Good morning. So we are absolutely delighted to welcome our American colleagues and friends. It's really wonderful that you are able to make this trip here to Paris and to share with us ideas, concerns, challenges, because all together we have one goal, which is to transform rare and genetic disorders. And we know that you have around 30 million in the US, we have 30 million in Europe. Each rare disease can be considered as sometimes just one in the world or two in the world. And so how do you establish economical models? How do you establish regulatory models that enable to actually treat these disorders that altogether encompass more than 300 million people around the world? And so we know that there's not just one type of approach. It has to encompass science. Should we think much more about a molecular way of classifying these disorders based on the therapeutic approaches we should be using? We need to think about the fact that these disorders are multiple. So how do you integrate common approaches for multiple disorders? And how can you apply, by having a different scientific point of view about rare diseases as a global entity, to actually transform the way the regulatory system and the economical models move ahead? And so we are in two different countries, two different continents, but I think there is a common big desire for transformation. And so here in France, we have, put our forces together with Frédéric Reva, who is from Genotea and also AFM, and Marina Cavazzana, who has been also key in implementing with Alain Fischer the first therapeutic trial for SCID that was able to reconstitute the immune system, and Romain, who is the bridge between the two countries, and Emily, who has really helped tremendously in the organization of that meeting. So I'm not going to keep on talking, because I think what's most important is how this day was organized, really thinking about those different aspects and working together to establish the program. And what we hope is that there will be a lot of exchanges, that we all need to be very open-minded, and that we learn from each other. So I wish you really a great day of science and also exchange. And I'll let some words for Romain.
Thank you very much. It's my great pleasure to introduce our speaker, David Liu. David has many hats. He is the founder of the Center for Therapeutic Genetics. He's a core institute member at the Broad Institute and the director of the Broad's Merkin Institute for Transformative Technology in Healthcare. He's an HHMI investigator. At Harvard, he's a professor of chemistry and chemical biology. And his lab has made a lot of contributions in the field of genome editing and protein evolution. A chemist by training, David and his group has made a numbers of landmark discoveries in the field of genome editing, including base editing, prime editing, but also in other fields such as PACE and DNA template synthesis. All these technologies being used by thousands of labs to study genetic disease and enable next-generation therapies. David's contribution have been recognized with many prizes and awards, including the 2025 Breakthrough Prize in the Life Sciences. He is an elected member of the US National Academy of Sciences and the US National Academy of Medicine. And truly, it's a pleasure to have you with us today, David, and a distinct privilege to welcome you to the stage. Thank you.
All right, it's a great pleasure being here. I'm grateful to Bana and Roman and everybody for organizing this very special symposium. And I remain in love with Paris, even though it's 37 degrees or more here. So today I'll describe three distinct strategies for therapeutic genome editing which is really performing precise chemistry on the genome of living cells in cultured cells, in animals, and ultimately in human patients to treat genetic diseases. So these three strategies provide examples of how genome editing technologies enable a variety of therapeutic approaches, some of which have already begun to have a substantial clinical impact. But I think they also demonstrate how scientific advances can help address some non-scientific bottlenecks, some practical and regulatory and economic challenges that we'll hear about throughout the symposium. These are often the current limitations on connecting the science of gene editing with patients. Okay, so the problem that unites, I think, everybody in this room is the one summarized here. More than 200,000 known mutations in our genome, more than 95% of which are small substitutions or insertions or deletions, cause more than 8,000 known genetic diseases that collectively afflict at least 400 million patients and their families. And so we developed base editors and prime editors to enable the first strategy that I'll talk about. This is the installation or correction of specific mutations in living systems. Base editors and prime editors don't exist in nature, at least we don't know of any examples in nature. They are instead engineered proteins that make specified changes in DNA without requiring double-stranded DNA breaks or donor DNA templates. We completed the development of the first base editor in 2016. This is the cytosine base editor, and it's a fusion of three different proteins: a catalytically impaired Cas9, which is shown in blue, which cannot cut the DNA double helix but retains its ability to bind a target DNA sequence in a guide RNA programmed manner. Second, natural and currently a laboratory evolved cytidine deaminase enzyme, which catalyzes the hydrolytic deamination of cytosine to uracil in single-stranded DNA. And third, this purple protein, a uracil glycosylase inhibitor, or UGI, which protects the uracil from removal by the cell. So in the presence of a target DNA sequence, the Cas9 domain of the cytosine base editor, the CBE, opens up the target DNA site and pairs one DNA strand with the guide RNA. That's what CRISPR systems naturally do. The unpaired DNA strand forms a single-stranded bubble within which the cytosines, within about canonically a 5-base window, are deaminated into uracil. That creates a UG mispair, and then the UGI domain protects the uracil in that UG mispair from being removed by the cell. So to coax the cell into replacing the G on the opposite strand with an A, so that this process doesn't happen, we designed the most efficient base editors to nick the G-containing strand, which we found will stimulate the cell into remaking that strand using the uracil-containing strand as a template. And so the result is conversion of a targeted CG base pair into a UA, or eventually a TA base pair. And so in our original report in 2016, we showed that if you simply fuse dead Cas9 with a cytidine deaminase, you end up with a very inefficient base editor, but we could still detect it. If you add the UGI domain, the purple protein, you observe improved editing efficiencies. And if you, in addition, add the nicking feature, creating what we call BE3, the third generation base editor, we saw a substantial improvement of editing efficiency. Okay, so by converting targeted CG base pairs to TA base pairs, these cytosine base editors can correct up to about 14% of known pathogenic point mutations, which are shown here in the blue wedge of this pie chart. But correcting the lion's share of disease-associated point mutations requires a different kind of chemistry, converting an AT base pair to a GC base pair. It's actually the converse of what the cytosine base editor achieves. So in 2017, we reported the development of an adenine base editor, which achieves this conversion. So adenine base editors, or ABEs, catalyze the hydrolytic deamination of adenosine, which yields inosine. And inosine is read as G during transcription or translation. So in principle, we initially thought this will be easy. We'll just do exactly what we did with the cytosine base editor. We'll replace the cytosine deaminase in BE3 with the deoxyadenosine deaminase, and then the ABE will open up the target DNA, deaminate A to I, will trick the cell into replacing the non-edited strand containing the T by nicking it, and that will result in the conversion of an AT base pair to a GC base pair. The main problem that we quickly encountered is that there is no natural enzyme that performs the necessary chemistry on DNA, that deaminates deoxyadenosine. And so to overcome this problem, we used laboratory evolution to evolve the first enzyme that deaminates deoxyadenosine. We created a Darwinian selection system that rewards deamination of adenine in DNA with survival in the presence of antibiotics. And then we made large libraries of mutant TAD-A tRNA deaminase genes. And after 7 consecutive generations of mutation, selection, and characterization in mammalian cells, we succeeded in evolving this enzyme, which we called ABE 7.10, which acquired the mutations shown on the right that together allowed this enzyme for the first time to deaminate DNA rather than tRNA, and also that importantly generalized its sequence compatibility to accept adenines in a wide variety of sequence contexts, rather than the UAC context strongly favored by wild-type TAD-A. And the result was the first editing agent capable of installing targeted AT to GC edits in human cells. So base editors are these engineered proteins that can precisely correct pathogenic transition mutations, A to G, G to A, C to T, or T to C, to install or correct the other types of mutations, the transversions, the insertions, the deletions. We develop prime editors. Prime editors are fusions of programmable nicases with engineered reverse transcriptases. They use an engineered prime editing guide RNA, shown in green here. We call this a PEG RNA for short, which not only specify the target site for editing, but also encode the desired edit. Prime editors nick the target DNA site, and then they use the 3' end of the freshly nicked DNA strand to prime reverse transcription of an extension on the PEG RNA, shown in green there, directly onto the end of the nicked DNA, target DNA strand. So the engineered reverse transcriptase then copies the desired edit directly onto that target DNA strand, which creates this precious 3' flap that contains the edit. The 3' flap is in an equilibrium with the 5' flap structure, and fortunately cells preferentially remove 5' flaps because they're very common intermediates in DNA replication and DNA repair. And therefore, eventually, this equilibrium becomes a heteroduplex of DNA with one edited strand and one unedited strand. And then the most efficient prime editing systems, just like our base editing systems, then nick the non-edited strand to cause the cell to remake that strand using the edited strand as a template, which completes the permanent editing of both DNA strands. And since the PEG RNA's reverse transcription template is specified by you, the researcher, you can make virtually any substitution, deletion, or insertion of up to maybe 100 or even 200 base pairs using this strategy. Since our initial prime editing report in 2019, our lab and many others have improved virtually every aspect of prime editing systems: protein architectures, codon usage, nuclear localization signals, PEG RNA stability, the evasion of cellular mismatch repair, which impedes prime editing, the continuous laboratory evolution of both reverse transcriptases and of Cas domains to increase prime editing efficiencies. 3 new developments we published just over the past 2 or 3 months include the use of AI protein redesign to improve the cellular stability of prime editors, resulting in these PE8 prime editors that have up to 163 amino acid substitutions compared to their PE6 progenitors. The directed evolution of new laboratory-evolved 3' PEG RNA motifs that enhance the lifetime of PEG RNAs in cells, and the development of OptiPrime. This is a machine learning model based on the mechanism of prime editing that generates high-performance PEG RNA sequences. These new advances were specifically designed to increase prime editing efficiency in primary cells and in animals, which are often more challenging settings for all types of gene editing. Okay, so prime editing and base editing have been used by many laboratories around the world to treat animal models of genetic disease. And in the interest of time, I'll just cover one story, which was co-led by a team that includes Alex Sousa, who's here at the symposium, in collaboration with the labs of Kat Lutz and Al George. This is an example of in vivo prime editing to rescue alternating hemiplegia of childhood, AHC. This is a severe neurodevelopmental disorder that presents with paralysis, involuntary movement, and seizures. There's no effective treatment known. We developed base editing or prime editing strategies that can correct with, in this case, about 40 to 90% efficiency, 5 mutations in ATP1A3 that collectively are responsible for about 70% of the ATP1A3 associated cases of AHC. Although 4 of the 5 mutations are transition mutations and in theory could be corrected by base editing, we ended up choosing prime editing to correct 3 of these 4 transition mutations due to the presence of non-silent bystander edits that resulted from base editing these alleles. We then used AAV9 to deliver in vivo optimized prime editing systems to the central nervous system of 2 AHC mouse models, yielding about 30 to 50% bulk brain cortex correction and about 50 to 90% correction among the transduced cells. When we looked at the correction at the mRNA level, it was even higher, suggesting that the prime editing occurs more efficiently in cells that express ATP1A3, which likely reflects the neurotropism of AAV9. Importantly, the treated animals showed some restoration of hippocampal ATPase activity of ATP1A3, the affected enzyme. And both of these different prime editor injected mouse models showed strong amelioration of stress-induced seizures, movement and cognition deficits, and dramatic extension of animal lifespan. And so this work suggests the feasibility of prime editing as a potential one-time treatment for AHC, and to our knowledge also represented the first use of prime editing to rescue a neurological disease in animals. And now we are actually advancing this work towards a clinical trial together with patient advocacy groups led by Nina Frost and others, physicians and scientists really across the world to try to use this case as what would end up being the first clinical prime editing in the central nervous system. AHC is one of many severe genetic diseases that have been successfully ameliorated in animal models using base editing or prime editing approaches that target a specific pathogenic mutation. 3 examples subsequent to using base editing or prime editing to correct AHC are actually shown on the right column of this slide, using base editing to rescue Zellweger spectrum disorder, Dravet syndrome, and prime editing to rescue phenylketonuria in animal models. Together, these and many other efforts from laboratories around the world, described in more than 3,000 publications to date that use base editing or prime editing, have provided the foundational data needed to advance these technologies into human clinical trials. And today there are at least 25 ongoing clinical trials. This number just increased twice over the past month that have begun using base editors or prime editors to treat a wide range of diseases, both ex vivo and in vivo. The majority of these trials are now in vivo. They include blood disorders, heart disease, metabolic disease, amyloid disorders, muscular dystrophy, a neurodevelopmental disorder, and more. 10 of these clinical trials, shown in green, have reported results. And happily, thus far, in all 10 cases, the base edit or the prime edit has been shown to benefit patients. And so it's really an honor to highlight some of the amazing clinical base editing and prime editing work of scientists and clinicians around the world. My lab does not directly run clinical trials, so all of the work I'm about to talk about is really a testament to the hard work and the courage of physicians and patients. This is Alyssa Tapley. She was a then 13-year-old girl in the UK with T-cell leukemia. She became the first patient treated with a base-edited therapeutic in 2022. Her T-cell leukemia was refractory to bone marrow transplant or chemotherapy. And so she was infused with CAR T cells containing 3 base edits in a trial led by Wasim Qasim at UCL and Ghosh. And following treatment with these triply base-edited CAR T cells, her T-cell leukemia went into complete remission. Now, more than 4 years after treatment, her cancer remains undetected. And she's back in school and actually hopes to pursue a career in biomedicine. I had the honor of meeting her for the first time in person last April. Brandon Baptiste, shown here, suffered severe pain crises. He had both of his hips replaced and acute chest syndrome, a very dangerous complication caused by blocked blood vessels in his lungs because he is a sickle cell disease patient. And so he received an autologous transplant of his own hematopoietic stem cells that were base edited in the fetal hemoglobin promoters to reawaken fetal hemoglobin expression in a trial led by Beam Therapeutics. And the result is that Brandon is now free of pain crises and acute chest syndrome, and indeed is no longer even receiving any medication for his sickle cell disease. In late 2023, clinical trial results were announced from the first in vivo base editing trial. This is a collaboration between Verve Therapeutics, now owned by Lilly, and Beam Therapeutics. Patients with very high levels of LDL cholesterol were given a single injection of an adenine base editor that was programmed to precisely disable a splice site in PCSK9, with the result that LDL cholesterol levels in these patients are durably, probably permanently lowered by up to about 60%. And so this clinical outcome is important in a number of ways. The first in vivo base editing trial, but it also demonstrates the potential of in vivo base editing not just to treat serious disease, but actually to lower future disease risk. In this case, the edit is not introduced into a pathogenic mutation. The edit is made into a wild-type gene that all of us have. One patient who received this base editor was Alice Thomas, a 64-year-old woman who had an LDL level of 190 and suffered 2 strokes. She was dosed in late March, just this past March, and 2 weeks later, her LDL levels are 50. In March of last year, one of these base editing trials achieved what I think is another milestone in medicine. This is the first correction of a pathogenic mutation in a patient. Scientists at Beam Therapeutics used an LNP-delivered adenine base editor to correct the most common cause of alpha-1 antitrypsin deficiency. This is a fairly common, as rare genetic diseases go, a fairly common serious liver and lung disease that in the US alone affects about 100,000 people. And as many of you saw last year, a team led by Kiran Musunuru and Becca Ahrens-Nicholas in Pennsylvania achieved another medical milestone. This is the first use of a bespoke gene editing therapy to treat a mutation found in a single patient. This is baby KJ Muldoon, who was born with no working copies of CPS1 and therefore suffered from a urea cycle disorder with a 50% infant mortality rate. He was compound heterozygous, inheriting different premature stop codons from mom and from dad. And in a remarkable testament to the dedication of this team and the regulators, as well as the translation capability of the science, an adenine base editor was developed in only about 6 months and dosed less than 7 months after diagnosis. This is a process that I would normally associate with maybe 7 years rather than 7 months. And the result is that baby KJ is now thriving. And here's a more recent picture. The baby is also unusually cute, so has managed to sort of become viral and is all over the news. FDA recently cleared the first clinical trial of a prime editing therapeutic. This actually occurred only 4.5 years after we published the first prime editing paper in October of 2019, just before the world shut down from the pandemic. So scientists at Prime Medicine developed an ex vivo prime editing treatment for chronic granulomatous disease, CGD. This is a debilitating and lifespan shortening disease that is caused by mutations in NADPH oxidase subunit genes. So a prime editing system that corrects one of the most common mutations, which is a 2 base pair deletion, the loss of a G followed by a T in a gene called NCF1, resulted in very efficient editing and then engraftment in mice of long-term hematopoietic stem cells from CGD patients. The treatment restored NADPH oxidase activity in cells derived from the edited HSCs, and genome-wide, transcriptome-wide, and targeted amplicon assays collectively uncovered no detected off-target edits. This is consistent with an emerging picture that the prime editing mechanism is inherently resistant to editing at off-target sites. This trial has yielded clinical results from the first 2 patients treated with a prime-edited therapeutic, one of which is Tracy Atterbury, shown here. So hematopoietic stem cells from these 2 patients were efficiently prime-edited, 60 to 90% correction, reinsertion of the missing GT into NCF1, and then transplanted back into the patients. Engraftment was very rapid, suggesting the edited cells were not left in a compromised state, and the neutrophils that repopulated the patient's bone marrows in both cases showed dramatic restoration of the missing NADPH oxidase activity, about 60 to 80% restoration. That's far higher than the 20% anticipated therapeutic threshold needed to restore immune system function. And you can see from this testimony from Tracy that he's already shown clinical benefit, and his brother unfortunately passed away from the same disease a day before his brother turned 3 years old. And Tracy speculates that if this therapy had been around when he grew up, he may have grown up with a sibling. All right, so a major challenge for genetic medicine, despite all of these results that I just showed you, is the rarity and the allelic diversity of most genetic disorders. So I showed you that base editing and prime editing can be targeted towards the correction of specific mutations, in some cases with substantial clinical benefit, but it is currently not practical or economically viable to develop thousands of different gene editing drugs, despite this promise, needed to correct even a modest fraction of known pathogenic alleles. So to help address this red part of the graph, there are many possible solutions, and we'll hear later from Winston and from Tim about the creation of some, I'll say, systemic catalysts to help address this problem. But there are also some scientific ways that you might think about addressing this problem. And so our lab has been working on the development of ways to edit DNA that would allow a single treatment to benefit more patients than those that have a specific rare mutation. So that brings me to the second approach that I'll talk about, which is an approach very familiar to this audience because it was first addressed with clinical success by Alain and Marina 30 years ago. And that problem is that in many cases, these genetic diseases are loss-of-function diseases caused by hundreds or even thousands of different mutations in the same gene. If you introduce a healthy copy of the gene into cells, again, this idea was first reduced clinically successfully by Alain and Marina and others with the development of SCID gene therapies. In theory, you might benefit any patient suffering from a loss-of-function genetic disease regardless of that patient's specific mutation. The modern twist on this idea, what I sort of sometimes colloquially call gene therapy 2.0, because it really builds on the foundation of the pioneers, is to introduce the healthy gene copy at a targeted location in the genome of the patient, such as its endogenous genomic locus, so that we can preserve as much of the endogenous regulatory context as possible, or perhaps at a safe harbor locus where its expression is less likely to interfere with other genes. So one approach to achieving this goal of targeted gene size insertion in human cells is to use prime editing to install a recombinase landing site, like a BXB1 recombinase landing site, at a target DNA locus, and then to use a BXB1 recombinase variant that we evolved in our laboratory using the PACE system to integrate with high efficiency a cargo gene into the prime edited site. And so recently we reported the use of this approach, which we call PASSAGE, to efficiently install cDNAs into therapeutically relevant targeted genomic sites in human and mouse cells. A second method to achieve targeted gene integration that we've worked on is the result of a 3-year collaboration with Sam Sternberg's lab in Columbia to evolve CRISPR-associated transposases, or CASTs, to work efficiently in mammalian cells. So the type 1F casts shown in this slide they use a cascade complex, so a type 1 CRISPR complex of Cas 6, 7, and 8 to engage a target DNA sequence in a guide RNA programmed manner. The TNIQ and the TNSC proteins then connect the cascade-bound target DNA with a heterodimeric TNSA, TNSB transposase. The transposase then mediates the integration of donor DNA, which can be kilobases or even tens of kilobases long. And the integration occurs rather precisely, about 49 base pairs downstream of the target site. And because this integration occurs in a single process without free genomic double-stranded breaks, Cas do not generate genomic indel byproducts. And in bacterial cells, these type 1 Cas are almost unbelievably perfect in their properties. They're extremely efficient, very specific, and yield minimal byproducts. Unfortunately, in mammalian cells, all the naturally occurring Cas that have been reported to date show very low activity. Sam's lab did a screen to identify the most active of these type 1 Cas homologs that show detectable activity in mammalian cells, and you can see the most active of these might support still 0.1% targeted gene integration into the genome of mammalian cells. We speculate that unlike the CRISPR-Cas9, for example, these bacterial immune systems, transposases probably never evolved to have high activity because there's no real fitness advantage to high activity transposition, and the possibility is that you incur a host cell fitness penalty. So integration in human cells, we speculated, might be limited by transposase activity or by DNA engagement. And since at the time the molecular determinants of these Cas properties weren't known, we thought the challenge of developing Cas that can function robustly in human cells is ideally suited to protein evolution. And so this is a nice example of how we apply our phage-assisted continuous evolution, or PACE system, to try to overcome a challenging, complicated problem in molecular protein science. This is a work that was initiated by former graduate student Shannon Miller and co-led by Isaac and Simon in our lab and by George in Sam Sternberg's lab. They used PACE to evolve Cas through thousands of generations of mutation, selection, and replication. That's really the special benefit of PACE, is that we can perform dozens of generations of evolution, which would normally take dozens of weeks. We can perform dozens of generations of evolution in 24 hours. So as a result, once you develop different PACE selections that can link phage propagation to the activity of interest, in this case, to Cas activity, and in this case, we integrate a promoter in front of an otherwise promoterless gene 3. And so that enables gene 3 expression, which is essential for phage propagation, to be linked to the activity of a CRISPR-associated transposase encoded by that phage. So then you can just drop the wild-type Cas gene into what we call a lagoon, and the system will continuously evolve that gene, dozens of generations per 24 hours, until you characterize the resulting gene variants. And so we use several different PACE selection circuits to evolve a wild-type type 1F Cas. I don't have time in this lecture to cover all of the findings, but I'll just say that the PACE campaign that yielded the most improved Cas variants is shown here. And while we observed mutations in all of the different Cas components, most of the key changes evolved in TNSB, which is the catalytic subunit of the transposase. So evolved TNSB variants, such as the one shown here, P4-15, survived hundreds of different generations of mutation, selection, and replication, which again, using traditional laboratory evolution methods, would have taken decades. numerous mutations in all of the cast components we noted, but the ones that contributed the most to the new activity, again, were in TNSB and occurred typically at the interface of this catalytic subunit of the transposase and other components of the cast complex. And reversion analysis, which I'm not showing here, showed that all of these mutations actually contribute to improved cast activity. So how do they perform in mammalian cells now? While wild-type Cas mediated, as I showed you before, less than 0.1% genomic DNA integration in HEC293 T cells, the paste-evolved Cas mediated guide RNA programmed DNA integration more than 200-fold more efficiently. And so the variant that I showed you in the last slide could integrate test cargo into 3 genomic sites in human cells with a typical efficiency of maybe 10 to 20% without any enrichment for transfected or edited cells. We speculated that based on the location of these mutations that were responsible for this benefit, that PACE probably enhanced multiple functions in TNSB. And indeed, we identified at least 2 of these enhanced functions. On the left, that PACE improved transposase binding of the transposon ends in human cells. And then on the right, PACE also improved the integration kinetics of evolved transposases, at least as assayed in E. coli cells. To determine if these evolved Cas can function efficiently in therapeutically relevant applications, we tested their ability to integrate donor DNA relevant to the treatment of genetic diseases into about a dozen genomic sites of clinical interest, including 4 safe harbor loci, the TRAC locus for CAR-T engineering, and 7 genomic loci where loss-of-function mutations cause Stargardt disease, cystic fibrosis, Fanconi anemia, sickle cell disease, beta thalassemia, SCID, Rett syndrome, or phenylketonuria. And once again, we generally observe DNA integration efficiencies in HEC293 T cells of about 10 to 30%, in this case with an average efficiency of 13% at these therapeutically relevant loci, and an average improvement compared to wild-type Cas of more than 350-fold. We anticipate that the efficiency of EvoCast can still be further improved, and we have a manuscript we just submitted on optimizing some of the nucleic acid components involved in EvoCast. But these integration efficiencies, if they could be translated into a clinical setting, which I admit is a very tall order, are already sufficient to offer potential benefit to patients suffering from a variety of loss-of-function genetic diseases. We extensively characterized integration products and found that the engineered and evolved Cas systems retain many of the favorable attributes that drew us to the original type 1F Cas systems in the first place. They're quite regioselective for cargo installation, again, predominantly occurring 49 base pairs downstream of the target sequence. The EvoCast integration is also predominantly unidirectional, with over 90% of integration events occurring in the single orientation. And consistent with the fact that the integration proceeds without formation of genomic double-stranded breaks, we observed no significant indel formation at the target sites. And so together, these results show that you can evolve and engineer Cas to show quite substantial activity in mammalian cells without sacrificing some of the key features of naturally occurring type 1F CRISPR-associated transposases. Now I think one of the outstanding challenges in the targeted gene integration field is to develop ways of delivering or producing the donor double-stranded DNA template inside of cells in a manner that is maximally compatible and minimally provocative of, for example, the C-gastine cellular immunity pathways. Okay, so I'll end with a third therapeutic gene editing strategy that's designed to maximize applicability to as many patients as possible by enabling a single composition of matter to rescue multiple unrelated genetic diseases. We have a scientific retreat, an offsite every year for our lab. And about 3.5 years ago, as an example of what we call science karaoke, where everybody has to present a new idea for 10 minutes that our lab hasn't worked on, I gave this idea as an example. We call this concept disease-agnostic therapeutic editing, and it sounds a little bit crazy, so I'm grateful to Sarah, Stephen, and Kita in our lab for leading this effort. So premature stop codons are a cause of about 30% of genetic diseases. Baby KJ suffered from 2 premature stop codons, for example. And about 1/4 of all known pathogenic alleles in ClinVar are nonsense mutations. Suppressor tRNAs, which are, of course, tRNAs that have an anticodon that complements one of the 3 stop codons, can rescue nonsense mutations. But suppressor tRNAs have proven challenging to develop into human medicines because of the need to provide them for the lifetime of the patient, as well as their modest potency. If you only change the anticodon of a tRNA, you tend to end up with a fairly modestly potent suppressor. And that has caused people to overexpress the tRNAs, the suppressor tRNAs. And tRNA overexpression can lead to global perturbation of translation. That said, suppressor tRNAs are generally well tolerated in eukaryotes for a variety of reasons, if you don't overexpress them. Cells evolved multiple safeguards to distinguish premature stop codons from natural stop codons and to minimize the read-through of natural stop codons, because all of you are probably wondering what happens to the natural stop codons that terminate the translation of all of our endogenous open reading frames. So first, the representation of premature stop codons versus natural stop codons differs statistically. For example, TAG is the most common premature stop codon, but the least common natural stop codon. Second, natural stop codons are often clustered, and all of those redundant stop codons would have to be suppressed to lead to read-through of the natural stop codon. Third, cells have evolved release factors that terminate translation very potently and can outcompete suppressor tRNAs. These release factors are typically recruited to the 3' UTR, which is near natural stop codons by definition, but is not typically close to premature stop codons. And fourth, there are non-stop decay pathways that degrade proteins that are translated into their 3' UTRs. And even the way our genetic code is laid out results in many 3' UTRs, if translated, generating degron sequences that stimulate rapid degradation of those proteins. Importantly, there's human genetic data to indicate that your tRNA genes are maybe more redundant and dispensable than one would think. First, you have 418 high-confidence tRNA genes, and you certainly don't need that many. There are apparently healthy individuals that are missing entire tRNA genes, or that have tRNA genes that are mutated in their anticodon loops to become suppressor tRNAs, or even miscoded, mischarging tRNAs, miscoding tRNAs. Yet they appear to be healthy in some cases. So our idea is to take advantage of prime editing's search and replace capability to convert a dispensable endogenous tRNA gene into a suppressor tRNA permanently. If this tRNA conversion can be done safely and efficaciously, now a single prime editor, a single composition of matter, might provide a one-time treatment for multiple different genetic diseases that are all caused by the same flavor of premature stop codon. And now the math starts to look more favorable. There are thousands of patients that suffer from premature stop codons that cause different genetic diseases like cystic fibrosis and Stargardt disease and phenylketonuria and muscular dystrophy that in principle could benefit from a single prime editing drug, or maybe a small collection of prime editing drugs that can unify patient cohorts both within a disease and even perhaps among different diseases. And so we began this effort by using prime editing and 17,000 pegRNAs as a tool, as a research tool, to convert each of your 418 endogenous human tRNA genes into all 3 types of suppressor tRNAs by replacing their anticodons with either CUA, UCA, or UUA. We performed this editing using a lentiviral library, and we assessed nonsense suppression activity by the ability to read through a premature stop codon in GFP. This screen identified several families of tRNAs that can serve as suppressors upon simple anticodon-only replacement. But consistent with what I just told you, the best of these anticodon-only suppressor tRNAs, when installed at native genomic tRNA sites, so just single genomic copy, showed low potency, supporting perhaps 5% of the amount of full-length GFP from a positive control GFP that lacked any premature stop codon. So to identify improved suppressor tRNAs, we generated tens of thousands of variants of the best performing suppressors in the last slide, including every possible paired and unpaired nucleotide substitution, every possible single base pair deletion, as well as many natural and synthetic leader and terminator sequences, and repeated the GFP nonsense suppression screen on these lentiviral libraries. We learned many insights into the determinants of tRNA function, such as the importance of matching endogenous leader sequences with their cognate tRNAs. But in the interest of time, I'll just focus on the discovery of the suppressor tRNA variants shown here, which showed a dramatic increase in suppression activity, such that now a single endogenous tRNA converted with prime editing into the best of these super suppressors could yield 35% of wild-type GFP protein levels. This level of suppression efficiency from a single genomic tRNA gene was exciting to us because it raised the possibility of rescuing potentially different loss-of-function genetic diseases caused by nonsense mutations without having to dramatically overexpress a suppressor tRNA or dose a suppressor tRNA for the rest of the patient's life. After identifying a highly active engineered human suppressor tRNA, we next optimized prime editing systems to efficiently convert an endogenous human leucine tRNA that is redundant. You have 4 copies of this particular leucine tRNA, and there are other, of course, leucine tRNA genes in your genome, into this optimized suppressor. And after exploring pegRNA and prime editor choices, we achieved about 80% prime editing efficiency to install the optimized suppressor tRNA into these 6 different human cell lines. These are cell models of Batten disease, Tay-Sachs disease, and Niemann-Pick disease that all arise from 6 different premature stop codons in 3 different genes. In the first 4 of these human cell models, the nonsense mutations that cause Batten disease and Tay-Sachs disease, a single prime editor and pegRNA restored about 20 to 70% of the wild-type enzyme activity levels. The NPC1 gene that causes Niemann-Pick disease type C1 encodes a cholesterol transporter rather than an enzyme, so it's a bit harder to assay, but we could still observe substantial rescue of full-length protein production upon treatment with the same prime editor. So this slide is an example of one prime editing agent that can partially rescue, at least at the molecular level, 6 different premature stop codons in 3 unrelated genes that cause 3 distinct genetic diseases. We can quantitate the relative abundance of our prime-edited installed suppressor tRNAs versus the wild-type tRNA at the same genomic locus that we replaced. And it turns out that the optimized suppressor tRNA is not overexpressed compared to the tRNA, the leucine tRNA that we replaced. Indeed, it's only expressed about 40% of the normal level of that endogenous leucine tRNA. And since a known consequence of tRNA overexpression, as I mentioned, is global perturbation of translation, we were encouraged by the possibility that we could achieve potent suppression without overexpression because it might suggest a higher prospect for safety. And so one of the ways we assessed perturbation of the cell in ways that were undesired was to look for, first, perturbation of transcripts that differ more than 2-fold in expression between hex cells before or after prime-edited mediated conversion of the tRNA. We call this PERT, prime-editing mediated read-through of premature termination codons, PERT. And we did not observe any transcripts that differed more than 2-fold. We also performed whole proteome mass spectrometry on trypsin-digested protein from hex cells with a premature TAG stop codon in GFP before or after PERT, and the only protein that was significantly different in abundance was full-length GFP. So by transcriptome and proteome changes, PERT, again, perhaps because we're not overexpressing this suppressor tRNA, was not highly perturbative. We then explicitly looked for peptides corresponding to read-through of any of the 4,036 natural human proteins terminated with the TAG stop codon. While we observed robust read-through of the premature stop codon in GFP, resulting in the detection of several peptides after the premature TAG stop codon in GFP, we did not detect any peptides consistent with translation past the natural TAG stop codon for any other protein. It's hard to interpret a negative result, and we were surprised by the outcome. So we increased the sensitivity of this experiment by performing a target ion peptide mass spec of predicted peptides from the 69 most abundant human proteins that use a TAG natural stop codon. So you just have the mass spec sit on the ion of the peptide that you expect, and you count how many ion counts. And once again, the only peptides we observed in these experiments were those corresponding to read-through of the premature GFP stop codon. So these data suggest that, at least under the tested conditions, PERT does not induce significant natural termination codon read-through, probably due to the mechanisms that I already mentioned by which cells distinguish premature stop codons from natural termination codons. And I also think the short lifetime of the resulting proteins that do get suppressed through their natural termination codons is probably a factor here as well. Finally, we tested the system in vivo in animals. Our first disappointing discovery is that mouse tRNA genes are different from human tRNA genes. So we had to redo everything I just presented for mouse tRNA genes. Then we injected AAVs encoding a prime editor targeting a mouse tRNA, converting it into a suppressor tRNA into reporter mice that contained first a premature stop codon in GFP, and that resulted in about 25% of wild-type GFP levels among transduced tissues. And then we used the same prime editor, so the same AAV, to inject a mouse model of Hurler syndrome. So this is a severe lysosomal storage disease caused by a premature stop codon in a gene called IDUA. We observed about 7% prime editing and about the same amount of IDUA enzyme activity compared to wild-type mice. But as is the case with many loss-of-function genetic diseases, this amount of wild-type enzyme activity should be sufficient to rescue the disease. And indeed, by a variety of histological and phenotypic analyses on multiple tissues, we observed complete or near-complete rescue of Hurler syndrome pathology in these mice, including foam cell accumulation, vacuolization, and glycosaminoglycan accumulation. All right, and then the last slide before I summarize is just really a teaser for what Winston will present later on in the symposium, which is really the culmination of our recognition that the status quo with for-profit industry leading most drug development efforts will not bring this science anytime soon to the vast majority of the patients who might benefit from it. So we've started a nonprofit effort called the Center for Therapeutic Genetics, which is a partnership between the Broad Institute, Boston Children's Hospital, and Jackson Laboratory Rare Disease Translational Center. Representatives of CTG are sprinkled throughout the audience, and we'll talk much more about CTG later. Okay, so to summarize, base editing and prime editing are engineered molecular machines that enable mutation-specific therapeutic gene correction and are in at least 25 clinical trials. I updated this just a few days ago and forgot to update the summary, with some positive patient outcomes. Targeted gene integration in human cells, really sort of the next generation legacy of what Alain and Marina created 30 years ago. You can perform this in human cells with prime editing plus recombinases or using evolved CRISPR-associated transposases, although there's still substantial work needed to connect targeted gene integration this way into clinical use. And then finally, I showed you an example of how you can use prime editing to convert endogenous tRNAs into engineered suppressor tRNAs to allow disease-agnostic rescue of pathogenic nonsense mutations in human cells or even in mice with a single composition of matter. And so that leaves me just to thank a really remarkably talented group of graduate students, postdocs, undergraduates, and especially our collaborators shown in yellow that made all of this work possible. Base editing and prime editing's developments were originally led by Alexis Comor, Nicole Godelli, and Andrew Anzalone, but many, many people in our lab, many collaborators shown on the left, and indeed many people throughout other labs in the world have contributed to their development as well. The use of prime editing plus recombinases in evolved recombinase was pioneered by Simriti and Daniel in our lab in collaboration with the other scientists shown there. The EvoCast work, the evolved CRISPR-associated transposase, as I mentioned, was a collaboration with Sam Sternberg's lab, co-led by Isaac, George, and Simon. And the PERT project that I ended with was co-led by Sarah and Stephen, together with the contributions from Mark Osborne's lab. I'm very grateful to these agencies and others for funding, and I'm grateful for your attention in this beautiful symposium and city, and so thank you very much. I don't know if there's time for questions or if...
Yeah,
okay.
There is time. Thank you very much for the very impressive talk. Any question in the audience?
Thank you, David, for this fantastic review of all of your work over the years. You showed us examples of efficacy in different tissues, different cells with the different tools you have developed. But still, are there, as a function of cell type, as a function of the fact that cells are cycling or non-cycling, stem cells, not stem cells, etc., are there differences of sensitivities of different cells and tissues to different tools developed?
Yeah, so I think base editing and prime editing have now been shown to not require cell cycling, in that at least in non-cycling cells in a dish like terminally differentiated neurons, you can achieve both types of editing. You know, of course, clinical editing, there can be surprises. So we have not yet achieved clinical readout of base editing or prime editing in the brain, for example. That's actually something that our new nonprofit center is trying to establish. So I'll say that the cell work and the animal work is suggestive that base editing and prime editing are compatible with a broad set of cells. I'm always hesitant to say that means absolutely all cell types until we really have more data, and especially clinical data. But we do know that in contrast with nuclease-mediated homology-directed repair, for example, which definitely requires certain cellular DNA repair processes that are only expressed during certain stages of the cell cycle, that these technologies do not require that. And they have been used in cells in vitro and in animals, spanning a pretty wide range of cell types, including some terminally differentiated post-mitotic cells.
Any other question?
Similar line of question. When you show, for instance, that PERT is only providing 7 or 8% of correction in the cells, are you limited by the amount of tools that actually reach the nucleus? So could you give more, several times, can repeat? Or are you actually facing also perhaps restriction factors from the cells, or are you lacking?
Yeah, it's a great question. You know, there are many transmission coefficients that all get multiplied, and unfortunately they're all less than 1 in order to get to the final outcome. In the case of that mouse experiment that resulted in about 7% prime editing, it's a combination, I think, of AAV delivery, the editing efficiency itself. You can redose editing agents, but not in the clinic for high titer AAV, because when you give a patient a high dose of AAV, you usually provoke high titers of neutralizing antibodies against that AAV serotype. So AAVs are currently thought to sort of offer one big opportunity for patients. There are immunosuppression regimens, but I don't know if anybody has really overcome with positive clinical data repeat dosing of high titer AAVs. However, people have repeatedly dosed LMP editing agents. You saw a picture of one of them because baby KJ was actually dosed 3 times. First, because it was a baby, just a dose that we didn't even anticipate would be therapeutic, but turned out to be slightly therapeutic. And then a second dose when that first dose showed benefit measured by a decrease in blood ammonia levels, dosed a second time. And then finally a third time with a substantially larger dose, which brought blood ammonia levels in baby KJ down to normalish levels. And that was the limit of how many doses FDA cleared for that emergency N of 1 trial. So that was the end of the dosing regimen. But you can redose the editing agents. You know, there are some long-term questions about especially durable expression of gene editing agents, whether they might provoke immune responses that in the worst case scenario might cause elimination, the immune system to go after the edited cells. We don't know really the answers to those questions yet. For sure, the I think the preferred way to perform gene editing in patients is to transiently express the editing agent. That doesn't necessarily mean non-viral only, because there are methods in development now to transiently express the editing agents, even when taking advantage of the amazing tropism of certain viruses to deliver these editing agents. So something our lab and others are currently working on.
We'll take a last question.
Amazing talk. Thank you very much, David.
Thank you, Marina.
To come back to the hematopoietic system, have you an idea of the level of polyclonality of with your different tools? Are few clones that expand, that are base edited, prime edited, or you have proven that there is a polyclonal engraftment in your animal studies?
Yes, so in the ex vivo base editing and prime editing studies, there have been measurements of how many different non-edited versus monoallelic versus biallelically edited hematopoietic stem cells there are before and after engraftment. I'd say overall, there's no obvious bias. It depends on the disease, of course. In some cases, there's selection based on the disease. But the cases, for example, editing fetal hemoglobin in hematopoietic stem cells, or even correcting the HBB mutation, that is the sickle mutation, back to wild type using prime editing, we didn't observe any obvious bias before or after engraftment in the allelic distribution. And I'd say the non-edited versus monoallelically edited versus the biallelically edited distribution also follows roughly the total potency. Of course, if you increase the potency of the editing, you get more biallelically edited. But it's not as though we observed an obvious bias for or against, homozygous or heterozygous edited cells.
Yes, but in this way you take the assumption that the diversity of stem cells is spread when you can have biallelic deletion or correction and have a multitude of different hematopoietic clones.
No,
I don't think that.
Oh, you're saying, could you end up with a chip-like situation from selective expansion? I don't think we have data to really comment on that or not. Yes.
But I think it could be very interesting to know.
Yes. Yeah. No, I agree. Thank you for pointing that out.
So thank you very much. I think we can all thank again Professor David Liu.
Thank you.
So now I'm very happy to welcome to the stage Dr. Romain Lévy. So Romain is Associate Professor of Immunology at Paris Cité University. He's a clinician, the Pediatric Immunology and Hematology Department at Necker Hospital, and he's affiliated researcher in the French-American lab Human Genetics of Infectious Diseases here at Imagine. So after completing his residency in adult and pediatric immunology, he earned his PhD in 2017 in the same lab, where his work used next-generation sequencing and molecular genetics to discover and characterize new genetic causes of immune defect. So since 2017, alongside his continued research into the mechanisms of rare diseases, he has practiced as a clinician in the bone marrow transplant unit of Necker's Pediatric Immunology Department, caring for patients with rare immunological conditions. And he's here now because since 2024, Romain he joined David Liu's lab as a postdoctoral researcher, where he works on gene editing strategies for rare immunological disease. Romain, the floor is yours.
Thank you. Thank you very much for the introduction. How do I pass the slides here? Sorry. Do you know how I pass the slides? Did you have just this?
I use my computer, so.
Uh-huh. Do we have a way to pass the slides? Is it this?
Press the green button.
Yeah, let's see. No? Yeah. That's the green button.
Do you have a piece of
I
chalk
even
instead?
No, I even tested the red one.
Red usually goes back.
Do you know how? Oh, no, it's working. Thank you. Yeah.
Is that
Just,
working
yes,
now, or is
yes.
that
No, it's just I got the sign there. Thank you. Well, thank you very much for the invitation to give a talk on therapeutic genome editing in the clinic. We just heard about a series of genome editing tools, current ones already implemented in the clinic, and next generation ones, such as BERT, CAST, and other tools that are in the works. And we heard about their precision and the demonstration of their efficacy in a number of diseases led by academic or industrial partners. These strategies come in addition to other ones that are already implemented in the clinic for the past decade in gene therapy or ASOs. And today we'll have an overall discussion across talks and panels across different fields, discussing the how and how to leverage the full potential of these approaches and how to implement them and bridge them to the clinic where patients actually need them when the science is mature and when the medical rationale is strong already. It's a very timely discussion as there is no a roadmap on how to do that, at least like a roadmap that would be applicable to any territory or any model. So this is what I want to cover during that talk. I will talk about genetic disease and architecture, and it's probably information that many of the room know about. I will discuss about a trajectory in concept from disease target identification to a treatment in a patient or a group of patients with a severe disease in the clinic. I will discuss about this expanding toolbox we just heard about in details of programmable medicine, of a gene editing solution that are at disposal now that made their demonstration in patients actually. What are the unknowns that we discussed about a little bit with David in the field, and the next horizon, the frontiers, where actually a lot of work and innovation is done already. About positioning, and I will stop about the case of blood diseases, because I think, although it's also my background, it's also a practical case of discussion of interest. I will take a disease case example of something that started here at Necker, crossing the path of a patient, became actually a scientific effort carried out in the lab, and to frame perhaps overall the discussion we'll have about implementation of these strategies. I will say a word of N of 1. It's a term that we will hear about many times during the day, and I will finally conclude with some remarks. So genetic diseases are frequent globally. They affect about 30 million in Europe, about the same numbers in the US. There are about 7,000 genetic diseases that have been reported to date, and this varies across databases. Importantly, 85% of those are considered ultra-rare, as we just heard also. They affect children in most majority of the case, and they are severe. In 3 out of 10 cases, these children won't live beyond the age of 5. And there's no treatment in about 95% of the case. Things that we can say about these numbers, again, that many of you know about, is that this is not a static picture. The map of genetic disease is expanding with steadily across the years discovery of new gene defect across all medical fields, mostly thanks to the wide use now of next generation sequencing that was actually done in academic labs 10, 15 years ago, well funded, to now routinely prescribed in the clinic in some hospitals like this one, where whole genome sequencing are prescribed when you suspect a genetic disease. In the field of inborn errors of immunity of blood disease itself, the number of genetic disease was multiplied by 3 in about 15 years. And importantly, and about our discussion, in 20% of these genetic defects were reported in unique patients. The second point is that as this map of genetic disease expanding, in parallel, our understanding of the underlying biology explaining this disease is actually getting more granular, more comprehensive, which is definitely a prerequisite when we want to discuss about precision medicine, such as genome editing. Despite this discovery, the path for development of such program for precision medicine, gene editing or ASOs or gene therapy is challenging. It's challenging for a number of reasons that will be discussed across the day, and this list is not exhaustive, but in the very nature of this genetic disease, their medical and genetic heterogeneity is a challenge, with population size that actually range from 1 to a couple hundred globally. In that space, as I said, ultra-rare or unique patients are more the rule than the exception. We lack natural history data in many cases, with new diseases being reported. We lack biomarkers or surrogate markers to design trials and assess efficacy. The timelines for the development of precision medicine and its approval versus the therapeutic window we have or a patient has is sometimes conflicting. We heard about baby KJ, and the efforts that was made to develop a drug in about 6 months. And now my slides are becoming smaller. That's fine. Complex manufacturing also is an issue. Finally, something that is important is the fragmentation of medical expertise with, again, this very rare disease followed by experts across the globe, and the fragmentation in the regulation landscape that is also something we'll discuss about. All these reasons, in addition to many other ones I may have forgot, explain why most of these programs are commercially not viable. So what we would like to achieve as clinicians, as scientists in the field of rare disease is to go from disease target to development of a drug in a given patient or a group of patients by first identifying an unmet medical need, which is something we do every day in the clinic, by simply looking at the alternative treatments available and the expected efficiency of these treatments for a given patient or group of patients. Having a biological rationale is very strong, is very important, meaning causal relationship between a given genetic trait and a list of symptoms or disease, which is sometimes the focus of labs for years to establish causal relationship between a given genotype and a given phenotype. Having some idea of the window of intervention is actually key, whether you have hours or years, and evolving in a development or an ecosystem where you can foresee a path forward to development of a cure, meaning from the designing of a strategy, a gene editing strategy or nasal strategy or any else strategy, to the delivery to the right cell or the right tissue, and evolving in a regulatory landscape when you can actually have an approval or at least permissive to such efforts. And this is not a straight line. It will require some science done in the lab to basically make the demonstration that you can actually correct the mutation and you can deliver it by optimization in cells, which is the first brick of the work, safety assessment, which we call off-target in the field of genome editing, animal disease modeling with mice or primates, and other inherent considerations for any drug development in terms of toxicity studies, manufacturing consideration, regulatory considerations, to end up with a treatment and treat in time a patient and be able to repeat the procedure for another group of patients or another patient. And this is obviously very challenging. So what are the tools that we have at our disposal now? Of course, viral gene addition, gene therapy, have been here about more than 20 years with remarkable success with different platforms such as AAV, lentiviral vectors, retroviral vectors in a series of diseases such as the CNS disease or blood diseases. These approaches, as David mentioned, are beautiful in the way that they are mutation agnostic, meaning that with a single treatment, you can actually address patients with the same disease but different mutations. We have about 20 years of clinical trials and safety data in that space. And so we know the safety profile and we know their pros and cons. The considerations for this strategy conceptually is that they are limiting to the type of mutation and mechanism of disease that actually explain the disease, limited to mutation causing loss of function mutation or absent protein. The gene expression is also regulated by artificial promoters, which in some cases can be of concern, not always. The transgene size is limited by these vectors, but it's true for any delivery modalities. And depending on the platform we consider, they have specific constraints or considerations such as immunogenicity in the field of AAV mostly, or insertional mutagenesis. And again, on these two topics, some talks can be done and nuanced depending on the type of vectors we consider, type of promoters done. And we have genome editing. So genome editing are, as you learn and listen to, are a set of programmable and modular set of gene editors. And this is an expanding field with new type of tools that are actually now available in the clinic or preclinical models. The consideration that are shared by these models is that these are newest platforms, of course. There is established clinical efficacy demonstrated for some organs, such as the liver, the blood, the eye, and the knowledge for their development is not generalized. Gene editing can be split in two kinds, I would say. Cas9 nucleases on the left, that are these molecular scissors, the first generation of gene editors that can actually are very good at going to a specific place and disrupt a gene, abolish gene function. And they are the basis of one FDA-approved drug for sickle cell disease, and ongoing programs are underway for disease for which the purpose of any of these strategies is always to disrupt gene expression. And on the right, we have these precision gene editors that we heard about, base editing and prime editing, but also other strategies that are endowed with rewriting properties to not only be specific at the locus, but enable molecular correction or versatile molecular correction of any kind almost of mutations. Importantly, these strategies preserve natural gene regulation, which again, in some cases, is actually key if you want to provide clinical benefit. So what works in the clinic, and really to summarize what is actually already demonstrated to work in humans right now, is to deliver base editors to the liver in vivo works. We heard the story of baby KG as a bespoke base editor, but industrial programs have also shown that it's something feasible. Ex vivo base editing and prime editing for blood cells, hematopoietic stem cells, and CAR T cells have been done with success. And these platforms rely on mRNA and its delivery, either ex vivo or with LNP formulas. One of the major bottlenecks remaining is, of course, to deliver these gene editors beyond the liver in vivo to organs of interest, such as the brain, the stem cells, the lung, and so forth. And on what's possible in 2036 and on the delivery bottleneck and other challenges that remain in 2026, I just want to comment on the progress made, on the pace of innovation made to solve these problems. Among the next frontiers are the ways of people are trying to solve this problem. Of course, the delivery problem is one of the area where a lot of progress and creative ideas have been actually emerging. The idea of being able to deliver a one-shot injection of a base editor, prime editor, or another technologies to the liver of interest, go to the target cells and correct the mutation, has been an effort with AAV engineering in that space, strategies to increase packaging limits, which is also an issue for genome editors, brain coursing capsids for delivery in the brain, immunogenicity concerns or work towards the problem of immunogenicity of AAVs. In the field of LNPs, colleagues have been working in academia or industry with different LNP formulas or targeted LNPs to deliver cargoes beyond the liver to HSCs, hematopoietic stem cells, or T cells, or even in the lung. And also some other strategies of delivery, such as viral-like particles to deliver to HSCs, T cells, or the muscle. Another frontier we heard about is the editors themselves. With new editors or new generation, we heard about PE8 as the next PE strategy, getting smaller, higher in their efficiency or precision. Another frontier is multiplex editing, the ability to target multiple loci in a given gene or different genes to provide benefit and tackle some polygenic traits. Targeted gene size integration, we heard about CAST and other strategies out there to mediate targeted gene integration at the locus. And of course, agnostic approach, more creative ways to target different disease, different mutation with a given single composition of matter. So discussing about all these strategies in this tool set as a clinician, and it's an ongoing discussion I had with my colleagues from the Department of Immunology and Hematology, an interesting discussion about the positioning of these strategies in particular the field of blood disease. I couldn't comment on other fields of medicine. This is a field of interest because these tissue, the bone marrow, actually can be replaced entirely with a procedure called bone marrow transplantation that has been actually used for decades now with remarkable progress in the way we manage these patients, thanks to the management of infectious complications, chemotherapy regimen, dosing, and many other improvements in that field. You can also take the cells ex vivo out of the host in the body and manipulate these cells with gene therapy products or gene editing, and also provide benefit to the patient in the clinic. This is, on the left, all the immune defects or blood disease that are reported, and of course it's probably not up to date. As I said, there is close to 600 immune defects reported in that space. And this is a single chart, and you can go into more granular talks about the survival rates and the EFS survival in that field of bone marrow transplantation for inborn errors of immunity. But as you can see, these strategies work in the clinic. They save lives every day. Although these are very expert procedures that are performed in very expert centers, such as this hospital or academic hospitals across the globe. And you still have bottlenecks that you need to fix, graft-versus-host disease, of course, graft failure, and other complications. And so there is a need for other alternative strategies in that space. Gene therapy on the left works also for a series of immune defects, as we just heard, pioneered here in this hospital, these first trials manipulating HSCs ex vivo and providing benefit in the series of immune defect. You have here the survival curve of ADA SCID with about 100% survival in patients treated with a vector delivering a healthy copy of the gene in the stem cells. And so we have licensed product in that field, and ongoing trials are underway in some immune defects. So gene therapy is definitely on the table, and gene editing works too. It works in sickle cell. I didn't have, and I'm sure we'll hear about sickle cell because it's a topic by itself. It works in sickle cells with base editors, published and reported last year and recently. And in CGD, as we just heard, another classic immune defect where prime editing actually provided strong benefit in the patients. And so positioning of these strategies in blood disease is actually interesting and important. So where genome editing may have the upper hand, I don't believe there is one technology for all disease. I think these technologies have pros and cons and should be discussed patient by patient. Conceptually, genome editing, as we heard, may have some interest because it actually corrects the gene in situ, and in some cases, correction in situ are needed to preserve physiological regulation for efficacy reasons and safety reasons. There are a series of genes that actually you want not to add, but basically to correct in situ. Allele-specific correction for some dominant traits, as we heard, conceptually, this gene therapy adds a new copy of the gene, whereas gene editing can actually manage some other mechanism of genetic disease, such as gain of function, dominant negative traits. Of course, the packaging limits of some vectors is of concern, but it's also true for gene therapy in some aspect. And I'd say another front is, which is very preclinical, is the prospect of in vivo delivery of gene editors to hematopoietic stem cells. So as I said, right now, to perform gene therapy or base editing or prime editing to hematopoietic stem cells, it requires an infrastructure which is very complex. You need to take the cells ex vivo, manipulate the cells ex vivo, condition the patient with chemotherapy, and infuse back the corrected cells in the patient, which can be done in very expert centers that are not available in any country or many countries. Whereas in vivo genome editing on the right would provide a solution, in theory, to this problem by opening the access, this treatment, of course, but also to deliver the cargo directly to the stem cells and make the correction. And in that space of in vivo delivery to hematopoietic stem cells, although this is preclinical, a lot of work has been done with remarkable or promising progress, either using LNPs that are either LNP formula or targeted LNPs in academic labs, or our colleagues from industry have reported some promising results to basically be able to edit stem cells in the bone marrow niche or in the periphery. And other strategies such as VLPs have also shown some promising results in animal models. So I'd like to stop here with a case example of what started again in this hospital, crossing the path of a patient, and which became a scientific effort now in the lab. So this patient was a teenage girl when I met her. She was 12 years old, and she has been sick since she was the age of 2 with, that's why I saw her in the immunology department, with recurrent infections in the lung and the ENT. By the age of 4, it was quite clear that she had neurological symptoms, which we call ataxia, which is basically an issue for balance and an issue for working correctly. This was actually even more clear by the age of 5, and the patient was diagnosed with a disease called ataxia telangiectasia by the age of 6 by Sanger sequencing. This patient ended up in a wheelchair by the age of 10 to 11 years old and was on supportive care with immunoglobulins to prevent infections. She needed full-time aid at school and at home. She developed a lymphoma, which is a type of cancer, when she was 14, and by the age of 17, she was admitted in the ICU for severe pneumonia, and unfortunately, she passed away. So ataxia telangiectasia affects thousands of patients in the US, Europe. It's a disease with no solution since its description in the early 50s, with neurodegeneration and patients that end up in a wheelchair by the age of 10. It's a fully penetrant disease with an immune defect also and a cancer predisposition. And as I said, there is no treatment, and the life expectancy of these patients is around 20 to 25 years old with like supportive care and experts in the disease. So thinking about that disease actually from the clinic and thinking about strategies, there is a rationale here to develop precision genome editing. Why? Because this this genetic disease is heterogeneous genetically. We have basically one patient, one mutation, or also with some hotspots, but basically we have thousands of mutations described. the gene size is about 10 kb and cannot be delivered with any AAV strategy today. You would need 3 AAVs to deliver the full gene to the right cell or the right tissue. It's also a gene that you need to regulate really tightly. You cannot just deliver the healthy copy. It's a highly regulated gene. Second, it's a DNA repair gene, so you cannot actually cut the DNA with Cas9 nucleases, for example, to mediate targeted gene integration. We have animal model available to study the work. And interestingly, newborn screening for another disease called SCID can actually pick up patients early on, years before the development of symptoms, enabling us to have this window of intervention I was talking about. Finally, some data in actually patients suggest that low therapeutic threshold might be beneficial to these patients. So we looked at the databases across the globe. Patients' foundation were actually key, and we discussed about their role here in the research, and found actually thousands of mutations responsible for ataxia telangiectasia, thousands of mutations reported in patients. We looked in silico at the proportion of mutation that could be actually accessible with a base editing strategy, and roughly 80% of this pathogenic ATM mutation could be, in theory, accessible to a base editing strategy. And in experiments done in the lab in cells, we confirmed that 70% of this mutation causing AT or being pathogenic for ATM can be actually corrected with a base editing strategy, a bespoke base editing strategy with about 20% correction or more, which is above what we think is a therapeutic threshold, providing that we can actually access to the cell type. We then carried out this demonstration in patient cells directly to correct a given mutation of interest and show that we can actually correct the mutation up to 60% in patients' fibroblasts, and similarly confirm that we can actually confirm the functional rescue of ATM protein in the patient cells. Finally, we moved in vivo with an animal model, and with a great collaboration we have with UCLA to deliver the base editor using an AAV platform, showing that we can actually transduce the key cell type, which are Purkinje neurons, but also other type of neurons in the animals, and correct the mutation with in the cerebellum and in the forebrain. So it is an ongoing work, and of course it's not absolutely not done. There is important research needs to be done to make a proof of concept that we can do something to mitigate the phenotype or actually alleviate the phenotype of AT in a mouse. There is a lot of optimization to be done and science to be done in terms of delivery route, vector design, dosing. Other complementary approaches are also being tested in the lab. Prime editing approach, agnostic approach we heard about, as AT is caused by, in 30% of the case, premature stop codons, a strategy that David described, enabling to basically read through about 30% of these patients might be beneficial. ASOs is another strategy. And so just to foresee this project further in about 2, 3, 4 years, if eventually the science is mature and strong, and if we have complementary data in the clinic for other kind of neurological disease and demonstration that base editors can work, these diseases would require personalized treatments and therefore supporting ecosystem to do so. I'd like to finish with the end of one, because this personalized ecosystem and this personalized medicine is something that has been discussed many times. And this is a quote from Claude Grisely we heard about, founder of this institute and founder of the Department of Immunology, Hematology Department. And you can read this quote. And basically, the idea of treating one or studying one is actually beneficial to many, something that have been absolutely key. It's true in the studying of immune defects or any genetic disease. It's true in the therapeutic field too, in the ASO field with this first patient treated with an ASO, or this first bespoke base editing, treating a baby with a metabolic disorder. These efforts, or these pilot programs designed for N of one or N of few, are definitely important to trigger or reshape the way we think about this disease. So to conclude, I would say that the toolbox of precision and programmable medicine is expanding. Genome editing already corrects a variety of genetic disease in patient and preclinical models. Different diseases require different technologies, and the programmability, the modularity, and the versatility of new gene editing offers new avenues for personalized treatments. And safety, steady progress is made to address the key bottlenecks we discussed about deliveries, one, manufacturing regulation that we will discuss about today. And finally, I think part of the discussion is, of course, to define or to discuss about a pathway to clinical implementation of these strategies for the patient, again, when the science is mature already and when the medical rationale is clear. I'd like to thank many people, and this is important. I will take 30 seconds on this because this is important. I would thank David Liu, of course, and the lab members. It's been great to work with them for the past 2 years or so. The colleagues working on the AT effort, Team U being here with us today, Paul Matthews at UCLA, our colleague from the Broad Institute or UMass. I would like to thank the Patients Association that it was a first for me. I mean, I've been working with patients for 20 years, but that's the first time I actually interact with Patients Association, I would say on a weekly basis, in Europe or in the US on the AT effort, and they have been actually key for our effort here. I'd like to thank the Necker Pediatric Immunology and Hematology Department, Benedict Neven, for their support in this project. The lab, I'm still affiliated with, the Human Genetic Lab of Infectious Disease, Paris Cité and the APHP for the Mission Temporaire program, and the Image Institute also for the mobility, the International Mobility Program. Thank you very much.
Thank you very much, Romain. We'll take maybe 2 to 3 questions. Is there anyone in front here? Maybe I can start with the question on the direction we should all take. We usually oppose, as you did, the ex vivo versus the in vivo strategies, and the in vivo strategies are getting better and better and better. Do you think we should still go, so as friends, in the direction of ex vivo gene therapies, or we should directly just aim for in vivo? I think this might be one of the main questions for today.
Thank you. I will not discuss about the pathway because it's a very complex topic with different things to consider. But the ex vivo and in vivo, again, I don't think there is one strategy fits all. I think these strategies are complementary depending on the disease you consider and the problem you face. So ex vivo and in vivo are definitely pathways to work into. Of course, the next frontier to have a drug delivered in vivo where whatever is the disease would be ideal, but we have to work everything together.
Is there any question in the audience? Yeah.
Many thanks for this accessible talk for non-scientific people like me. You say to us that this is a toolboxes. Those approaches are complementary, but time is key, especially for the therapeutic window in the real life. Investment is huge to be made. So how you as a scientist, as a clinician too, you prioritize on those, between those approaches, and how you can help us as decision maker to put the money on the best value, on the best rationale for the kids on nowadays and tomorrow.
Well, that's the biggest question. So I will answer that. I think I will split the problem in many parts. There is the science problem. I mean, there is the science we do in the lab, which is, of course, to design strategies that can actually solve problems right now. But so what we heard about a neurological disease in vivo delivery, there is also an aspect of what is mature already, what is shown to work in the patients in the clinic. We heard about these strategies. I'm not going to repeat them. And here there is the question not about really the science itself, but how to implement it in the clinic, which is different by the scientific efforts. Of course, it's a field that is at the intersection of what we do in the lab and in the clinic. So part of your question will be discussed across different aspects. I would say that the funding of the research is definitely a question to anticipate on the next problem. Going from a proof of concept to implementation in the clinic is a big question here, and the partnership with different institutions, the collaboration is also a keyword, I think, in that space of the rare disease. Sharing the data, collaborating on the patient cohorts is actually important. So there are different ways to answer your question, but I would say that fundamentally you have to split perhaps what is mature already and that we need to implement to respond to new problems, and what we need to work to anticipate on the next strategies to answer or tackle other problems. But this is definitely part of the panels we'll discuss across the day. We have dedicated panels to discuss all of these aspects.
Any last question? No? Because otherwise we'll take a 15 minutes break. So just in front of the auditorium. Do you hear me well? Well. Um, Let's start. So we have just heard about American excellence in almost genetic surgery with David Liu. Now let's hear from the French excellence with Geneton at the Imagine Institute. So the first one, Geneton, is represented today by its CEO, Dr. Frédéric Reva. So he's directing Geneton since 2010 and is president of the Genoterre, so French cluster of excellence dedicated to gene therapy. Before joining Geneton, Frédéric has had multiple leadership roles in pharma and biotech, and he started his career as a 10-year scientist at the Pasteur Institute here in Paris. So Frédéric, the stage is yours.
Thank you for the introduction. It's a pleasure being here today with you all. I'll be presenting today the latest developments we have at Geneton, both in terms of products, in terms of therapies, but also in the way we address the certain of the limitations that have been underlined by the previous speakers for the treatment of rare genetic diseases. So before doing that, let me present you Geneton in some quick slides. So the first point is that we were created by we are a not-for-profit organization. We were created by a patient organization, the AFM Telethon. All French people here know the AFM Telethon. They all know the Telethon, a yearly charity event. So the AFM Telethon has been organizing the Telethon since 1987 and has invested since then more than 1.8 billion euros in innovation, out of which half has been dedicated to gene therapy, and at a time where there were not many believers in the field of gene therapy. So we deciphered the map of the human genome between 1990 and 1996, and then moved to gene therapy in 1997. Just coming back one step, when I said that we're created by the AFM Telethon, we're not only created, we are funded massively by the AFM Telethon. And as a CEO, I report to the board of Geneton, and the board is chaired by the president of the AFM Telethon, who is herself the mother of a child that died of Duchenne muscular dystrophy. So clearly, patients are in the driver's seat whenever it comes to defining what our strategy is. We have a robust pipeline of treatments for rare genetic diseases ongoing. I'll go a bit more into the detail. One of our major achievements is having been the originator of Zolgensma, so the first ever demonstration of the efficacy of gene therapy in spinal muscular atrophy mice came from Geneton. We have the founding patents for the product, both for the use of AV9 and for the product itself, that were licensed to Avexis and now which is part of Novartis. We were pioneers in the field of manufacturing of viral vector, and as we know, manufacturing is one of the key elements in the value chain of gene therapy. We have established multiple partnerships with pharma and biotech companies. There's 15 products stemming out of our research or in collaboration that are today in clinical trials, 7 programs that are on the way to reaching clinical trials, 800 patents filed, 240 scientists at Geneton, and this is based on Science of Excellence with the number of publications that you see here. Now, let me just take you through rapidly take you through the pipeline. So a drug approved, that's Zolgensma by Novartis. We have several drugs in phase 3, and I'll give you some more detail about our Duchenne muscular dystrophy drug. We're a sponsor of the ongoing pivotal phase in Europe for GNT004. There's a Lumovoq for optic neuropathy, which is held by a an outside company. There's a product in pivotal trial for Crigler-Najjar syndrome. And the Astellas product for myotubular myopathy was also originated from Geneto. Several programs in phase 1-2 for limb-girdle muscular dystrophy. We also out-licensed the technology to support AskBio's Pompe trial recently, and then several products which are at the IND CTA preparation level. Now, what's the organization to support this pipeline in our research? You could say that we are a mid-size, not-for-profit biotech organization with approximately 150 in the 4 research translational teams, and most of the leaders of these translational teams are here in the room today. We have a bioprocess and technology development group, which is very important. CMC is of the utmost importance for these drugs. Innovation for CMC is also very important. I'll tell you more during my talk. We have platforms which serve the whole organization. We have program management, we have clinical development and regulatory. And I would say the complementary arm of what we do is really manufacturing. We have a spin-off, which is Europe's largest manufacturing capacity for AAV and lentivectors, which is Hyposchesi, which is basically across the street. And having access to this manufacturing capacity is very important. It is one of the key elements in being able to move those drugs to the clinic and one day, hopefully, on the market. Hyposchesi, that really stems out of our initial bioproduction, biomanufacturing department, is now 10,000 square meter, 100,000 square feet, able to manufacture AAVs, both adherent in suspension, for both for clinical trials and for commercial phases. And it's a key element both for us and of the global ecosystem in France. It is at the service of the global ecosystem in France, and we'll hear about some clinical trials here at Imagine, for which the vector has also been manufactured at Hyposchesi. Now let me take you through one of the examples of ongoing clinical trials, which is Duchenne muscular dystrophy. Let me show you a short video of Duchenne kids. So these are old images. They date back from the 50s. In fact, these are the images of the kids from the founder of the AFM Telethon, Madame de Quéper, who had 7 children, out of which 4 were affected with Duchenne muscular dystrophy and died all 4 before the age of 17. You see here this very typical gait of these kids before they lose ambulation. All kids will lose ambulation before the age of 12. They will then need respiratory assistance before the age of 20. 20 years ago, they would all die before their 20s. Now, with introduction of corticosteroids and standard of care, improvement of standard of care, they would live in their 20s and certain in their 30s. But it still is a very lethal and very severe and lethal disorder. We have this program for which that stems out of our research and for which we are sponsor of the development, which is GNT004. It has the specificity of being a lower dose gene therapy. When I mean lower dose, it is not the only microdystrophin-based gene therapy under development, but it's the most advanced in Europe, and it's the one that uses the lowest dose. We're at 3×10^13. All others are above 10^14. It is an important element. It is an important element both in terms of manufacturing, cost of goods, and also in terms of safety. We know how important it is to inject the lowest dose possible of these AAVs. We have over 2 years efficacy data. I'll share some of them with you. And these data show sustained clinical efficacy in the first patients treated. And the pivotal phase is ongoing in the EU. It is a double-blind, placebo-controlled clinical phase. Now, I will not go into the details of the construct, but you know that dystrophin is the largest gene of the human genome. In its coding, the coding sequence is much too large to be encapsulated in an AAV. Thus, the approach to reduce the size of the therapeutic transgene by developing microdystrophin these microdystrophins will recapitulate the functional characteristics of dystrophin, the main functional characteristics, and be small enough to be encapsulated in AAV, the maximum encapsulation capacity of AAV being a bit over between 4 to 5 kb. So we have a unique combination as compared to other drugs. We have used an AAV8. We know that AAV8 is efficient both in skeletal muscle and in the heart. It has shown important safety features in previous trials. We use a SPC 5.12 promoter, and we use the microdystrophin with the exons that are listed here. Now, let me show you some of the long-term data that we have accumulated in the first patients treated. So we're not talking about the patients from the pivotal phase. This is blinded. This is ongoing, so I cannot. There's no results that I can have access or present today. But let me show you some results from the sorry for that. Let me show you some results that we got from the escalation phase. Now, on the upper left side, you have an important marker of muscle suffering, which is the levels of CK, creatine kinase. And what you see in all 3 kids from this treated at this dose in the escalation phase, you see a strong drop in the CK levels in these kids, approximately 70%, mean of 70%, which is long-lasting. You see the data here at 2 years, and for the one has been treated for longest time, now at 3 years. So we see a strong and sustained decrease in CK level, which is clearly one of the best biomarkers that we have for muscle suffering. You see strong expression of microdystrophin in muscle, which is important, which is nice to have, but we know that expression of microdystrophin cannot be a marker of efficacy. It is one of the elements. It cannot be a marker of efficacy since it is a microdystrophin. It is not microdystrophin. It's not the whole microdystrophin, but combination of this expression and the functional elements that we show with CK and others that I'll show in a minute is a very positive point. Now, if we look now at clinical markers, and in particular the clinical scale, the NSAA, which has been developed specifically for Duchenne patients, we look here at the evolution of the NSAA in the kids treated with the evolution of the NSAA with kids that are followed at the same time. So this is not historical natural history data, but it's really kids followed at the same time with the same characteristics in the same clinical centers. And you see a very strong drop in the kids which are who are not treated, and with at 2 years a difference in NSA of 9 points, knowing that 2.4 to 3.4, 3.5 to 3.5 are considered as clinically meaningful. So basically what we see here with this NSA scale, which is a scale that goes up to 34 points, this difference in 9 points shows both a very strong divergence between treated and untreated kids, and in treated kids we see a stabilization and even a small increase in the clinical scale. Now, other important elements are here the MRI outcomes. And MRI outcomes, I mean, you can have kids prepared for the NSA scale test, but you cannot have kids prepared training for the MRI outcomes. And here you see the MRI outcomes. On the left-hand side, you see the fat fraction. And on the right, you see the contractile cross-sectional area in these kids. Again, in green, the treated kids, and in red, the untreated kids. You see a very strong difference at 24 months between untreated and treated kids for the fat fraction. And fat fraction is really a sign of muscle degeneration because it measures the speed at which fat cells will replace degenerating muscle cells. And on the right-hand side, you see the volume of the muscle. And here again, you see stabilization of the volume of the muscle in the treated kids as compared to the untreated ones. And both of these measures are beyond what you would expect for MCID. Now, what does it mean in the daily lives of kids? Let me show you here. So this is one treated kid, treated in September 2022. This video was taken in 2025 for his 9th birthday. His parents took him to the Eiffel Tower. He ran up the Eiffel Tower, 2nd floor. I personally would not do it, but second kid is from London, and you see here. Now you try to do that. Okay, so I think these videos speak for themselves. So this is an ongoing clinical trial. This really summarizes the elements I just shown you. I'm not going to come into the detail, but keep in mind that this is a low-dose gene therapy, 310 to the 13, and clearly this is an important element in that. Now, how long? An additional element on the duration of the effect. Of course, duration of the effect is key. How long are these results going to be sustainable? So we have followed cohorts of GRMD dogs. So GRMD dogs are golden retriever dogs that naturally show mutation dystrophin in the dystrophin gene, and who show all the phenotypes of Duchenne muscular dystrophy. And these are the most relevant animal models for Duchenne muscular dystrophy. So we've followed a cohort. We've injected a cohort of dogs, and we kept one of the dogs for close to 11 years. And this is you see here the untreated dogs in red. They will all die before the age of 2. And you see this dog from the cohort that was kept for 11 years with a stabilization of its clinical performance on the left-hand side, with the stabilization of the cardiac systolic function in this dog. And just to, I don't know if anybody here has had a GRMD dog. Oops, here we are. So this is an old GRMD dog. I mean, 10 years is really an old dog, and we see how performing this dog can be. This is things that never happen in GRMD dogs, and of course we have performed the dog was euthanized, and we have all the parameters, long-term parameters showing expression, long-term expression of the transgene in these animals. Now, starting from some of these examples, our question, and which really is convergent with the questions that was asked by both David and Romain before at the beginning of the morning, how can we extend the successes of gene therapy to the benefit of the largest populations of patients possible? We know that we have some specific hurdles, and I'm talking mostly here about in vivo gene therapy and AAV-based gene therapy, but the questions are really common to most of the platform technologies. So we have high manufacturing costs, and clearly this is a key hurdle to bringing more of these drugs to patients and developing more of these drugs. We have a business model which is based on a single shot treatment, meaning that when you are a company, you have to recoup your investment on a single injection. And then there's a specific question of the ultra-rare disease, the very rare disease, with limited profitability outcomes, and how do we make sure that we leave nobody behind and that we address these patients, which represent the vast majority of rare disease patients. So now the answers are, and the levers are multi-layered, of course. It's about science and technology. We've seen some of this morning. It's also about regulatory, and we'll have a regulatory roundtable a bit later today. It's also about organizing the ecosystem, and I'll tell you a few words about that, and really developing public-private partnerships. Now, on the science and technology side, how do we address, for instance, the question of decreasing cost of production? If you want to decrease the cost of production, well, you have to be innovative in terms of manufacturing process. And clearly here, innovation in the manufacturing process is clearly per se a perspective for improvement and innovation. It is often neglected, but if we invest sufficiently in a sufficiently wise way, we should be able to decrease in a very dramatic way the cost of manufacturing. Also, decreasing cost of production means having to produce less of these vectors. So if you have vectors that are more potent, efficient at lower doses, then it means that you don't have to manufacture that much, and you're able to decrease the cost of production. Now, on the first part of it, how do we innovate in manufacturing? Again, this is a multidisciplinary approach and has to be taken as such. It's a mixture of systems biology. You have to better understand how these vectors assemble in cells. And it is not an easy task. You have to use improved producer cell lines. You have to explore for novel producer cells. You can introduce, and this is very exploratory, this is what we do, you can explore plant-based systems. You can explore novel transfecting agents, improve plasmids. You have to work on analytical methods to have analytical methods that are more efficient, that inform you better on what's happening in your bioreactors. You have to be more aggressive with the use of AI with the objective of developing digital twins. So this is what we've been doing at Geneto for years, and here are some of the landmarks that we have achieved over the years. We have 45 people working in the technology innovation at Geneto, and the head of technology innovation, Patrick Santambia, is also in the audience with several of his colleagues. We have developed 17 processes at a large scale, 200 liter scale, before sending them to GMP manufacturing. And over the years, we have improved the yield of the process. We have multiple ongoing innovation process working both on AI, expression systems, upstream plasmids, downstream analyticals. We're doing this either internally or with external partners, but clearly if we want to move there, this is really a interdisciplinary approach, and we are leading this. Just to give you an example of one of our recent achievements, we were starting from a cost of goods per patient of 1.2 million euros in one of our indication, a program which is currently in clinical trial. We could, both by changing the cell line, using new transfectants, optimizing downstream, we could decrease this cost of goods from 1.2 to less than 100,000 euro per patient. So this tells you, and clearly, this is not ancillary. This is not anecdotal, because when the cost moves from 1 million to 100,000, it means it's really the difference between being able to do the clinical trial or not being able to do the clinical trial. So we'll continue working in that direction. Now, improving the vector specificity and potency is also very important. Being able to develop vectors that are efficient at lower doses, and I borrowed this slide from Giuseppe Ronzetti, who's in the audience, and with here an example of a new generation AV vector with a 10 times increased myotropic activity used in the Pompi mice model, showing that you could achieve the same or even better decrease in glycogen content in muscle by using 10 times less AV than you would need AV9. So this is clearly a way, an approach in decreasing the quantity of a vector that you will be using. Now we want to be much more systematic in this approach, and I borrowed this in the next slides to Isabelle Richard, who's in the audience. We have developed AI-guided rational design for interspecies organ specificity. What do I mean by interspecies? We have developed an approach by which we would graft, aided by AI and rational modeling, peptide specific for given receptors on cell surface, those cells that we want to target, introducing an appropriate peptide, and in a way that we would both target the organ, detarget the liver. It's interspecies in the sense that in this very pharmacological approach, you can design your peptide so that it will bind to your receptor not only in mice but also in primates and in humans, meaning that, of course, once you have validated the approach globally, this approach allows you to be using mostly mice rather than going through the primate step, which is laborious and costly. Then we have put in place a very systematic AI-guided incremental multi-VR library screening, where we would integrate not only specificity, potency of the vector, but also the immune response, the immune susceptibility, which is something key, and the productivity of the vector, because you could have the most specific and immune stealth vector. If you're not able to produce it, it won't be of much use. Here, some data about the myotropic vectors that we have developed using this approach. You see here in a mouse model of a limb girdle muscular dystrophy, you see here in different muscles the differential expression of the therapeutic transgene in muscle as compared between AV9 and the two vectors here, the LICA1 and LICA3 vectors. The differential expression in muscle on the different bar graphs on the right, and the very strong liver detargeting that you achieve as compared to AV9 with these novel vectors. These novel vectors, when used in primates, show also both improved transduction as compared to AV8 here in this case in primates, and strong liver detargeting versus AV8. So we're pursuing this approach, and hopefully we'll have a clinical trial soon based on one of these novel vectors. Here, the question of CNS being also of utmost important, a result with the intravenous injection in HPs of one of our novel capsids, GNT0014. We show comparable transduction in the brain, comparable transduction efficacy as AV9, which is known to transduce to cross the blood-brain barrier. So we have a product that a vector that does cross the blood-brain barrier, but a strongly reduced targeting of liver and heart, which are at the origin of several of the side effects linked to AV9. So I could give multiple examples, but clearly this is an important approach that we follow. Now, another key question, it was raised by Romain, I think. It's a question of redosing. If we're able to unlock redosing, we would not only have a road to improved efficacy, but it would open a new model for these gene therapies. A new model because you would shift from a unique injection to a repeated and why not a chronic treatment and a totally different pharmacoeconomics equation. Now, what we know is that the redosing of gene therapy products is limited by immune response. We have been using imlifidase, a bacterial IgG degrading enzyme, which is approved by EMA for the use in HLA sensitized patients undergoing kidney transplantation. And we took a first patient with pre-existing neutralizing antibodies against AV8, which was a patient affected with Crigler-Najjar syndrome, and I'll tell you a little bit more about that in a second. And he was pre-treated with imlifidase and then successfully treated with an AV8-based therapy. So this is clearly an interesting step in order to being able to dose patients with pre-existing immunity. Is this a first step to redosing? Hopefully so. Maybe not with this exact enzyme, but maybe with improved enzymes we're working on. So just let me show you here the antibody in this patient with pre-existing anti-AV8 neutralizing antibodies. We see at day 0 the level of neutralizing antibodies. We treat with imlifidase, and we see at day 2 a very strong reduction, opening a window for treatment of this patient with AV8 for Crigler-Najjar syndrome. So what is Crigler-Najjar syndrome? It's a very rare disease. It's a bilirubin, bilirubinemia disease causing jaundice, but also the accumulation of bilirubin can be strongly neurotoxic if it is not controlled. These kids and adults have to undergo light therapy for up to 12 hours a day, each day. And this phototherapy is clearly today the treatment of reference. We have started a clinical trial with a gene therapy, an AVA-based gene therapy for Crigler-Najjar syndrome, led by, from the scientific point of view, by Giuseppe Oronziti, and from the development part by Gérald Perret, who's here in the audience. We are in phase 3, but for the time being, all patients treated were patients that were not seropositive for AV8. So for this first time, we did treat seropositive patient, and this is the result that we've had. We see here bilirubin levels in this patient. So first treated with imlifidase, then dosed with the Geneton product to treat his Crigler-Najjar syndrome. And what you see here is that after 15 weeks, we could stop phototherapy. So at treatment, you see decrease of bilirubin. So the patient is still under phototherapy, and the decrease is really due to the treatment. When you stop phototherapy at 15 days, bilirubin increases again, meaning that, of course, in the level of bilirubin, there's the equilibrium between phototherapy and the AAV treatment that we've introduced. But what you see is that although bilirubin increases again, it increases to the same level where the patient was before treatment under phototherapy. So we have a patient that now has the same bilirubin as initially, but without phototherapy. So it's a patient without phototherapy. Now, is this going to be a first step to redosing? We are actively working on that, because redosing means treatments can be repeated. And I'm talking now from an investor's point of view. It means sustainable revenue. It means stronger value proposition. And clearly, this is one of the avenues to transform a one-shot treatment into a potential chronic treatment. Let me now address something that was already addressed both by David and Romain, which is the case of ultra-rare disease. And let me be a bit provocative here. Should we allow profitability to decide who is worth treating? Are there patients that are too rare to be worth treating? I mean, this is clearly the question that we are facing today. We know that 85% of patients with rare disease have a prevalence of less than 1 out of 1 million. It's only 4% of rare diseases that have a prevalence of 1 over 10,000. And whenever a for-profit actor is interested in rare disease, he would be interested by one of these 4% disease, so leaving all the rest of the patients on the side. And we know that there are hundreds of treatments in development already in the clinic for ultra-rare diseases that have been abandoned in recent years due to financial consideration. So how do we address that? Beyond the scientific approaches that we've been discussing, clearly it's a question of financing. And however you turn the problem, at the end of the day, if you want to address this in a systemic way, financing is not going to come from for-profit. It cannot be based on public generosity either. I mean, there's 7,000 rare diseases. I mean, public generosity cannot compensate for what is a market failure. So if you want to address this market failure, it is the state's mission, it is the public mission to correct for market failure. And we, from that perspective, there is a very interesting draft legislation which was passed in front of the French National Assembly. That was a few weeks ago, which we think really goes in the right direction. And we've been lobbying for these types of legislation for some time. The objective is really to fund innovation for pediatric cancers and rare disease, address market failure in pediatric drug development. The funding mechanism is simple. It's a mandatory pharma contribution of 0.1% of their revenue. And the estimated annual budget is around 50 million euros, between 40 and 70. So the use of funds would really be to target the drug development and clinical trials. We know we have the means to do research. We have the means to prepare clinical trials. When it comes to executing clinical trials, when it comes to manufacturing what is needed, clearly the funds are not there. We believe that this is a very interesting step moving forward. Now it is under review by Senate. We'll see what the political environment is, but it has to be adopted by both the Senate and the National Assembly to become an active law. So we'll be following this in a very active way. And the few last minutes, just let me finish around the fact that answering those questions and the challenges of genetic medicine is really a question of creating the appropriate ecosystem. And this is why the French government has funded in an important way the creation of an ecosystem around gene therapy, which is called Génotaire. Which is so the French government has allocated 70 million euros to the consortium, and the participants themselves have allocated another 70 million, matching 70 million euros also. So an overall sum of 140 million for 5 years, regrouping academic institution, translation and clinical research institutions. So Imagine is part of it. Pharma, biotech, industrial partners, both French and foreign. We have, for instance, Nava Therapeutics, which is based in the US, was part of the consortium. We have AskBio. We have several foreign companies which are also part of the consortium. We have investors, we have local authorities, we have higher education. And the idea is really to put together to crystallize this ecosystem of excellence, building on a very strong history that exists here in France and several times I was reminded that the first ever successful clinical trial in gene therapy was done here at Hôpital Necker. And the idea here is really put these worldwide recognized leaders in a common transversal organization, create cutting-edge technology platform accessible to any project holder that wants to transform a project into a therapy, into a clinical trial, into a company. To provide training programs to the ecosystem and to create value by accelerating creation, incubation, development of high potential startups. So the CEO of Genoterra is here in the room, Marco Pintore, and really it is the first European biocluster dedicated to genetic medicine, both dedicated to innovation in a public-private way. Biomanufacturing is an important part, clinical and regulatory, and also nurturing startups and talents. Having said that, thank you again for your attention, and if there's any question, I'll be happy to take them. Thank you.
Someone want to start? No? Yet one.
Hi, thank you for your presentation. For those who, like me, don't work on muscle cell biology, as the treatment is an AAV, so non-integrative, how does it work with really long-term persistence with muscle cell renewal, reparation? How does it work since it's not integrative?
I will let Serge Braun, who's our DMD guru, he's in the room, answer that question. Did you get the question, Serge? No.
I was about to leave the room, but when I heard the question, I said I should stay for a few minutes. Actually, the muscle tissue is a fairly stable tissue, as long as it doesn't degenerate. And there is probably a time window for an optimal treatment, which is around 3-4 years and 10 years. Before it's too early because of muscle growth and because of the lack of integration of the vector. And if too late, there is too little amount of muscle tissue to correct. And even if the muscle tissue would regenerate, there will be a sort of dilution of the vector, of the amount of genome transferred with the vector. So probably an optimal time window would be necessary with this particular disease.
So in this case, what do you think about the integrative strategies with the VLPs and the Cas9?
The condition for efficacy of this type of approach, there is a prerequisite for that, which is you have to target the muscle stem cells, the satellite cells, and so far, to my knowledge, there is no vector that is able to do that. So there is an absolute need to find a way to transfer the genetic material in satellite cells, which is not possible as of today.
Any question in the audience? No? You mentioned also down to 62K per dose, or 26. How much do you think we can still go lower than this?
No, there's no simple question because there's no simple answer, sorry, because of course it depends on the dose. But I guess in that specific indication, I think we could go down to 10 to 20,000 K per dose.
So thank you very much, Frédéric.
Thank you.
So now it's my great pleasure to introduce our next speaker, Professor Marina Cavazzana, So as you may know, Professor Cavazzana is a gene therapy pioneer, as she developed the first effective gene replacement therapy for patients with severe combined immunodeficiency. She's a recipient of numerous awards, and a recent example is the 2026 Antonio Feltrinelli International Prize for Medicine. Professor Cavazzana is director of the Necker Hospital Biotherapy Department, where she developed gene therapies to treat beta hemoglobinopathies, such as sickle cell disease, as exemplified by the ongoing clinical trial Drepamir.
Thank you very much, François, for this kind word. Thank you, Bana and Frederic, for providing me with this opportunity to try to describe the organization and the innovative protocols ongoing in this campus. So, we are very proud to have organized a sort of very compact, fruitful bridge between discovery, that is essentially located at the Imaging Institute. And before Imaging Institute, it was spread out in all the campus. And the patient that the different reference center take care at Necker Hospital. This bridge is did by several very integrated units that are able to guarantee the complete value chain from discovery to patient bed and beyond. In particular, this contains a first piece of the puzzle where we have a clinical and translational investigational center located here at Imaging. It is able to go from discovery up to the creation of a GMP clinical trial that is secondary transferred to and scaled up to the academic GMP facility that is in Necker Hospital. But this clinical translational investigation center is also very important for the follow-up of the patient, because we tightly follow them, as we can see just later on. And it is able to invent or set up new analytic tests following the different strategies and the pathologies that we try to cure. The ATMP facility center is in the Necker side. He got all the EMA and French authorities authorization for all type of ex vivo manipulation of hematopoietic stem cells and T lymphocyte that span from cell therapy expansion of the cells up to base editing. That is something that we acquired recently for the more disruptive technology we are setting up. But also we have we have another very important piece of, we have all the regulatory and clinical units able to support us from the writing of the protocol, submission, the file to the EMA authorities, and follow-up of the patient, pharmacovigilance, and relationship with the regulatory authorities in the case of severe adverse event. This integrated center for molecular medicine also benefit of the experience of all the reference center in inherited disease. One example has been provided this morning by Romain about the capacity of to get a tight collaboration between inborn error of immunity and this integrated center of molecular medicine. So, since the already several times reminded that is not, I'm not proud because you can calculate easy my age. So, from the very first one, SKIDEX 1 clinical trials up to now, we have set up 12 clinical gene therapy trials across 9 pathologies. The last 4 ongoing at different phase of maturation is 2 clinical trials for 3 clinical trials for 3 inborn error of immunity and 1 clinical trials for sickle cell anemia. So the great advantage to be co-localized in the same area where all the actors are present is that we are able to set up a very important virtuous cycles, because long-term follow-up of each treated patient learn us deeper inside on the pathophysiology, not forecast in advance, better clinical trials, improved genetic tools, and of course at the end improved patient outcomes. To give you just 2 examples of this virtuous cycle, I would like just to underline the role of clonal tracking of a vector integration site and the chronic inflammation environment on the outcome of the trials. So clonal tracking. Clonal tracking, as I underlined before, is very important for 2 reasons. Polyclonality is a guarantee of safety in the case of a gene addition vector. That we have reproduced or renewed all the hematopoietic stem cells reservoir, but also provide us important information on the possible emergency of oligoclonal clones or monoclonal clones, as we have faced years ago. In the case of the first in the world trials on beta thalassemia major, we used a particular vector with an insulator, hoping to avoid completely the problem of insertional mutagenesis. At our surprise, we have had the hyperexpression of this gene, HMGA2, that was important also for the benefit of the patient. But this clone was integrated in very long-term precursor cells and not in the stem cells. So at the end, the clone disappeared with no side effect for the patient. So this is the reason that I think polyclonality has to be studied in the case of application of hematopoietic stem cells. This is the great work made by Emmanuel Six in collaboration with Fred Bushman, that he studied also from this genetic study who have been harvested a lot of insight of hematopoietic stem cells. First of all, we can say how many stem cells intake in this patient, over 7 different clinical trials, and which are the characteristics of these hematopoietic stem cells tree. And Emmanuel has defined the existence of not only very balanced hematopoietic clone able to give rise to all the blood compartment, but also myeloid and lymphoid dominant clones. So inflammatory condition impact on hematopoietic stem cell. Studying from 2 particular conditions completely different in terms of pathophysiology, we have underlined the impact on the biology of hematopoietic stem cells of chronic inflammation. Chronic granulomatous disease evoked this success this morning for one two patients treated by prime editing, and this is a particular form due to X-linked deficiency of GP91 and the sickle cell disease. What is in the common with these two disease? In the common is the chronic inflammation that dysregulated the hematopoietic system, and facing the failure in both for two patients in both gene therapy protocols, we try to understand the pathophysiology of this failure, because in terms of transduction, CD34 harvesting and so on, conditioning regimen, nothing was changed between patients. So thank you for the great computational skilling qualities of Emmanuel 6 and adenine. We make a single cell RNA sequencing of all the stem cells present in CGD patient, and through a logistic regression method we that could be implemented and tested with more than 500 single stem cells. We have validated a method that allow us to distinguish patients that go to have a successful outcome for patients that can fail for inflammation reason the gene therapy. So we characterize 51 inflammatory inflammatory 51 gene involved in the inflammatory pathway, and we could try to implement this with the next clinical trials. We applied a little bit the same pre-screening model to sickle cells, obtained the same result, different pathway of inflammation, and we have also repositioning a new drug that is in capacity to bring back the hematopoietic stem cells to a dormancy, because this is a very important characteristic of stem cells if you want that the result of gene therapy are stable over the time. In fact, chronic inflammation make the stem cells go out from the dormancy, biased towards the myeloid compartment, and have all the hallmark of aging and exhaustion. So the idea of this 3 months of anti-inflammatory treatment is to bring back the cells to their capacity typical of an healthy subject. This could also impact gene editing in vivo, and we are exploring with Anarita Micho this possibility. So this is the pipeline we have ongoing for a viral approach at our campus. And with 3 inborn error of immunity, sickle cell, but also neurometabolic disease that we try to expand in collaboration with Professor Alessandra Biffi from Padua. But we have also a very amazing clinical trial for recessive dystrophic epidermolysis bullosa, that is a topical treatment very efficient for this devastating SCID disease. So just one word on SCID, because you are all here very attached to this form of inborn error, because essentially for the severity, the young age of the patient. And this particularly, I am very happy to share with you this result, because this particular form of SCID, Artemis deficiency, the name has been done by Despina Bonchus, a very brilliant physician in the unit of pediatric immunohematology, that has discovered this gene, gave them a Greek name for a number of reasons. So from this discovery in, say, in the imaging, we make the POC, the patient cells prove, and everything the NSM ask to be in clinics, and finally we have the clinical protocol approved. Maybe, David, you can ask why this SCID Artemis. SCID Artemis is a DNA repair defect. This is a gene that allows to resolve some double-stranded break. So the reason why there is no TAB cells. But there is also a great hypersensitivity to irradiation and all conditioning regimen causing double-stranded break. So this is the reason why it is the allogeneic bone marrow transplantation are particularly worsening, despite a lot of improvement in other indications. So 4 patients with a typical SCID T-B- have been treated. 2 of them were no infected at the time of the transplantation, thanks to the neonatal screening, and have been transplanted within the first 3 months after birth. P1 has been diagnosed later on, has a very severe maternal graft versus host disease requiring 3 lines of immunosuppression, but also as typically in this type of patient several opportunistic infection. To note the post-rotavirix vaccination gastroenteritis that is now can be avoided thanks to the neonatal screening. Same for P5, the diagnosis was done only at 7 months of age. He suffered of pneumocystosis, hepatitis, adenovirus, rotavirus replication in the stool. So all the 4 patients recovered for any infection, restored very early after transplantation, between 3 and 6 months after the injection of gene therapy. All the T and B cells immunity go back very early and are free now of any type of medication. So the more important parameter that indicate us that this gene therapy has been successful is the high polyclonal repertoire of this patient, already present at P6. And we compare the Shannon index, that's the index that indicates the quantity of the diversity of the VDG recombination for each rearrangement. We see that using the data already published by Gigi Notarangelo with the study of Del Monte in JACI, that the polyclonality of the VDG compartment has been obtained and highest of this threshold of 7-8 that is required to avoid any second treatment, a second haplotransplantation, or to avoid any comorbidities in very long term. Other important achievement is this as early as the children are treated, the thymus is rebuilded, and you can see here the imaging of a P5 and P3 thymus that is largely overlapping to that of a subject of the same age. So if you want to compare the post-transplant toxicity and relative to the outcome of the gene therapy, you find here some figures. The data are taken from 10 patients haplotransplanted in the unit, in the pediatric immunohematology unit directed by Benedetta, but over course of this is a ultra rare disease, so the data are not completely the data that we can have today, but being this rare, ultra rare disease, 1 out 70,000 of patients. The 10 haplotransplantation are realized in a very long period, so some improvement have been registered, luckily for all of us. Nevertheless, because of this hypersensitivity of irradiation, these patients are particularly sensitive of GVHD, very difficult to control once it occurs, and also they have a lot of endothelial toxicity related to the immaturity of their system, and 3 patients in the haplo cohort died. So in the post-transplantation comorbidity, there is a very long period of hospitalization. 4 patients have enteral nutrition. This is very frequent in this class of SCID patient because of the presence of disimmunity, probably aggravated by the conditioning regimen. We have had only 1 case of very short enteral nutrition, and the patient have been very shortly hospitalized, longer than we could, because 3 out of the patients come from Europe. So the national procedure, how we can implement our health system with this therapy, we have just submitted to the NSM the ask for the hospital exemption. I can enter in detail later on if necessary, but it is a sort of market authorization limited to the France and to the French patient, and this is an important limitation for Europe. So the second procedure in parallel we are doing is we have obtained early this month the orphan drug designation that allow us to obtain a prime designation and EMA accompaniment to make this drug product a therapy. So beta hemoglobinopathy, maybe our colleague and friend can ask me why we continue to work on beta hemoglobinopathy with so many trials ongoing for gene editing, base editing, prime editing, ex vivo, in vivo. The reason is over there, accessibility. We have collaborated with 2 important biotech, Bluebird Bio and Vertex. We have no accessible drug product in France. And to give you an idea of a number of the patients affected by this disease, we follow in Necker Hospital a cohort of pediatric patients of 800 patients and a cohort of adult patients in my service of 700. So this is the real. So with the great help of Mario Amendola and Megan Brousson, under the direction of Anna-Rita Micio, that we have the great pleasure to listen just after me, we have put together a new lentivirus vector with an improvement towards the previous generation, because it contains a microRNA in intron 2 able to completely inhibit or greatly inhibit the expression of hemoglobin S. So we have made all the proof of principle in vitro, in vivo, in the animal models to show that the presence of this microRNA allow a significant decrease in the production of hemoglobin S that is in parallel with the number of the vector copy number. The association of this vector and the use of this vector and the anti-inflammatory treatment, we really hope to obtain in the patient this result. This is an inversion of the ratio between the therapeutic globin and hemoglobin S. It is to note that each time hemoglobin S is around 50% or more, this is not a cure. This is a transformative for a sickle cell patient because hemolysis continue and we continue to progress in terms of endothelial damage. So, if all this approach we have, because in meantime waiting for the implementation of the disruptive technology that we are setting up and for which we upgrade our GMP facility, patient need to be cured. So the other idea is to take all this antivirals approach and to continue to implement their use through and in vivo gene therapy through the use of viral particles. Here again, Anna-Rita Micio and his student Mathieu Van Jost are taking the lead of this project that we are helping to arrive as fast as possible to the clinics. So, in meantime, as I explained, we have two operating modes. One of larger implementation of vintage strategies, but very useful to cure patient that we need to cure, limiting as much as possible the toxicity. We are implementing the editing approach for many genetic diseases that are in the preclinical study, and I don't want to disclose anything that Anna-Rita go to tell you in detail just after my talk. So the other idea that is very common in the field and we as many other try to implement is the idea of a platform that we can have l'antivirus AAV platform, gene editing based editing in vivo ex vivo. I am very happy to present this slide that has been did for this morning by Alessandra Biffi, because taking into account all the reference center of this campus, so we need to enlarge the strategies and not to be fixed only on the hematopoietic stem cells. But hematopoietic stem cells is very useful across system and across organ, and can be very useful also for neurometabolic disease. This is the platform strategies developed by Alessandra that could be really an asset for our campus, where she mixes cell therapy with pre-committed stem cells towards the microglia compartment, AAV, but also targeted integration in particular locus and CRISPR technologies. So, in conclusion, I hope to have convinced you that we have a vivid academic pipeline with an increasing number of eligible patients. We make also many efforts in parallel to try to reducing cost and to pass to this, to transfer these advances to our healthcare system. So through technological innovation, we go to speak in the roundtable about automatization and securization of the manufacturing process, biological innovation and regulatory one. We are living a very rich moment with two innovation spread out by EMA. One it is the Biotech Act that we try to influence as much as possible, and the other one the pharma package that go to change in the good direction our strategies. It is also a great European momentum, as reminded this morning by David, but also Romain and Frederic, we need a strong collaboration, not only between Boston and Paris, but also to avoid the existing fragmentation in Europe and to try to speed up treatment driven by our major concern is to treat as many as patients as possible with the less toxic effect. So the number of people that I would like to thank is very long, but at least I would like to underline the great role as director of LTCG of Elisa Magrin, the great work did along many years by Chantal Agrel and Emmanuel Six. The Hemoglobinopathy Center is directed by Laure Joseph. I also benefit a lot of the experience of the immunohematological and pediatric unit, as well as of the adult hematological unit with the two chief of the service, Professor Neven and Professor Hermine. And I would like really undertake this opportunity to thank Annalita Micio, with whom I have the pleasure to collaborate since the very beginning of her arrival at Imagine, but also a number of immunological researchers, very important in my life, because they discovered the gene that I have the honor to bring to the clinics for a major benefit of the patient. Everything can't exist if, and maybe Winston, you are interested in developing this value chain from discovery up to patient, without the regulatory and pharmacovigilance present in the care. We have everything that allows us to go to the bedside, and I benefit of a great sustain from the direction of clinical research of our administration hospital with Milan Lazarevich and Laure Bouquet, but also since the very, very beginning we benefit of the invaluable sustain by Geneton and IFM. And also I am very grateful to Frédéric Reva and Anne Gali. Of course, patient families are very strong and courageous, because I think it is not really simple to accept to participate in a phase 1-2 clinical trial, and you for your patience. Thank you. I propose you that we listen to Anna Rita, and then we have a common discussion.
Exactly. So Narita, she's also a researcher at Imaging Institute, and over the past 10 to 15 years, Dr. Micio has pioneered the development of a wide range of gene editing approaches for treatment of beta hemoglobinopathies, establishing herself as one of the Europe's leading experts in genome editing. Narita, please.
So good morning, everybody. I'm sorry we're supposed to be eating now, but I will be relatively quick. So basically, my idea for today was to present to you the gene editing program that we have at Imagine and in the Necker campus. So we have 29 programs spanning over around 13 diseases. So we have both ex vivo and in vivo program using either viral or non-viral approaches. And as you can see, we're using a variety of editing tools, CRISPR-Cas9, base editing, prime editing for targeted integration, and epigenome editors. And again, also the indications are very broad, a lot of hematological disease, of course, but also metabolic disorders, epilepsy, and skin disorders. And so as a consequence, we also have a lot of target organs, such as the hematopoietic cells, liver, skin, and CNS. And so basically the most advanced project on genome editing, and the program that is most advanced is on beta hemoglobinopathies, and in particular on sickle cell disease that is caused by a single point mutation that causes hemoglobin polymerization, formation of sickle red blood cells, and a very severe clinical phenotype. So, as you probably know, many editing strategies have aimed at reactivating the expression of the fetal gamma globin, which is usually silenced soon after birth. And this is because the clinical severity of both beta thalassemia and sickle cell disease patient is strongly mitigated by the persistent synthesis of fetal hemoglobin in the adult life. And so a while ago, we tested several strategies targeting in particular the promoter of the gamma globin genes. And so we use in particular a base editor to generate a different mutation. And these are point mutations that they either evict the repressor from the gamma globin promoter, or they recruit potent activators in order to achieve fetal hemoglobin reactivation. And so we finally choose this mutation that we are generating in sickle hematopoietic stem progenitor cells. And this is a mutation that generates a binding site for a potent transcriptional activator, KLF1, that in our hand was the most potent one in terms of fetal hemoglobin reactivation. And so, therefore, in 2024, we started the BE-DREP program to bring base editing to the clinics for patients with sickle cell disease. And this was really a joint effort between the Imaging Institute, the hospital, and with Marina, the INSERM unit ARTTG that is headed by Angali, and of course IFM Teleton and Geneton and Genoter Initiative, and in particular the gene editing at the ATMP platform. So what are we doing? We've been developing the GMP processes, performing biosafety studies, and now we are currently transferring all this process to the GMP, hopefully to start soon a clinical trial. And so in our first preclinical data, we were able to achieve around 60% of editing efficiency in hematopoietic stem progenitor cells from sickle cell disease patients, with also a good viability and a good cell recovery, especially compared to the same cells that were treated with the CRISPR-Cas9 strategy that, on the contrary, is generating DNA double-strand break. And so importantly, HSPC-derived red blood cells show high level of fetal hemoglobin expression at both RNA and protein level, and importantly, we observe a strong reduction in the frequency of sickling cells. And importantly, we also showed that the base editors were safer compared to the CRISPR-Cas9. Indeed, here we analyzed the transcriptome of human HSPCs from sickle cell disease patients that were either treated with adenine base editor or with CRISPR-Cas9. And as you can see, we observe much fewer dysregulated genes in base edited cells compared to the same patient samples treated with the CRISPR-Cas9. And interestingly, we observed that CRISPR-Cas9 basically in sickle HSPC induced the expression of genes involved in interferon alpha or gamma response, in inflammation response, as well as in apoptosis. And this was observed also with another base editor that we use as a control. This is the cytosine base editor. But most importantly, this did not happen with our favorite base editor, which is the adenine base editor. And interestingly, then we look at patient by patient, with the P1 being more inflammatory than P3, followed by P2. And as you can see, Cas9 exacerbate the inflammatory and apoptotic profile in patient 1 and patient 3, while it induces the expression of genes involved in inflammation in P2, who at steady state condition does not really show an inflammatory profile. But what was interesting was also to notice that both AB and CB did not further exacerbate this inflammatory profile. These were analyses in bulk population that contain only a tiny fraction of hematopoietic stem cells, and therefore we performed single-cell RNA-seq analysis to be able to look at bona fide HSCs. And as you can see, we confirmed that the P1 and P3 have inflamed HSCs, and that Cas9 induces an inflammatory profile in the 3 patients, is also observed by the increase in the interferon gamma score. And importantly, we also wanted to evaluate the efficacy of this strategy in long-term repopulating hematopoietic stem cells, and therefore we transplanted a human HSPC from sickle cell disease patient in immunodeficient mice, and cells were either treated with CRISPR-Cas9 or with adenine base editor. And so the chimerism, meaning the frequency of human cells, was variable but similar in the different group. But most importantly, we achieved the highest editing efficiency using adenine base editor compared to the Cas9, and most importantly, also the highest level of fetal hemoglobin in the progeny of stem cell-derived patient cells. And so, as I mentioned to you before, we're collaborating with Anne Galli at ARTTG, and so she's basically developing the clinical cell manufacturing process, and so far she achieved very good viability and editing efficiency. Of course, as you know, this strategy have already hit the clinic, and we had the chance to collaborate with Correct-seq that started a clinical trial using a base editing strategy targeting the gamma globin promoter. This trial was mainly done in a patient with beta thalassemia, and so these are the 5 first patients that are now transfusion independent thanks to the high level of fetal hemoglobin. But of course, there are also other strategies such as prime and epigenome editing strategy that are currently being explored and optimized to develop a therapeutic strategy for beta hemoglobinopathies. And in particular, as you know, prime editing allow all base conversion, deletion and insertion, but also combination of this mutation. And therefore, we decided to introduce multiple mutations in the gamma globin promoter with the idea that together these can further boost fetal hemoglobin expression. And so in the work led by Megan in my lab, Maria and Anne showed that indeed the introduction of multiple mutation in the gamma globin promoter leads to higher fetal hemoglobin expression compared to the introduction of an individual mutation. So at the time that we published this work, we achieved only up to 5% of editing in HSPCs, but now actually we are optimizing the protocol and we were able to achieve a higher frequency of editing recently. And then we decided also to explore the epigenome editing technology in order to reactivate fetal gamma globin expression. So basically, epigenome editor can modulate gene expression by changing histomodification or, for example, DNA methylation. And of course, we decided to reactivate fetal gamma globin expression by depositing active histone marks and by removing repressive marks such as DNA methylation. And so first we confirmed that when silent, the gamma globin promoter are highly methylated, while when cells start expressing high level of fetal hemoglobin, the DNA methylation level are significantly reduced, and the cells also acquire marks of active transcription, such as H3K27 acetylation. And therefore, we decided to basically use epigenome editing in human sickle HSPCs in order to demethylate the gamma globin promoter. And so in this case, we use an epigenome editor containing this TET1 enzyme. And also, we also deposit histone acetylation using an epigenome editor carrying the CBP acetyltransferase. And so this led so far to around 40% of fetal hemoglobin and a good reduction in the frequency of sickling cells. I have to say that this is the frequency that we observe in asymptomatic sickle cell disease carriers that are subject to this very same assay. So I think overall this work showed that DNA methylation matters at the gamma globin promoter. This was something that was challenged during the years, but we confirmed it also in collaboration with a group of Mitch Weiss in Memphis. We achieve therapeutic fetal hemoglobin reactivation. However, we are further optimizing the protocol. And in addition, we are also evaluating additional targets. And so in conclusion, for beta hemoglobinopathies, I think that CRISPR-Cas9 strategy, for sure, they are an excellent strategy and are already in clinic with approved clinical therapeutic drugs, although if they are not available in France. But I think that still CRISPR-Cas9 exacerbate the sickle inflammatory profile, and this, I think, it can contribute to the exclusion of many patients from the treatment. On the contrary, we really think that base editing strategy are very promising with all the first clinical trial that already started. And again, we hopefully would like to start this clinical trial quite soon here in France. And finally, we provide a proof of concept for a prime editing of epigenome and an epigenome editing strategy that, however, still needs to be ameliorated, for example, in terms of efficiency for the prime editing strategy and in terms of persistent of sufficient fetal hemoglobin expression for the epigenome editing approaches. But of course, and Marina stole the slide from me, actually, these are, we're all discussing about ex vivo strategies in which we collect the cells from the patient, we then genetically modify them in vitro, and we inject them into the patient. However, of course, this patient then needs to undergo myeloablation, which is one of the main cause of the secondary severe adverse event that we see in the patient. And therefore, for the future, the idea is to develop an in vivo strategy based on the injection of viral or non-viral particle directly into the patient, where they directly would go and correct the hematopoietic stem cells. And of course, this would reduce the complexity and the cost of the therapy, and also probably the toxicity, because we will not need any more myeloablation. We just really correct inside the hematopoietic stem cells. And therefore, as anticipated by Marina, Mattia in my lab, we decided to use the syncytin, that is a human endogenous envelope that also has immunosuppressive ability. So she used this as envelope for lentiviral vector to target specifically hematopoietic stem cells. However, syncytin recognizes these two receptors, ACT1 and 2, that are expressed on a wide variety of cells, and so I decided to retarget it by fusing it with a binder that is targeting CD117, which is a marker of hematopoietic stem cells. And so in this way, he was able to basically meet two main requirements for the in vivo strategy, which is the low or no immunogenicity and the specificity. And therefore, it tested the vector that are pseudotyped with syncytin alone or with syncytin targeting CD117 in hematopoietic stem cells that were sorted based on the expression of CD117. And he could see that targeting the lentiviral vector to CD117 allowed efficient delivery in CD117 high hematopoietic stem cells. And it also showed that the system is adaptable to other cell types, such as, for example, T cells. And so in parallel, we are also working with several collaborators and company for a non-viral delivery to hematopoietic cells. And this is a work that was done by Annalisa, PhD student in my lab, in which she compared electroporation, which is the current method to deliver editing tools to the hematopoietic stem cells, with LNP in human sickle HSPCs. And she showed that LNP are safer in terms of viability and cell growth, while preserving the cell identity. And she also was able to achieve a similar editing efficiency compared to the electroporation. And importantly, of course, in our model, a strong reduction in the frequency of sickling cells. However, I think that when we discuss about in vivo gene editing, we need to go back to the pathophysiology of the disease. For example, in the case of sickle cell disease, the HSCs reside in a bone marrow niche that is characterized by ineffective erythropoiesis with apoptosis of erythroid precursors, ROS, inflammation. And so this alteration can impact, for example, the targeting of HSCs or the editing of HSCs. For example, this is a biopsy from a patient from sickle cell disease that showed an unusually high number of adipocytes, which, as you might know, are negative regulator of HSCs. And so I think that in general, it's very important to study the sickle cell disease bone marrow niche with all the components in order to optimize and maximize the success of in vivo gene therapy approaches. And finally, ideally, it would be great to treat this condition before the onset of irreversible damage. For example, during the fetal life. This is not something that we are doing now, but we can think about that for the future for several diseases. In this case, for example, for stem cells, we show that the hematopoietic stem cells at this stage, they are healthy. Of course, the small fetal size will require just little editing reagent. And also we have, in the development, we have a window in which we have a tolerogenic immune system. And we are working on this project in collaboration with Dr. Shangaris and Professor Kypros Nikolaides in London. And finally, a few words of the other editing programs that we have here at Imagine, although again, sickle is the one that is more advanced. So we have been trying to explain to you now the different modus operandi that we have here at Imagine. So we have been correcting really specific mutations, specific disease causing mutation, either more common mutation like in thalassemia, but also really rare and unique mutation like in this case, where we use adding base editor basically to efficiently correct a mutation that is caused CGD. And in this case, we were able to show a restoration of the expression of the oxidase complex and also functional restoration of ROS production. But of course, this is a very specific strategy in the so-called N-of-1 gene correction approach. But I think that we need to still think about a more universal approach, for example, universal gene correction approach using these tools that allow the targeted integration of the full gene so that we can correct all the mutations that are affecting one gene and causing a specific diseases. But we can also think about universal gene modulation approaches, such as in the case of sickle cell disease, in which basically we can modulate the expression of a disease modifier. So again, this is the example of the sickle cell disease, in which we reactivate the expression of the fetal gamma globin in order to compensate for the defect of the adult beta globin. Of course, this can be extended to many other diseases in which we can, again, modulate the expression of disease modifier genes that can be fetal gene, embryonic genes, or even paralog genes. And this can be applied to a variety of diseases that are affecting a variety of organs. And of course, in my lab, we started with another hematopoietic disorder. And so Ellen is my lab is trying to fix alpha thalassemia, which is caused by the defect in alpha globin expression. And in her case, is trying to reactivate the expression of the embryonic zeta globin gene. And of course, then we heard about that, and we probably will discuss furthermore in the afternoon about a universal approach for multiple diseases or for classes, let's say classes of disease. So the so-called platform technological approaches, in which, for example, we can choose a class of base editor, the favorite base editor, our favorite delivery tools, and of course our favorite organ, the bone marrow for myself, in order again to develop treatment to treat a class of diseases. And so in conclusion, I think that I hope that I explain well to you that we have several, we are editing programs here at Imagine based on different editing technologies with different modus operandi, ex vivo and in vivo, viral and non-viral. And I think this is made possible thanks to collaboration with in the French biosystem with all the actors that already mentioned, but of course also thanks to the collaboration with many international scientists and clinician. And of course, we'd like to bring this to the clinics, and this will be further discussed today. And so we have ongoing discussion with the INSM and EMA. Also, the Imagine is a founder of the European Genomic Medicine Consortium, which brings together clinician, patient, scientists, all the stakeholders in order to bring editing approaches to the clinic. And finally, as mentioned with Marina, is heading basically the Future Center of Excellence for ATMP European Network. And finally, so that we can go and eat, I would like to thank all the members of my lab that have contributed to the work that I show you, all the funding agency and all the national and many international collaborators, and of course also all the clinician and the patient, and all of you for your attention. Thank you.
So we'll take 2 questions, and after you will have until 1:30 to eat. You want to start? No burning question. So maybe I have one for the epigenome editing. So we'll allow the reactivation of the gamma globin. I was wondering what would be the advantage of this approach compared to just gene addition, just either beta globin or gamma globin addition? Yeah,
I think in our research experiment, I think reactivating an endogenous gene is quite potent and might be easier to have a higher level of therapeutic hemoglobin concentration. And we also started because still, as Marina showed, hematopoietic stem cells from a patient with sickle cell disease are very hard to transduce, but I did not show the data. Still, there are also some stem cells, the same stem cells, some of them, when there is a lot of inflammation, they are also hard to edit. So at the beginning, I was convinced that it was easier to edit, but maybe still we need to face some of the problem, at least for some particular patient that show a high inflammation profile.
Any question? No? Okay. So in this case, we're going to eat. So go back here at 1:30.
Good afternoon, distinguished colleagues and friends. It's a great pleasure and privilege to introduce you, our next speaker, Professor Tim Yu. Professor Yu is a physician scientist at Boston Children's Hospital, professor of pediatrics at Harvard Medical Schools, and associate professor of the Broad Institute of MIT. His research focuses on human genetic neurobiology, and precision genomic medicine, with particular emphasis on rare genetic disease. He is internationally recognized for his pioneer contribution to personalized medicines, and today we are honored to have him with us to present his lecture entitled From One to Many, Scaling Individualized Medicines. Thank you. You have 30 minutes. 30 minutes.
All right, thank you. Thank you very much, and it's an honor to be here. I will share on a personal note that one of the treats for me is to come here to hear about and hear directly from the scientists who launched the pioneering gene therapy programs for SCID, which were what I was actually riveted to during my MD-PhD training in San Francisco in the 90s. And there was a time when I actually really thought very seriously about becoming a hematologist-oncologist, because of this amazing opportunity to bring the bench together to the bedside. Sadly, I failed to do that. I've been a neurologist and a neurogeneticist at Boston Children's now for 20 years. But I'm glad to say that it was worth it, because we are now seeing these moves come back, these worlds come back closer together in a way that I was worried that I might miss if I did neurology, but I'm very glad to see that I was wrong. So I entitled my talk From One to Many, Scaling Individualized Medicines. And I'd like to pass over many of the same themes that we talked about earlier in the morning from the lens of work that we've been doing the last 10 years on N-of-1 style therapies for neurologic disorders. And this review, which will skip over my disclosures real quick, all involves medicines that are for conditions that are decidedly non-commercial, sadly. And this work that we're doing together is to think about ways in which we might actually change that. My lab here started off working on gene diagnostics. We were lucky to be one of the very early adopters of next-generation sequencing, and we used this to discover dozens of neurologic disease genes from 2007 to 2017. And then our work, which was supposed to continue in that vein, stopped because we met a patient that switched us to this entirely different space of N-of-1 therapies and interventional medicines, interventional genetics. And the challenge that we've been talking about today is this rare disease challenge, the statistics which we've already covered. The point that I'd like to draw your attention to is the shape of the distribution of these diseases, which we all know looks like a dinosaur with a head and a very, very long tail. And all of the activity in the space is concentrated at the head. If you walk through the halls of biotechs and pharma in Boston and New York and San Francisco, in Europe, you will take tallies of programs that suggest that there's tremendous activity in genetic therapies around the world. It couldn't be a better time in the world to be having a genetic disease if you have one of these 25 conditions that are the most frequently encountered. And all of the activity, some of these programs may have 80 different active pipeline drugs being developed for them. The sad reality, though, is that once you get to the 25th program, TTR amyloidosis, which actually is pretty well subscribed with 8+ programs, as you fall below that threshold, most of the programs have, of the 7,000+ other monogenic conditions, have 0 or 1 program in the pipeline. And most of the patients in the pediatric neurology clinics at Boston Children's Hospital who are coming in or being hospitalized will have one of those. This challenge has been addressed and is being addressed in many different ways. But the most interesting opportunity to think about this challenge in a new way comes with the advent of technologies, programmable therapies like the ones that David invented, that allow us to now look at the problem not just from the left, to try to enhance the efficiency of the commercial system to develop medicines for progressively less common conditions, but actually to flip the way that we look at it, to look at it from the right, to start at the long tail, at the most rare conditions, maybe even the ends of ones. And that's the work that we've been doing now for the last 10 years. As folks know, because it's been widely publicized, back in 2017, we met a patient with an ultra-rare neurologic condition that was progressive and fatal. We were able to diagnose her because of advanced technologies available to us to look for cryptic mutations that were at that point clinically inaccessible. She had a deep intronic mutation that allowed us to envision a way to treat her that actually we realized was within our reach to develop as a laboratory that did not necessarily require biotech investment to advance. We used a technology called antisense oligonucleotides, which are now very well understood. At that point, there had been one approved drug, Spinraza, which had shown remarkable efficacy in neurology. And what we learned from that experience is that this type of drug, which are 20 nucleotide snippets of synthetically modified nucleic acids, can be reprogrammable and used to develop therapies very effectively, especially for central nervous system disorders. And in thinking about their use in the central nervous system, for this type of looking from the right interventional genetics approach, there are 3 properties that I'd like to highlight about them that make them, that are especially relevant. The first actually is the one on the right, it's delivery. That of the different modalities that are available to us, there are very few that actually can penetrate the brain in a broad, widespread manner. And that's really critical because the brain remains very mysterious and very heterogeneous with many different substructures, many different circuits. And our ability to know which substructures, which cell types, and which circuits need to be targeted is incredibly limited right now. Most of the pediatric neurologic conditions that we are studying, we've only discovered in the last 5 to 10 years. And our ability to then be able to make a specific biological, precise, mechanistic prediction does not exist. So a broadly deliverable therapy like an ASO, which can go and span brain and spinal cord and multiple cell types, is incredibly useful because you can use it to modulate circuits, gene circuits in situ, as opposed to more specifically. And I will also just point out that one of the advantages of this modality is that it's titratable and reversible, meaning that you can adjust the doses as you go. And you can stop if you encounter a toxicity. And so if you are thinking about venturing into a space with less than a traditional pharma package of data to advance into it, these properties are very important because they allow you as a clinician scientist to be more empirical and more methodical in your approach. Instead of having to choose one dose and hope that you got it right, you can now deliver doses that allow you to adjust on the fly and make adjustments as you need to. As folks know, with this type of technology, we made a medicine for this patient. We made it in 10-month span, and that was a big deal back then when we first described it publicly, 2018, 2019. It got attention because of the speed. It got attention because the symptoms in this patient seemed to improve. We saw reductions in seizure intensity, seizure frequency, duration. Sadly, it was not enough to save her life, and she did pass away in year 3 of the trial. But her gift to us through this effort showed us what's possible, and her postmortem gift to us that the family granted to us also showed that the drug was bio-delivered exactly in the distribution that we hoped it had been delivered, in a gradient, highest in the spinal cord, but also with significant penetration into the brain. And there was a 1-to-1 correlation between the distribution of the drug and the target engagement reactivating gene expression, building scientific credence that this type of work could actually have the type of influence on gene expression that could be useful for other patients. We've been working now with the FDA for 9 years since then to continue to build on that concept. Our first meeting with them, of course, was negotiations around the FDA's our IND in 2017. But we had a follow-up meeting with them 2 years later to begin to first actually propose this idea of individualized medicine for fatal life-threatening conditions, pointing out that for single gene disorders that are clinically severe and lack existing treatments and are not public health hazards, like N of ones or very small numbers, and using well understood platform technologies like ASOs, but someday extending to editors and other modalities, that we could actually ask for and receive certain allowances from the FDA to streamline the submission of these packages, including accelerated reviews, accelerated manufacturing programs, and accelerants for animal toxicology studies, for instance, allowing single species toxicology instead of two species toxicology. Fast forward 10 years, and the field has come a long way. There are now multiple FDA-approved ASO drugs available for clinical use in a variety of indications. Several of these are neurologic. These two in yellow are specifically central nervous system targeting agents, where I've had the most focus. And there are many intrathecal ASOs in clinical trials in the central nervous system that are looking extremely promising. And this space has matured a lot. There have been also a growing number of parties now working in the N of 1 CNS ASO space, beginning with our work at Children's, extending to Columbia, which ran the second program, UMass, which ran the third program. Actually, Helene Tran is over here. I may embarrass her as being the first author on that really important program. And then many others, including activity in Europe, like the Dutch Center for RNA Therapeutics. There's the 1M1M Association, a nonprofit that has started here to coordinate European activities in this space, and our own N of 1 collaborative. This space now has seen 35 N of 1 ASO programs submitted and approved by the FDA, and with patient experience now accrued on over 80 patients. One of these sponsors is actually Enlorum, a nonprofit started by Ionis, and just 2 weeks ago they announced their dosing of their 50th patient. One single nonprofit making a large impact in the space. What are some of the lessons from these programs that are relevant to our thinking about ways to collaborate and move forward together? Here are a few of the lessons that I'll draw from our programs, which, since we started them early, are some of the longest standing programs that have been currently running. First, we start with ataxia-telangiectasia that Roman described and introduced already this morning. And as we've heard, this is a condition that's associated with immunodeficiency, but the immunodeficiency is relatively mild. It's predominantly a neurodegenerative condition with progressive ataxia as its primary disabling feature. And as we've also heard, it sometimes can be picked up on newborn screening for severe combined immunodeficiency by looking at T-cell receptor excision circles. Patients with this condition can be picked up, like this one, at birth, and then diagnosed with AT, and then their parents are told that she was relatively asymptomatic now. She would be relatively asymptomatic for the first few years, but then she would begin to neurologically deteriorate at age 5 and be wheelchair requiring by age 10 or 11, and that there may be fatality by young adulthood. And there are no treatments, and as we've heard, it's too large to package this gene in, say, an AAV virus. We've run this trial for this patient now for 6 years using a drug that we designed targeting one of her mutations. This is published work that we first put out there 2.5 years ago. I'll give you clinical updates on the case that are unpublished. We published how we were able to find a mutation in this patient that led to creation of a new splice site in the middle of exon 53. And we published how we were able to design an ASO to block that splice site, to rescue RNA levels and rescue signaling through the ATM kinase pathway. And we explained how we started this trial at age 2 years, 10 months for this first index patient. This is the natural history of AT, a composite neurologic index called the AT-NEST score. Higher is better. And patients in the beginning have a period where they show relative stability of their neurologic symptoms. it's not perfectly stable. They are mildly ataxic, but their development in motor patterns or wiring at the same time that they're slightly deteriorating. So the net effect is relative stability. And then they undergo the sharp decline between ages 5 and 10, and then they plateau. Our trial began at age 2, and it is now extended to age 9. We've had to be very patient because the first few years we knew we were going to be in this period where not a lot was expected to change. But now that she's 9, we are beginning to report what we're finding, which is that she's not shown clinical progression through age 9. These are AT-NEST scores, a composite score here, and then subdomains shown here. Again, higher is better, and our patient is in the red asterisk. The box and whiskers plots show AT-NEST scores from age-matched individuals with AT who are plus or minus 1 year of age, and of course they have not been treated with any investigational therapy. And our patient is doing better than expected in all of these domains. We have stability also supported by secondary outcome measures. I'll show you just a few to give you a flavor. Here is a wearable device that we periodically have her wear when she's willing to, on her wrist or on her ankles. She wears it for a couple days at a time. And when she's had it applied at several points during the course of her trajectory. Our colleague Anupam Gupta, who has pioneered this AT digital wearable, has shown that neurotypical kids have scores that look like this, and children with AT have scores that look like this, clearly separable between the two populations. And what we find is that our patient lies right here in between the two, performing with the best of the AT children and maybe the slightly less coordinated of the neurotypical children. We've also done sub-analyses that if you look at the mild mutations, that these mild mutations, individuals with mild mutations show up in purple, and so our patient looks like those. We've seen other promising signs too, stabilization of serum biomarkers, somatic growth signals, etc. The point is that we think that this is a sign of a clinical response that would be the first successful staving off of progression in AT if this holds. And back to the theme of my talk, from N of 1 to N of many, we've actually found that this mutation that we've identified in her is actually a founder mutation in Turkey, and we found 10 additional children in Turkey and recently received funding to expand our trial with an FDA-funded R01 grant to bring this from an N of 1 to an N of, well, at least a few more. We also have a patient in Europe, in Germany, who's now being treated with our friends Mathis Anofsik and Rebecca Schul. We flew the family to Boston for a year to begin that treatment, and now he's been doing really well receiving this therapy the last 4 years in Tübingen. Other cases and other lessons. Our KCNT1 epilepsy program is one that we published just a few months ago, describing a very, very severe form of neonatal onset epilepsy. This is a condition called epilepsy of infancy with migrating focal seizures, and these children have neonatal onset in the first hours or days of life of dozens of seizures per day. The details of this are not important for the purpose of my talk, but just briefly, it's a toxic gain-of-function mutation that leads to channel hyperactivity that can be shut down with an antisense oligonucleotide. And this is a condition which is among the most severe of the childhood epilepsies, with profound neurologic impairment in patients, and half of these patients die young at a very, very young age. Using ASOs to reverse the expression of this hyperactive gene, to rescue the electrophysiology of iPSC-derived neurons, to rescue mice and do appropriate safety testing in human cells for immunogenicity, off-targets in iPSC neurons, animal studies, et cetera, trials like this can be launched, sometimes with positive effects. This trial here involved dosing a patient with intrathecal doses, escalating doses 10, 20, 30, and 40 milligrams, following her seizure course in response, and finding that her seizures went on average from 5 to 15 at baseline, and then they dipped after the third and fourth doses to between 0 to 2 for nearly 50 days. And that's a response that's not seen in this condition. However, I show this example to allow me to illustrate another difficult fact about this case. In these sorts of interventional genetics projects, progress is not always a straight line. This patient also had a serious side effect. And the reason that we stopped after the 4th dose, despite the seizure suppression, the remarkable seizure suppression, was that she developed ventricular enlargement and required a VP shunt. This, when we found it in 2021, was actually, no, 2022, in 2022, was one of the very first reports of this being a side effect of some ASOs. They were also being concomitantly reported in a Huntington's disease trial. And now we know that this is one of the potential toxicities of some ASOs that we have to watch out for. The happy news is that we were able to change our dose monitoring and our dose escalation scheme and to safely resume dosing in this patient. We were also able to switch to intracerebroventricular injection to be able to reduce the amount of drug exposure required in this family. And her seizures have seemed to respond. Sadly, another patient who was also in the same protocol for whom this also happened did not feel that a VP shunt was appropriate for their daughter given her very impaired neurodevelopmental status, and she passed away. Still, these programs have the ability to go in directions that offer hope to many other individuals. And working together with Helene, Sarah Vier built a program, a 2nd-generation ASO for this same disease. This is a trial called the CANDLE trial, which is now publicly open, first patients dosed in Europe. It's a phase 1/2 trial, first in human open label study of an ASO knockdown for this KCNT1 epilepsy with intrathecal dosing to begin. And this is a move from now an investigator-sponsored effort to a company-sponsored effort with the ability to impact many, many more. Other programs that we've launched include in the eye, this program for an inherited form of blindness. I'll skip over the details in these 2 slides, but I'll just show you briefly that this is a patient who has had deteriorating vision and also dorsal root ganglia sensory dysfunction, leading to congenital pain insensitivity. And she's been dosed with a splice modulating ASO, which restores expression of this gene FLVCR1, which is a choline transporter in those cells. Dysfunction of choline metabolism appears to be toxic to those cells, and her vision over time has declined. With dosing here beginning in yellow, we've been able to safely dose her now for 2.5 years, and we're waiting to see what the overall impact is going to be on her vision. It seems like it may have stabilized in the last 2.5 years. I can't say whether this improvement is real, but we're going to just keep on continuing to watch her. These are many programs that now have been launched by us and others in the space. And thinking about the lessons from these programs, again, 35 programs, over 80 patients, also N of 1 editing programs, that it now invites us to think, what have we done? What are we doing? And why are we doing it? This is a space now where our initial efforts were really motivated largely by compassion. There was the idea of establishing a scientific proof of concept to see if this type of work could be done at faster scale than is traditionally done. But it was primarily a compassionate effort to help patients without other options. As it scales, though, and one shows that it is possible to develop a fleet of drugs in this manner with a great effort, we have to think about what are the other motivations that we need to keep in mind to keep these programs well justified. One is that these programs have the chance to generate critical learnings for rare genetic diseases. And I would say there's a contrast in, depending on your specialty, as to how important this is. If you have conviction about the mechanism and target for your condition, say it's a severe combined immunodeficiency, you've identified and characterized and beautifully studied and elucidated the problem, then fixing it and developing the drug for it may not teach you that much more that you need to know. But in my space in neurology, with most of these conditions having only been discovered in the last several years, this is a critical area by which we can gain acceleration in the space, to test the suitability of a target, or to answer even fundamental neurodevelopmental questions about how the brain develops, works, and repairs itself, about therapeutic windows, and at what point in brain plasticity is correction of an underlying genetic defect too late. Last, if this type of work could be scaled to support scientific learnings, could it also be scaled to support medical application, clinical scale application, as a delivery model for precision medicine? This theme of one-to-many is one that I think folks in the room are well familiar with. Usually, thinking about the translation from insights from rare disease to common diseases, we often argue to our politicians, to our colleagues, to our reviewers, that our grants for rare diseases will lead to insights and treatments that may actually apply to many different common diseases. And the fetal hemoglobin switch might be an example, or PCSK9 might be another example. Those same rules may apply to the N of ones, where they provide pilot insights into a rare disease that otherwise will not see any therapeutic attention, that could open the door to a rare disease therapeutic that might have an insight to the common disease. Recognizing these opportunities is one of the things that a group called the N of One Collaborative has been working to try to standardize. We've developed a series of guidances to help recognize what types of mutations might open up a patient to eligibility for one of these approaches. And David Cherry, Marlon Laffer, Margaret Meserve, and others have run these, developed these rules and then applied them to various disease cohorts. 15% of individuals with ataxia-telangiectasia are eligible for therapy for a splice-modulating ASO. 16% of patients with early onset epilepsy are eligible in principle for an individualized ASO. 5% of patients from all comers from a single medical center at SickKids in Toronto appear eligible for an individualized ASO. That's just with an ASO. If you now turn this, of course, to the technology that David invented and that you are all advancing here for editing, these numbers become even more substantial. This is that group, our N of One Collaborative, which I'm proud to have been a co-founder of. Winston Yan, who will be speaking later, was our founding president of this group, and it's attracted international interest. This is a group that has advanced several different white papers to help bring folks along together in a cohesive way. Guidelines for patient identification, drug design, communication, and consent. Critical scientific, genetic contributions, but also critical medical, ethical considerations as we think about these types of programs and explaining them to our clinical colleagues and to our patients. This group has also allowed us to explore different ways of working and thinking about how you have been so successful in setting up the right mix of funding and science and patient application. One mini success that I'd like to briefly celebrate is that the N1C recently announced a collaboration with Lilly Genetic Medicines, where Lilly will provide its know-how, its technology, siRNAs, ASOs, shuttles, et cetera, to the orphan disease community, and working together with proposals that are advanced by members of the community. Lilly will provide and manufacture drugs for those indications that then can be used and tested by members of the community on an academic basis. And a critical thing is that Lilly is not asserting commercial rights over these programs. They are granting ownership of the programs to the community. And a lot of people shake their heads and say, why would people do that? Are they really going to offer that up? And the answer is that they really have. Their lawyers have written it up to make that very clear in the contracts. The reason for why is that their catchphrase that they devised medicines for patients, learning for Lilly. The concept is that these platforms that they're advancing are going to be benefited by seeing them more broadly applied. And if the results from those are published without restriction, but also shared back with the parent company, that becomes another type of virtuous cycle to add to the list of justifications that we use to help to advance science and advance this work. On the regulatory side, it's necessary to think differently about these cases. We all know that in conventional drug development, drug development timelines are pretty impressively long, pretty impressively expensive. And the key is that each approval is meant to regain the investment via patents and market exclusivity. These all also require, from the regulator standpoint, for each new drug, even if it's targeting a single mutation, to be filed as a new program, a new submission with 1,000 pages of supportive administrative documentation. And in contrast, though, the types of portfolios of drugs that we'd like to make using ASOs and editing technologies really don't fit that model at all. We need to be able to now accommodate a technology that allows for drug design in weeks, end-to-end development in 7 to 12 months. This can be done actually reasonably inexpensively. Right now for ASOs, the prices are reasonably not prohibitive. It will take some time to bring editing costs down further, but these will involve potentially hundreds or thousands of drugs, each of which cannot be filed on its own. We're motivated to think differently because of that shape of the curve. That shape of the curve creates a gap that is not addressable except by being creative. And one of the parallels that we've thought about in the Center for Therapeutic Genetics is imagine two children, one born with a severe heart defect requiring an organ transplant or severe corrective cardiac surgery. We do have paths for treating these patients via expert clinicians, teams, surgical plannings, consents, ethical review. And those plans using standardized tools and procedures don't require filings with the FDA for every deviation that may be made from the standard on behalf of that patient. The system would not work if that were required. Can we imagine a situation where regulators borrow some of the aspects of this surgical model to inform a new modernized regulatory stance whereby this could be applied to custom oligonucleotides or CRISPR editing programs? Again, it's tackling this problem from the right, looking at patients and ultra-rare diseases in the rarest situations first, thinking about a procedural model for just-in-time treatments that is still informed by the FDA, but just in a different way. The FDA's guidances for individualized medicines have been followed up with their next generation version. In the US, it's something called the Plausible Mechanisms Framework. This is, interestingly, a guidance for industry, not for investigators. And it's explicitly targeted to industry because it is describing a framework for approving a medicine or a family of medicines. And this is a really interesting acknowledgement of the potential of these platforms to treat mutation-specific indications. And it recognizes the FDA's understanding that you need to unlock industry in order to make this field actually work. Another competing model, though, that we are considering, and some folks in the UK are advancing this concept, that instead of having an approval model, maybe we need an accreditation model that would be more flexible than a single point-in-time approval. And such that under this model, an accredited medical center could act and treat a patient and tell the regulators about everything that they do, but they wouldn't be required to stop and wait for a signal for each decision. Regulators could step in at any moment to withdraw approval or stop a program, but the stepwise back and forth that's required currently, which we know would not work if you're treating dozens or hundreds of patients, would be replaced by something like this. That is the basis, one of the bases for this concept of our Center for Therapeutic Genetics. This collaboration between David's group at the Broad, the Rare Disease Translational Center, ourselves at Boston Children's. It will be described in much more detail later by Winston. I'm just giving you a preview of it right now. One of the programs is going to be happily targeting the brain, but there are many other indications as well that we're excited to discuss with you. So to just summarize, from one to many, to me, there are many ways in which these N-to-1s have the ability to extend to impact many. Our first patient, Mila, inspired this pipeline that I described of over 80 patients treated in the years since. Our program in ataxia telangiectasia was built for one patient, but then target a recurrent variant, including this cluster in Turkey. It also allowed us to demonstrate that 15% of all patients could be treated using that same approach if we could scale it. Our program in KCNT1 for an initial index patient, Valeria, allowed us to generate data that supported the targetability of this disease gene and show marked seizure reduction that may have helped grease the wheels for this amazing new effort from Servier to expand this to many others. One point that I did not get to mention is it also turns out that lowering KCNT1 may not just treat KCNT1 epilepsy, but it may treat several other sodium channel-based epilepsies too. Mouse models of SCN1A, 2A, 8A have been successfully treated with this ASO. So all of that really pushes us to reimagine this model that we've always had, using genetics to go to neurobiology, to go to therapeutics, and to think about ways where we may actually now be able to rely on some of these insights, but go more directly from the genetics to the therapeutics. And we have this capability not only to do that, but then to let the therapeutic response reverse translate to teach us about the neurobiology. And to do this over and over again to expand this to the less and less common of the conditions. The conversation with all of you is about how to do this and to learn from how you've set up this institute to see how we might set this up in Boston. Thinking about academics and nonprofits, we're used to licensing our approaches to biopharma and having them develop the therapies. But maybe similarly, we can develop some of these therapies with pharma's important partnership, and then maybe bring lessons and learnings back to the companies that we're collaborating with. All of that requires lots more, not just science, but social discussion. And I will just put this up to point out that in talking about individualized therapy, we think differently not about the science and the regulatory, but also about the risk. And picking cases that are well justified, where risks and benefits are appropriately weighed, requires us to share this responsibility and have responsible public transparency as well. So I hope that with this, we can think together about building a system that's resilient to all of these interesting challenges, to take advantage of the opportunities, build something that's fit for this actual purpose, and resilient to risks that we might encounter. Thank you so much. I'll stop there.
Thank you very much. It's very beautiful. New platform, a lot of hopes.
Congratulations. This is really great. I have a scientific question about ataxia telangiectasia. If I remember correctly, you didn't show us the MRI of the brain of this child. It would be interesting to know how it is 9 years post-treatment.
Yes.
And also, how did you determine the dose of the product you injected?
Good question. So if I had 5 more minutes, I would have shown the MRI, which represent a really interesting puzzle. So in the manuscript that we're drafting to describe these results, the first result is that the MRIs have shown continued shrinkage of the cerebellum over this time period. It's a little bit of unsatisfying because actually the last year and a half of data points, the rate of deterioration seems to have deviated from the natural history, but there was a 4-year delay before we saw any change in the parenchymal volume. to us, the puzzle is that there seems to be a dissociation between the anatomic size of the cerebellum and the function of the cerebellum, which is actually something that as neurologists in ataxia and movement disorders we see quite a bit. But that is the important first interesting observation and caveat about that. The clinical data looks really good. The radiology has that extra wrinkle. Dosing for the atapexin program was calculated off of dosing for nusinersen, another splice modulating ASO of the same category in chemistry and the same target abundance. A lot of the initial dosing that we're doing, again, we're titrating it empirically based off of clinical response, and that's how we have targeted the doses for all of the 35 programs. They are largely empirically based.
Thank you for this great presentation. I was wondering, how do you see the future of ASOs outside the field of neurology?
That's a really good question. ASOs have got, well, gladly, some amazing competition outside of the brain. I think that for liver editing, I do think that the work that Kieran and Becca showed us at UPenn using David's tools is likely the most proximate path forward. I think that ASOs, however, in the eye, ASOs also do very well in the liver, and one of the 35 programs is a liver-targeted program using a GalNAc conjugated ASO. And there are other programs that, shuttles that look like they're going to be able to drive target specificity to many additional organs. So it would be premature for me to say what I think, where I think it would go. I think there's a lot of new delivery technology to other organs still out there.
One more question.
Thank you for the great presentation. Going back to Mila, where it started, you have shown targeting of the ASO to the brain, to different areas, and you have shown modulation of the pathways that are intended to provide benefit. But despite that, she had obviously a different course than we would have wished. What do you think is the limitation? Is it a matter of distribution, cellular targeting? What are your thoughts?
I think the primary issue is time. There's an important secondary issue, but the primary issue is time. In her case, she began having symptoms at age 3.5, and we began treating her at age 7. And by the time we all know that by the time you show symptoms, there's already been significant circuit injury on the way. I think that we need to look more for cases like ataxia-telangiectasia, where you can find these children at the newborn stage. The other advantage is practical, that if you find them at a uniform checkpoint, then your patient inclusion, your patient cohort that you are starting with is more uniform, and you are more able to extract signal as opposed to the very stochastic process by which a Batten patient otherwise might be diagnosed. The second consideration that was distribution, and that's also critically important. There are gradients of ASO distribution in the brain, whereby the deep nuclei might see 3x less drug than the superficial cortex. And for a cell-autonomous neurodegenerative condition, if that 3x, you either have to tolerate a 3x overdosing to be able to cross the threshold to save the thalami and other basal ganglia, striatum, et cetera, or we have to find smoother distribution mechanisms.
Thank you. We are already late. I think we can move to the next. Get the first one table. Thank you again. I'll ask to Marina Cavazzana, Dr. Han Gali, Giuseppe Ronzetti, David Liu, and Winston Yan to come. And we will discuss about from the bench to the first in human, preclinical proof of concept, quality control, and toxicity studies.
So thank you very much for agreeing to participate to this round table. Just a few words for introduce the participant, because probably we don't know each other. Oh yes, otherwise not. So I would like first initiate with Professor Anne Galli, that leads the accelerator of gene therapy. This is a public research laboratory dedicated to cell gene therapy. Anna is an expert in immunology and translational research, and she spent over 20 years developing innovative gene therapy strategies. Her current work focuses on accelerating the translation of next-generation genomic therapy into clinical practice by strengthening the interface between academic, R&D, and industry, and of course, patient care. So at this round table, she brings a combined scientific and pharmaceutical perspective on the major challenges shaping the future of genetic medicine. So thank you, Anna, to be there. So we have also Giuseppe Ronzitti. This is Giuseppe is the director of scientific strategy of Geneton, head of a team dedicated to the treatment of metabolic disease, and with a large experience which span from POC studies to clinical development with phase 3 and phase 1-2 ongoing clinical trials, as well as 3 projects in advanced preclinical phases. And then I think we don't need any other introduction for David. Conversely, we have the great pleasure to have also Professor Winston Yan, who is a physician scientist that is building a sustainable and scalable infrastructure for precision medicine and for the treatment of rare disease. He is a senior director of the Center of Therapeutical Genomics, because you changed recently from your at the Broad Institute, and he leads a non-profit drug development center advancing in vivo gene therapy. He is the senior director of this center, and so he can guide us and give us advices on how accelerate gene therapy for rare disease, and what is your plan. So thank you very much. I come there with you all. So I have prepared some questions just to be sure that we can interact actively. So maybe I can initiate with Anna and asking you, what critical safety insight have you gained from lentiviral vector experience, and how transferable are they to genome and the base editing approaches?
Thank you, Marina. So, you know, of course, that lentiviral vectors integrate into the genome. And we are now, using a battery of different new tools that modify the genome. So we have a commonality here to think, what type of genotoxic event can be induced. Perhaps the most significant thing is to remember that these side effects and these adverse events were also largely discovered by using these tools in patients, and were not always really predictable, at least initially, by models. So I think that should also serve us to really try to understand, how to think about that, and also to think beyond just the interaction between these tools and the DNA. And then, now we're facing with multiple possibilities of disruption of genes, and even epigenetic changes. But we have to also think in population dynamics, because I think it's pretty, at least to me, quite obvious that if you have high polyclonality and a very rich ecosystem of gene-modified cells, you have probably less likely chances to have the emergence of dominant events. So I think all of these have been really very valuable tools, and that we can probably use that together to try to predict and model and study, this genotoxicity of the new CRISPR base editor, prime editors, etc., etc.
David, would you like to react, or Giuseppe, have you any other thing to add to this?
I think that was a great summary. AAV, the same story is being played out now, and those who have followed some very recent publications know that while AAV integration frequencies are much lower in cells and in animals than lentiviruses, they're not zero. And AAV-mediated integration into the genome of animals has been associated with initiating cancer in those animals. It had not been seen until recently in an AAV human clinical trial. But in fact, Becca Ahrens-Nicholas, one of baby KJ's doctors, was one of the lead authors on this paper that came out recently reporting this event. But I think the important emphasis there was that even with this now confirmed clinical risk, in that trial in particular, the physicians assessed that the risk-benefit still favored treatment because of the effectiveness of the treatment. But it is a great reminder that, for a while, I think the AAV field held out hope that even though oncogenic integration was observed at a low frequency but detectable frequency in animals, that it maybe wouldn't happen in humans. That turns out not to be the case. It's not zero, at least. But it doesn't necessarily mean that they're not worth taking forward as therapies.
Of course. So, okay.
Yeah, no, I was thinking about what you were presenting before, because I think the inflammation part is also something that, the host vector interactions in the context of disease, these are also very relevant, and you can get this information from, as you showed, from lentiviruses or from genome editing, and I think it's very important because this can really influence the outcomes of the disease. So this is something that is also very important.
I'll follow up also on something you said earlier of the importance of clinical follow-up and pharmacovigilance, even as academic investigators. I think that's something where at an institute and at Necker, where there's such close collaboration that happens, and you use that as a way of learning from the patients and making treatments iteratively better over time. So I think with this, the oncogenesis that resulted from the early SCID treatments, as a field, we all learned, right? And I think that's a really important process, not just like technical process, what happened, but just the operational clinical process of how we rigorously follow SAEs and work them up and then share it with the field. I think that's a very important precedent to have set.
Yes, of course. So continue on this line and taking into account so many powerful tools you have provided the field, David, for correct genetic disease, any type of mutation. I would like to ask you what are your advice to rigorously assess off-target effect, genomic stability, and long-term risk in humans, and how you balance all this effect, because this morning we have had no time to discuss about off-target and genomic stability.
Yeah, that's right. It's a great question. Early on in the history of gene editing, at least framed from a therapeutic perspective, which really, of course, began with zinc fingers and tail domains, but then sort of exploded with CRISPR. During the early era, off-target editing was, really the topic that was most easy for the public to understand, and therefore probably the most common question by far that we would receive as gene editing scientists was off-target editing. Over time, I think the number of slides that I've put in my talks about off-target editing has paralleled the decrease in the focus on off-target editing as a concern. Not necessarily because I think there have been an overwhelmingly large number of clinical uses of gene editing, the 25 trials I listed, there are, if you add in all the public and unpublic data, there's more than 150 patients treated. But, if you do the math for what an infrequent off-target mutation that has a negative consequence, how much you would like to monitor that before it really descends into the background of human mutation, spontaneous mutation, you would need much, much larger numbers than that. I think, though, that with the positive clinical data, maybe people just, both in the scientific community and in the general public, have begun to appreciate that it's really a balanced perspective. Like everything we talk about that's critical here. I've certainly had patient advocacy groups and rare disease patient family members tell me explicitly, well, your community, I wish your community would just cool it with the obsession with off-target editing. I even had a parent of a rare disease patient tell me once, if you traded cancer for my child's disease, that would actually be a good trade, not a bad trade. So if that's really the only thing you really worry about for off-target editing, maybe you should consider that the disease is sometimes so much worse than the rare off-target event you're trying to prevent that your field's perspective is a little bit out of whack. I think, in reality, all we can do is try to take the best technologies, which constantly get better. We're always profiling off-target editing as we develop and improve these tools, we meaning the whole community, not just my lab, and continue to try to deploy them thoughtfully, using, as regulators are also, continue to require us to do, the most sensitive methods we can practically to detect off-target editing, but most importantly, just keeping in mind the risk-benefit analysis. So in the baby KJ case, for example, there was I'd say a reasonably good off-target analysis done, but it was all done on the timescale, knowing that the baby would have a short life expectancy if all of this didn't happen. So, as Karen and Becca very wisely designed, don't let the perfect be the enemy of the good. I think that's the overall one-line summary answer to your question.
So I don't know if Reid or Anna would like to complete.
Just one little comment, I think, of course, you cannot be 100% sure, right? You cannot sample all the cells, verify. So there will always be a leap of faith, so to speak. So we are enjoying right now a lot of, very sophisticated technology to model, the different side effects. But we are always sort of, modeling, what we know and, looking at what we expect to see. So ultimately, there's sort of a vision in science fiction that at least if we could, in a cheap way, sequence, fully the whole genome, looking at the whole transcriptome of every cell and then sample enough to be sure enough. That would be great. So I think there's still, some progress to be made at the technological level and to study patients. But again, I think at some point you just need to, trust and go on.
Yeah. Although if we reach that aspiration, the first thing we would realize is in your 27 trillion cells, this time tomorrow, you'll have maybe 100 billion new mutations. This is true. This has been measured. So our genomes are not perfectly static, but that does not absolve this field of the responsibility to try to minimize the additional mutational burden we add to patients, of course.
Yeah, and then perhaps if we go back to the issue with the AAV insertion we were talking about before, the most important thing in that publication is that that baby. Anyway, it was. I mean, the tumor was removed, and it was still progressing in terms of cognitive, so it was still treated by the AAV. So, beside the fact of the tumors, there was a clear impact on the safety. And this is, of course, it's always a matter of risk-benefit calculation, right? So, if you are dealing with such an aggressive disease, it will be a tumor, maybe even considered as something happening because of the. I'm thinking about the impact of the disease on the tumor formation. There may be some impact on the disease that you won't see because these patients were dying anyway, and so they were not developing tumors. So.
Okay, and if we try to see a little bit far away the problem of CMC, my question should be, with in front of us all these new technologies and with the new complexity and opportunities for simplification, how can you leverage this to redesign manufacturing workflows, and how we can plan to organize platforms in all your field of competence?
So David and I have actually talked to the FDA about this, about how do we, in the world with platform gene editing, streamline CMC requirements that were really geared towards small molecule drugs, large populations, as well as redosable and large batches of medicines. I think there are a lot of very reasonable things we can already start eliminating. Like, for example, David gave the example of that chronic granulomatous disease treatment that Prime Medicine did. They made one batch. They can treat all the available patients in the world. And then there's a CMC requirement to do 3 manufacturing runs just to say you can, you know how to do it. It's like, that you can do it consistently. Is that necessary? Is that reasonable, right? Other requirements, such as for very small batches, the cost for stability, the cost for validated versus qualified exercise. There's a lot of CMC nuance in there that actually is in need of updating, and that's a very expensive part of most drug development. I will say that there's platforms actually offer, from a pure technical standpoint, actually gene editing platforms actually offer, I think, more standardization of CMC than with gene therapy vectors of gene replacement. In most gene editing, like if I were to package a prime or base editor into an AAV and I want to change it to a different gene target, the amount of DNA and RNA that the DNA in an AAV or RNA in an LNP product that would change is much smaller than changing a whole new transgene. And that actually makes the characteristics, like the physiochemical characteristics of the product, much more repeatable from a CMC standpoint. And we can actually build up platforms versus having different, transgenes with different promoters that actually get expressed differently by the 293 T cells and such. So that's another reason why I feel like there are a lot of great things about gene therapy and where they've gotten us, but I do think editing as that next step becomes much more modular from a CMC perspective.
So I agree with what you say, of course, and think we should, aim to streamline our understanding of all the different tools. I just want perhaps to say also that, there's this impression that viral vectors are very complex and that we should move away from that, having more simple things. But I just want to remind that if you have an LNP with an antibody, targeted to target this LNP and then an mRNA, you have basically 3 components to manufacture. And, it's not necessarily always, simpler or maybe, I don't know, less expensive. So maybe we should keep this in mind and not, discard basically the old tools that have worked so well. But,
I'm actually very bullish on viral vectors because of all the great work that's been done, and it's more about replacing the cargo, not with a therapeutic transgene, but as with an editor that can then, do the specific base corrections. Yeah.
So, engineering vector, Giuseppe, identification of a new receptor, ligand modeling. What is the contribution of AI to all this progress, and how far we can go with this expectation of improvement for AAV?
Yeah, I mean, I think the speaker of this morning, they provided a clear example of how you can use AI and engineering in general, I mean, protein engineering in general, to modify the vector, to improve the distribution. But it's not just that. I mean, it's not all about biodistribution, because we have diseases to deal with. So there is also the transgene. The way we modify the transgene can really have an impact on the efficacy. and can also have an impact on the safety, because there is always the possibility of changing the profile of immunogenicity. This is something that has not been, that is somehow addressed, but it's clear that even the new enzymes, they may be needing some kind of tweaking to escape the immune system. Although I have the feeling that this is something somehow may be addressed also by biodistribution, by changing the PK, so changing basically the time the transgene is seen by the cells. After that, I think the one comment on AI, I mean, AI can clearly have an impact on the way we develop new medicines, but it can also have an impact on the way we produce these new medicines. Because, I mean, I'm speaking, of course, of my experience in AAV and what I've seen happening at Genethon and in other companies doing AAV development. You can clearly have an impact on manufacturing by applying the classical design of experiments, but together combined with the AI to identify the right way to produce your specific drug. I think this is very important. This may have another impact. And if we want to go really far from, I mean, what is my experience, or going into clinical experience, and this I think is particularly relevant in ultra-rare and rare disease, it may be the impact of AI in the way we design a clinical trial. So for instance, the use of digital twin for patients with rare diseases. We know that N will be anyway an N of few even in this disease, and so this may help a lot in improving the quality, and also the controls, like the control group, the synthetic control groups that we may implement in AI. So there is I mean, this is not exhaustive, of course, but this is what I've seen happening right now in the use of AI in the in the whole process. Yeah.
So in vivo model, preclinical in vivo model. So we have ex vivo gene editing, base editing, prime editing, and in vivo the same possibilities. So have you a global strategies for ex vivo gene therapy and in vivo ones to propose to the FDA to accelerate the arrival of the drug to the patient and avoid unuseful in vivo models?
I think there's no coincidence that all of the gene editing technologies' first clinical application were ex vivo. I think literally every gene editing technology that was introduced to a person, at least FDA cleared, started out ex vivo. Because if you're using the most popular ex vivo platform, which is probably mRNA electroporation into hematopoietic stem cells, the editor isn't even in the patients anymore. So you transiently express the mRNA. The mRNA transiently generates the editor in the cells outside of the patient, and the lifetime of that expression is typically less than the time before you actually transplant those cells, as you know well, and therefore the FDA, in principle, should have easier time not having to worry about the effects of this new cargo, the editing agent, in the patient, because in principle, the editor shouldn't even end up in the patient. The consequences of the editor will end up in cells that go into the patient, but that's very different than saying the editor will be in the patient perhaps for a long time. So ex vivo therapy, in addition to, of course, providing thus far the majority of patient outcomes that have read out, has played an important role in helping to de-risk editing platforms from the regulator's perspective. Of course, the future of any medicine is in vivo, because unfortunately we can't take the brain or the heart or the liver out of a patient, edit it, and put it back in. It's either impossible or deemed not practical or not safe. The majority of gene editing trials, at least the ones that I showed, are now in vivo, and I think that will only continue in the future, gated largely by delivery, in vivo delivery strategy, technology development, of course. But that's really what's needed to truly democratize gene editing for as many patients as possible.
But you forget to tell me which animal species have we used to the risk in vivo application.
I was hoping you wouldn't ask me. No, it's, you know, you can use large animals, and if you want human relevance, non-human primates, but the truth is, for efficacy, there are almost no non-human primate models that have genetic diseases. And there are many people, I'm one of them, who believe that it's not even obviously ethical to make non-human primate models of certain genetic diseases because the disease is horrible. But it will certainly take many decades. I'm not that patient. So, the vast majority of efficacy studies are done in mice, of course. We all know the limitations of mouse models. Of course, biodistribution, toxicology, pharmacology tends to be preferred in larger animals. And there, as long as the regulatory agency recognizes that you don't need target engagement and efficacy to go with your biodistribution, that's, I think, a reasonable compromise. But hopefully, because of the great modularity of all of the platforms that we've talked about in this symposium so far, regulatory agencies should realize that the great treasure, the great leverage of these platforms is that their biodistribution, when delivered using the same delivery method, should be consistent regardless of whether the guide RNA's sequence of A's, C's, G's, and T's, or U's, is one sequence or a rearranged sequence. So that's really, I think, one of the great hopes of how we can accelerate what is normally a very slow step in the process of large animal studies by obviating their need once a certain combination of cargo and delivery agent has shown enough biodistribution toxicology studies in large animals that you don't need to repeat those.
Any other comment, please?
Yeah, I'll have a couple of specific examples for how we think about development today and how I think we can accelerate in the future. I'm of the mindset that I think to be collaborative with our regulatory colleagues, we do want to establish the base case of how do we go in the full stack development from, maybe iPSCs to then, primary hepatocytes. I'm not talking about liver-directed gene editing to then mouse, NHP. You might need to do it once, right? But the beautiful thing about editing is that you can track a very well-defined target engagement marker, which is the percentage editing success you have across all of these. And then you can often correlate it with human genetics of what is a sufficient treatment dose, right? Many diseases are autosomal recessive, which means that if you have half of the copies, you probably are okay and you're healthy. So with that, I think you have this almost this like chain of evidence that as you run more cycles of it, you can start dropping out or abstracting different pieces. So for example, right now, in baby KJ's case, I don't think they created a full humanized mouse model of it. It was like a cassette. It was a Rosa locus that had the editing function, or it had the different alleles that was inserted. And they just show that this is the demonstration of editing inside an in vivo system versus editing of the specific disease model. And that was sufficient. Maybe we can, once we have enough experience with that, we can abstract it one back of just say, let's just do an iPSC. And if we get sufficient editing there from all of our data and using AI and inference models, we can say like, this should get us this level. And we can redose, so we can dose to effect. Like those are all things we can take into account to accelerate the development process.
And often the leaders at the very top of these regulatory agencies are very consistent in their view that animal models should be less relied on, that all of this should be streamlined, that many of the principles we've talked about are the future and should be the present of regulation in this space. But of course, when it trickles down to the individual regulators whose jobs, frankly, depend on making the right decisions, there tends to be more conservatism. And I think, to be fair to them, sometimes it's not so clear when a leader says, yes, let's get rid of animal models. Then the junior regulator is left saying, well, how can I convince myself that this treatment's actually efficacious without an animal model? So there is a tension that comes with the natural evolution of the field and the opportunities that everybody sees to streamline that we'll have to resolve. I know there are regulators in this room. I'm sure they're aware of the fact that the vast majority of the cost of developing these medicines and the vast majority of the time it takes to make these medicines is spent meeting regulatory requirements. That's generally a good thing because you don't want people to be treated with materials that we have no idea if they're safe or efficacious. But it does mean that regulatory requirements play a hugely outsized role in deciding how much of the science gets to patients and how quickly.
Can I just add one thing? I would like to really emphasize the importance of pharmaceutical quality when you manufacture the products. And I think that this is really critical for all of the difficulties of in vivo, which are biodistribution, immune responses, or even dosing. They need to have, very high quality pharmaceutical development and production and manufacture. And I think, unfortunately, we don't pay attention sufficiently to that. And even in the, groundbreaking use of in vivo CAR-Ts, vectors for in vivo CAR-Ts, they were barely well manufactured, or at least not very well described. So you can still, go in humans, for, very, severe diseases. But I think that if you want the field to move, this is a really important aspect that needs to be worked on to ensure high quality and also fast regulatory approval. Because for regulators, I think the, pharmaceutical quality of the product is absolutely a critical aspect.
I was thinking that in that aspect of, on the quality, of course, but also on the translation, right, from a preclinical model, whatever it is, if it's an iPS-derived organoids or a primate, perhaps the value of the N01 trials, they may be that they may create really they can give you a value of this translatability, because you can understand how much you have in your organoids, how much you have in non-human primates, and then you have in humans. And of course, perhaps that where the regulators may be more flexible in case of an N01 is when you have a high benefit, basically. You can have a high benefit, so perhaps you can go faster down the road and then have, in the end, a demonstration that your technology is efficient and safe in the best. I mean, I feel like this is something that you learn by doing translational, is that there is no perfect model for prediction, and so you really have to go as far as you can if you want to really understand what's happening with your technology.
Thank you very much. I would like to ask to the public if you have a burning question, because it is really a privilege for us to have this extraordinary panelist with us. So maybe somebody from Anna Rita.
Thank you very much. That was very interesting. So I'm now chairing the ASGCT Gene Editing Committee, and we're writing a draft to the guidance that FDA gave on off-targets. I don't know what you think about the guidelines that they gave, if you have worked with them, because I'm in this committee, but I'm European, so that was interesting. But I don't know if they discuss with any of you, with scientists, before drafting this guidance for sponsors. And
they didn't call me, but I have at least indirectly heard some of the requirements, because some of our IND-enabling studies are now meeting those requirements. I think I don't know how closely the FDA's released guidance or draft guidance mirrors what your committee came up with. I will say that one of the consistent off-target regulatory asks that ends up being a tension between the scientists in a program and the regulators, at least for FDA, is doing an off-target analysis in every type of tissue that the vector might get into, which for AAV or AAV9 is pretty much every tissue. That has always struck me as something an ask that takes a disproportionate amount of time and resources for the benefit. And in fact, I have begun to favor just taking naked genomes in a test tube, human genomes in a test tube, stripped of all of the protein and chromatin that could hide the genome from editing agents, exposing them to high doses of the editing agents, nominating all of the sites that are engaged by the editor, or at least the DNA binding domain of that editor, and then PCR amplifying those sites from cells that are treated in vitro with high doses of the editing agents, recognizing that you won't always be able to find every primary cell type that your vector may get into. In some cases, that may not even be known, what cell types. But it's hard for me to think that that's the best use of the time and the resources compared to the trying to balance the need from patients, especially for some of the ultra rare and more urgent conditions where you don't anticipate treating 1 million people needed to even see a 1 in a million sort of level off-target event.
Yeah, we highlighted exactly the same because it was strange that they proposed to do off-targeting in all the tissue that could be targets, and also they propose off-target in vivo, which I'm not sure it makes sense to look at off-target editing in vivo.
Yeah, well, you're not going to use humans, we presume, so that means you're going to be doing off-target analysis in a genome that's irrelevant for the patient.
Primates, I guess. I'm not sure that would be interesting.
Yeah, in my discussions with FDA, it's been a little bit of an adventure on this issue. Sometimes I had an Interact meeting with FDA in which, this is for our progeria base editing trial, in which they initially said we want off-target analysis in all tissues. I pushed back and said, we do this off-target analysis for this N of 1 trial, initially N of 1 trial, exposing the naked genome to high doses of editing agent, and then we, just what I just described, they actually paused the meeting and then had their own discussion where they go on mute and you sit there on the Zoom for 10 minutes, very nervous, wondering what's going on. They came back and said, okay, we agree. But then in the official report they gave back to us, they said, we changed our mind. And we do want you to do off-target analysis in those tissues. So, we're trying to now propose a sort of compromise position where we do it in enough tissues where if those tissues give back a consistent, enough cell types where if those off-target analyses come back with the consistent results, that they hopefully won't literally hold us to editing all of the tissues in that AAV9 gets to, which is, many, many tissues.
Very short. Can I ask you one question? Can this be offset, those requirements, can they be offset by the proposal of, doing a very thorough monitoring of the patient post-treatment, very thorough pharmacovigilance?
Well, the answer from my observation of FDA, not speaking as an FDA representative, is no. I think it would be reasonable. Almost always post-treatment monitoring is offered as part of a compromise to an FDA ask that the sponsor really feels is unnecessary or impractical. I'm not sure that's ever had a very conciliatory effect on the ask.
Thank you very much to all of you. Very great discussion. We have to stop, unfortunately, but we can continue in the break. Thank you.
Thank you very much. We will move to the next sessions. We will focus on patient organizations. I will ask to the moderator, Professor Banajabri, to come on stage, but also 3 speakers. François Lamy, Vice President of AFM Téléthon. And from the US model, Alexandra Souza on CGS patient organizations, Nina Frost from Rare Hope Founders. Please.
Yeah, I think we had decided that there would be 10 minutes presentations. No presentation.
Nina has some slides.
You have some slides? Okay. So maybe we'll start with, Nina. So Nina Frost is at the head of Rare Hope. organization. And so she trained, I think, first as a historian at Cambridge before becoming a consulting manager. And now she's playing a critical role, I think, in concert with David Liu's lab. And everything started also with the story of your child and the impact you wanted to have on rare diseases with neurological impact. So I let you present for 10 minutes and then...
So, here we go. Thank you so much for having me. I do appreciate being here and talking on a subject that's so near and dear to my heart. We talk about patient organizations frequently as advocates, but I want to talk about patient organizations as organizers, as architects, as builders, as strategists. I think that's the role that we've tried to take in the last number of years, and I think we've learned some good lessons, and I think we've really found a way that we believe we can really assist scientists in all the brilliant work that they're doing. So my organization, Rare Hope, was founded in 2017. My daughter, Annabelle, seen here the day she was born. She was born in 2015. And this is, you can kind of see the joy in this moment. You can kind of see her ear right in the middle there. My older daughter, Clara, is delighted to have her baby sister at last. And this was a really wonderful moment that I'll always remember. And we looked back in the months and even maybe years that followed, and we saw that within moments of Annabelle's birth, when she was born, they took her from me, wrapped her up on the delivery table, and right then and there, she was having a neurological episode. And no one in the delivery room noticed. No one was looking for a rare disease. No one, thought that there might be epilepsy or complications of any sort of condition. So we kind of had this blissful month or so, and then her symptoms progressed, and it became a challenging time for all of us, as for all rare disease family members, I think, who are in this position where they have a diagnosis they didn't expect. There's no treatment on the horizon. There's no hope for a better future, and you kind of learn, as we did, that you have to invent that future yourself. So in the years since Annabelle's diagnosis, we created our organization, Rare Hope, and we wanted to put together as many pieces as we could to build therapies for Annabelle's condition and for other rare conditions as well. I think all of our work really has been predicated on the premise that creating therapies for rare diseases isn't just a scientific problem. It's clearly a complex scientific challenge, but it's also an organizational problem. It's almost a business problem, and we had some of the capacity, I think, to take on that problem. So over the years, we've worked with a phenomenal group of scientists, innovators, field leaders, people who've done some incredible things, obviously. They bring incredible expertise to the table. I think we've found, though, over the years that scientific expertise is only one piece of a larger puzzle that needs to be put together. I think, you have all these scientific experts, you have all this scientific expertise, but each expert maybe owns their own piece of a larger problem. And no one really truly owns the entire problem. So we decided that our organization would take on the problem, we'd own the whole problem. So to do that, in our mind, was functioning as a strategic hub, thinking about all of the pieces that needed to be assembled in order to create a therapy for Annabelle, for other children like her. So what that means is, the researchers, the scientists we work with, they do an incredible job of optimizing their lane, their area of expertise. We're able to kind of take that 30,000-foot view and really optimize the whole system. So while we're thinking forward about what needs to happen in the future, we're identifying gaps, we're identifying capabilities to build, we're putting those pieces in place that we know are going to become relevant in the future. It would be absurd for us to think that David would be up at 2 a.m. worrying about whether we'd have access to the natural history data that is really siloed in our condition. But obviously, David doesn't have to. That's one of the burdens that we try to take off the plate. We try to tackle all the challenges before they become big issues. And part of what we do as well is, I think we're very confident that creating a therapy for a disorder is not nearly enough. I think we have determined that creating a whole ecosystem of research, a portfolio of different research projects that span therapeutics and multiple different types of therapeutics, that span basic science and clinical research and infrastructure building, things like biomarker investigation, animal models, things like that. Putting all of those pieces in place, which we've done over the years in Annabelle's condition and are starting to do in other adjacent conditions, I think that is the enabling piece that really lets these wonderful programs where we're collaborating with the Lou Lab and Alex, it really puts the pieces in place that someone's going to need, and often many people need. Or there are experiments that happen because the basic scientists have made great advances, and all of a sudden there's an experimental system that Alex and his team can use. So it's been very productive. We've also really enforced collaboration pretty aggressively where we could. The scientists in our world work wonderfully together, and I think Alex and the Lou Lab have been very influential in facilitating that collaboration, but that's been wonderful. So the last thing I'm going to say is I think patient organizations can also have a very important role throughout the full life cycle of therapeutics development. This is not a question of patient advocates going to the FDA at the end and asking for support, right? This is a question of patient organizations putting together the resources, pulling data together, understanding what patients experience, what patients prioritize in terms of symptom mitigation, implementing their priorities and experiences into preclinical experiments with researchers. Those are the pieces we've done so far for the gene editing project. We've created new humanized mouse models. We've put all of these pieces together to move things forward. We're now kind of in this intermediate stage where we are pursuing multiple biomarkers, where we're considering endpoints. We're meeting end of this week, in fact, with a big group of 35 experts to talk about endpoints for Annabelle's condition. So these are the pieces that we're doing now, and then we're anticipating those kind of regulatory interactions and the pieces that come towards the end of the pipeline with the gene editing work. So these are my perspectives. We hope that as an organization we've added value, but I think we'll see what Alex's perspective is, because I think in many ways what I'm saying is that his perspective is most important. So thank you, Alex.
Yeah, I'll push back and I'll say that your perspective is also astoundingly important, and I don't think one trumps the other in any case. So the session is the driving role of patient organizations, and I think like 3 words kind of define how I like to think about that and what we've been able to accomplish with Rare Hope, led by Nina, so effectively that we want to see with every kind of program that we run, especially rare programs. The 3 words would be partnership, which you started off on your title slide, and then catalysis, and then ecosystem. And those can be really well managed in collaboration with these patient organizations to have some of the most effective outcomes we can, at least in my experience, for doing preclinical work that we use for proof of concept or hopefully IND enabling, and definitely further down the line as we look towards more definitions of what a more late stage program might look like. And to kind of back up the partnership aspect of this, this is a proof of concept manuscript that we wrote for in vivo prime editing. David detailed it in very accurate and faithful summary earlier this morning. But Nina, her husband Simon, and their scientific advisor, Dr. Natalia Morsay, everybody from Rare Hope, our patient partner organization, they're co-authors on this manuscript. And this isn't, a generous gesture. They're legitimate research partners, and they've done data analysis with us. They've actually run independent arms of this entire project that I did not have any personal direct supervision with. Obviously, we saw the data and reviewed it as partners, as we would with any collaborator. But they do research. They do guide research, and we converse with them, and we make plans on how we interpret data and how this will guide what we'll do later on in our work. To get a little bit of an idea of what the catalysis part looks like, in 2020, Rare Hope reached out to the Broad, specifically to David, and mentioned, hey, we have this rare disorder. We've seen your work. We're building an ecosystem. We're building a set of researchers. We've already sponsored some other work with basic biologists. We have clinicians in mind. Here's what we have. Here's a menu of collaborations that we can facilitate for you. Here's the mouse model that we're building. You should really work with us. And they made such a compelling value proposition to kind of fill in so many gaps that we'd have to kind of start from scratch with if we were trying to do this on our own. If we were picking an indication out of a hat, that we said, well, we can't say no, right? We need to take on this. We need to take on this project. So we did. And it also really aligned with how we wanted to start using some of our most newest tech at the time, prime editing, and kind of push it to its limit and define what the envelope was for that technology. So as part of that catalysis word, I mean, the second thing that they did was they helped us identify support. They were able to help co-apply with us for $2 million in preclinical research through CZI, which we're still eventually grateful for. And really, the whole structure of that award was as a patient-partner collaboration. And working with the outcomes that were guided from that framework, we were also able to see the utility of having patient-partner collaboration. We were able to be put in contact with clinicians who would guide kind of our experimental design, our in vivo partners, who we'll talk about in a second, and then more basic biologists as well that we wanted to work with for the whole study. And part of that kind of shakedown of how are we going to make this application, how are we going to move beyond just the first initial in vitro experiments that we run at the Lou Lab, we built this kind of one-to-one-to-one partnership for the core AHC research team that ended up being between Rare Hope, the Jackson Laboratory, and the Broad Institute. At the Jackson Lab, a team led by Kat Lutz at the Rare Disease Translational Center, and at the Broad, obviously David's lab. And again, this is a partnership that was, catalyzed by Rare Hope because they had actually already been working with the Jackson Lab independently in pre-work for a gene therapy. And because of that, they had already onboarded a mouse model. They had derived a mouse model for their project, and they had insisted that that work be shared to other modalities where it may be beneficial, like gene editing. And this is just a nice case of having that 30,000-foot view, as Nina mentioned, where we can repurpose work done for one modality for another. And having patient organizations, kind of force these interactions. And it wasn't hard for us to work with JAX. We know them. We like them very much. But kind of, again, the holistic view that we have from Rare Hope really helped. And with that, we were able to get some of our best data for our proof of concept study, where we show a dramatic reduction in seizure in the orange PE-treated mice. We're able to show a significant lifespan improvement. But the real reason why I put the data up there is because, on the seizure experiments that we were doing, these are hypothermia-induced paroxysmal events. This is a feature of AHC that really captures presentation that we see in patients. The design of that experiment was very much guided by the patient experience. The kind of outcomes that we wanted to see was guided by our patient partners, how we'd interpret that. And we got significant community feedback on how we were going to actually run these experiments, what outcomes we would evaluate, how we'd weigh that, and even in the design, the Rare Hope actually had a really important role there. The survival study as well wouldn't have happened with nearly as successfully as it did if we didn't have Rare Hope. So I mentioned they had done a gene therapy study in parallel, and actually before we had started collaborating for gene editing. As part of that gene therapy study, we realized there was a major survival defect in our model mouse that had to be resolved. So first, Rare Hope went to JAX, had the mouse model derived, found out that it's a very fragile model, and actually compelled them to rederive it as a hybrid model between two different mouse strains. And that solved it. And by the time that our in vitro development work for the Lou Lab was done, we were actually ready to use this hybrid model on a silver platter for us and have our experiment that ended up taking a year work pretty much in the first try and get that work done in a very timely fashion. That wouldn't have been possible if we didn't have this partnership with Rare Hope, if they weren't already trying to bring together and have crossover between different functional groups of modalities. Really, again, just the driving role of the patient organization of building an ecosystem where there is a menu of options that we have to choose from for our work. And finally, for this proof of concept study, we needed to do work in primary human iPSCs. We needed to do work with biochemistry to show some kind of functional connection between gene editing and behavioral rescue in the mice. And that was very easy for us to do because, again, an ecosystem had been built by Rare Hope where they had already done basic biology and sponsored basic biology research at groups like Al George's lab at Northwestern, at Kathy Swender's lab at Mass General Brigham. And they said, okay, here's a menu of these scientists that we have available to us. Why don't you go see who we'd best collaborate with? And we're able to reach out to them. Instead of these collaborations, I don't have to justify who I am because Nina's there to say, no, they're very serious about bringing this all the way across the finish line. And it's very easy buy-in and very easy for them to be kept on track, and all of us to be kept on track for the final goal, because not only do I have a say as a stakeholder, we have the patient organization having a role as a stakeholder to kind of keep us all focused on the ultimate goal that we want to move forward, and that's getting proof of concept in this case. And that's what we did. And in 2025, we were able to put this manuscript out with a dramatic proof of concept that we're able to do postnatal rescue using an AAV-delivered prime editor or base editor. And now we're looking to see for the future how we can advance that towards the clinic. Still working with our partners at Jackson Lab, still working with Rare Hope, and also now with Boston Children's with Tim Yu's lab, again, which is part of an ecosystem that has already been built from other modalities, specifically ASOs, that Rare Hope had initiated in partnership with some other patient organizations for Tim's group. So it's a nice background function to have them running some pre-work all the time, and really just doing a good job of being the high-level stakeholder that keeps us all working in the same direction and fills a lot of gaps. That way we can focus on what we're trying to do best, specific to our own technical expertise. So that is the driving role of the patient organization. Yeah.
Hello everyone. So I do not have slides, but I will make a short introduction about AFM Téléthon. So since we are here in Necker Hospital and invited by Imagine Institute, my disclaimer is that we are among the co-founder of Imagine Institute a long time ago, and we are still present at the board of Institute Imagine. And we are also founder and funder of Geneton, which we have created more than 40 years ago. So AFM Téléthon is a patient organization which was created a long time ago, but it's very well known in France, especially thanks to the Téléthon. We'll celebrate this year the 40th birthday anniversary of the Téléthon. So Téléthon is a huge event in France. Téléthon is on TV. It was inspired by MDA, so it comes from the US. It was adapted to the French, let's say, culture. And 40 years later, it still exists, and with a great success. Last year in December, we've collected more than 100 million euros, and that was record, and so it was a great success 40 years after the first time it was created. And it's not just about money, it's also about the whole country assembling against rare disease. So because in every second city in France there is some events that is organized to collect little bit of money, to assemble, to do some event, some party to collect money. It's also a way to communicate and to educate the whole population about rare disease and the difficult concept of gene therapy, hereditary disease, and all these difficult things. So it's really something very important and a very regular event in France. It's like the national day in winter. Some say about this. So, and also the fact that the whole country is really aware of this and participating to this event. It also participates, I would say, more largely to the health democracy. So it means that every year we go to the poll and the people say, yes, what you're doing is good, going in the right direction, and we agree to push forward to accelerate on this matter. So over the years, we have collected a lot of money. About 1/3 of this money goes to the care, so to the emergency for the patients, to the immediate care for the patients. Because as a patient organization, we are only patient or parent of patients. I'm myself a father of a boy with Duchenne muscular dystrophy. I volunteer for this organization. And we are driven by two very important things. The first one is general interest. Since the beginning, patients and parents of patients have understood that they cannot fight for their disease or the disease of their kids. They need to think wider and to think rare disease, because only at this level we could find some solution. Back in '87, when there was the first Téléthon, only a handful of genes were identified, and the gene of Duchenne muscular dystrophy was a trigger for the first Téléthon here, but basically we knew nothing about genetics. And at that moment there was a big family gathering, a rare disease family affected by SMA. There were hundreds of families who assembled and collected their blood samples. That was back in 1988, so we knew not much about the genome at that time. The important other dates coming just after is 92, when Genethon has produced the first human genome maps, and then the scientific community was able to work more efficiently on the genome. And in 1995, here in Necker, in the team of Judith Melki, was discovered the SMN1 gene. So these blood samples that were collected 7 years before, the first genome maps that were produced by Genethon thanks to the Teleton, conducted to the discovery of the gene. From then, we agreed as our patient organization to change Genethon mind, instead of genome sequencing, to move to gene therapy. And at the beginning of 2007, I think, I'm sure, Martin Barkatz at Genethon made the first proof of concept for the gene therapy against SMA, spinal amyotrophy. Then this proof of concept took a return ticket to the US, and it came back in 2019 as a regular drug, and with a name called Zolgen SMA, and a price, which was a bit expensive. But still it was a success, because as a patient organization, our driver is to provide access to the patient. At the beginning of 2000, we were missing still a lot. We didn't have production facilities. We were not able to really go from the laboratory to the patient. All the regulatory affairs were not also ready. So we have worked since then. In 2012, we have opened the production facility for gene therapy, which became a GMP production facility called Hyposkesi. We have also built, as a spin-off of Genethon, another company called Genosafe, which is dedicated in quality control for this very accurate therapy, because all this field did not exist. Everything has to be created, invented, and we were really triggering all this creation of this ecosystem in France. And maybe before I give the microphone and we have this exchange, because we are here in Necker, I wanted to stress, to underline that AFM Teleton has supported the breakthrough work and research led by Alain Fischer, which on XKID, which really was really a big moment in the history of gene therapy. So AFM Teleton supported financially this. And today, AFM Teleton is proud to support Marina Cavazzana on her clinical trial, about to start clinical trial against sickle cell disease. And the difference between back at the year 2000 with XKID and today with a sickle cell gene therapy, we have built all this ecosystem. So the drug that was discovered by Marina Cavazzana was able to, travel also not far away, but this time to Évry, to the suburbs of Paris, to be optimized, to be vectorized, and to be produced by the facility and controlled by all the ecosystem that we have triggered and that we have contributed to create, and to make the difference and to put France as a leading part in Europe, but also in the world in the domain of gene therapy. Yeah.
Yeah, that's all. So I just wanted to say that Alexander Souza is a leading scientist, experimental scientist in David Liu's lab, because we didn't have a chance to introduce you. So it seems that what I find interesting here, both in what on the US side and French side was said, is that there's this close connection between the scientists and the patient organization. So Nina, because you didn't come at all from a scientific world, so what was your experience entering in a scientific lab, and what did you learn, and what did they learn from you? Because these are very different perspectives. And did you evolve in your way of thinking about science and how patient organization should actually help scientists? And how do you find the balance between being the parent of a sick child and having enough distance to also be able to think more broadly and beyond the personal story.
Thank you. I think that's a good question. My background, so I mentioned this when we spoke previously, my specific training was in masculinity in 17th century Bermuda. So not a terribly relevant topic for the world that I inhabit now. My husband and I, when Annabelle was first diagnosed, my husband started reading papers, and he would read a single paper, and it would take him 24 hours because he would understand every single concept in the paper. He would, whatever came up, and there wasn't ChatGPT for cheating back then. It was just full-on research of every concept, every term, and there are so many, obviously, in this world. And I was a little bit slower to get involved. I started with fundraising and other things. And then, of course, I thought I could do it better than my husband and dove right in. But I think for me, doing enough reading to make informed decisions was the most important piece, because I didn't want to go in and kind of give advice or try to diagnose a problem when I didn't actually know enough to have a credible response. I think at this point, and Alex might disagree, but I think at this point I am able to kind of make decisions about how we deploy very scant resources, and that's the level of expertise that I wanted to get to. In terms of maintaining, an arm's length distance emotionally from this, I think, it's my coping mechanisms are gym and working a lot, so it's very compartmentalized for me.
So,
I could also from science, I think the first thing I've learned in science, so I'm a bit provocative, but I would say that it doesn't work. So this is why it's slow. So when we see the result on the slides, it's extraordinary. You have the path, it goes from A to B, and it's straight line. But science, this is what I've learned, is not at all a straight line. It starts, it doesn't work. You need to go back. You start again. It doesn't work, and it's a very huge and long process. And I I admire scientists who have worked for decades on sometimes the same disease, and now sometimes finding possibilities to tackle a disease on which they have worked on their PhD or postdoc. So that's this is important for patient organization to be aware of the difficulty which lies behind science, and it's also explained why it costs money. And this situation I say was because I think it tends to not to disappear, but with a new tool, with the learnings we have made over the past decades, things are going faster, and this N of 1 process and the 10 months process to build a drug is getting possible today because we have learned about how this ASO work, about all these things. So this mass of knowledge that was assembled over these decades are really modifying today's science to be faster. So even though this doesn't work, a remark that I made is still valid, I think this is really going in a good direction, and especially because this slow process for patient organization is driven by our emergency to find a drug. I mean, we are assembling people who are affected by a very severe disease, who have death at the heels, and they want to be faster, to find the shortcuts. And I see that, and scientists work on accelerating, on going faster, and using all this knowledge that was collected to really improve the process to move faster. And this example of this clinical trial in preparation with the work done by Marina is a good example, because Marina made a discovery for a proof of concept, and then it goes to some other people who have other expertise to bring it to the next level with optimized vectors, which 20 years ago we wouldn't be able to do. So, yeah.
So, all pointing out is a little bit a division of work, a division of tasks between the patient organization and the physician, and the scientist. But you could also maybe argue, I'm going to challenge a little bit, that maybe people don't agree at all times between, you know, people who lead scientific institutions or scientific labs and the patient organization or the patient point of view. And so there is also, I think, learning how to build trust and give space. And so how is the decision making done? And for instance, at IFM Teleton, there is, you know, how are the priorities given? Who has the final word? And how is the balance done, you know, beyond actually distributing money, which has huge consequences?
So the first thing is that in our priority is to give access to drugs to patients. So we don't fund research for research to write nice articles. It really needs to have a target to modify the life of patients. So that's really the first driver for us. And this is why we don't fund all the labs, but only those who really are going into our direction as well. The decision process here in our organization, but I think it's the same in all patient organizations, we are not experts ourselves. The work that is done in the laboratory are very accurate, very complicated. So we have next to us a scientific committee. In the case of IFM, it is made of 80 experts who are here all year long and who rely on a network of 5,000 experts in the world that can be contacted to give their expertise on a project that is proposed for or submitted to ourselves for finance. To give you an order of magnitude, we found about 200 projects per year, so it's a massive effort just to assess those projects and to decide which one we will fund. So so there is this expertise or expert assessment that is done by a scientific committee, but the final word is on the patients, and sometimes we decide to prioritize one instead of another because it is targeting an ultra-rare disease that has no therapeutic option, so this can be a priority. Or it's a highly risky but highly innovative strategy, so this we like. It's a high risk that it doesn't work, when I refer to what I said about science. But this is really what will make the difference in the future, so we are aware of that, and we would give priority to this kind of projects.
Just for Alex, because I think you said something important. You were trying to say what Nina and her organization was bringing even to the science and defining endpoints, helping you design even experiments, it seems. So maybe you can tell us more about that, because I think this is not something that's ingrained in all mentalities and in all labs, to have, you know, patient organization within a lab and participating scientifically.
No, I think one of the best decisions we made when we started working with Rare Hope was unifying the science meeting across modalities. And for us, that meant that we were joining the gene therapy program monthly meetings that had already occurred for, I think, about a year, 2 years prior to you guys starting to work with us really intimately. From that, we were able to hear the progress they were having on mouse model characterization. As I said, they remade a mouse model halfway through that program. We were able to hear about some of the readouts that they were designing. And that was just such an astounding efficiency gain for us to be able to tap into that as we were still doing our in vitro workup for our editor design prior to actually making the first leap of faith into our first mouse model experiment. And then as we started designing those experiments, you know, when we start moving away from the things that are just strictly gene editing in vitro, we outline how we want to run these experiments and what the design of them might be. And at those meetings, we meet with our in vivo collaborators, the Jackson Lab from Cataletz's team. We have Nina's there as well, and so is Nina's science advisor, Natalia Morsai. And what we would find is on a monthly basis, we'd probably have an argument about the precise design of the experiment. And that's a good thing because it really shakes down presumptions that, I would personally make as somebody who is not an in vivo mouse neurologist, or presumptions they would make as somebody who is not an expert gene editor and has concerns about what materials we'll get as a readout, or presumptions that either of those two groups would make as people who don't ultimately need to be the end user for an envisioned product. And if there's some other tangential readout that we can get, be it a seizure assay, be it some kind of recovery time period, things that maybe we would look past blindly because we're just not thinking about what additional information can we extract from this assay that will be a minor add, but will be so informative for the patient community or so informative for some other kind of trial design in the future, that we should do that. I mean, and that's a great place where Nina and Nina's scientific advisor Natalia and Simon come in to kind of bring that all back to us. And then as we have this tech develop and we see that it's working, as well, Nina started to push us to collaborate with people outside of that core working team to make sure that we have some other orthogonal readouts, such as in vivo electrophysiology teams at the University of Utrecht. Like we're trying to do that now because there are additional learnings that we can have there, and we wouldn't necessarily go after that because we're focused on our core competencies. But she's been to these meetings, she has made contact with other people, she's introduced us to these teams at this point, and now we have the ability to include that in our research strategy because it is clearly an important aspect of the research to prioritize per our consultation with the patient organization. So that's where a lot of that comes in. And then again, the pre-work that was done prior to interaction on just establishing a gene therapy study with other scientists is also essential for enabling us to have the most efficient step forward.
Very interesting. It's unusual. Maybe I'm just looking at the time to 3:30. Yeah, maybe one last question. Maybe the audience, if somebody has a question. I have one question then for you. It seems that, something that has been said a lot this morning was about considering rare diseases as an entity, and also how regulatory mechanism could influence advances. So what do you see your roles in the US and in France as influencing or helping shape the regulatory system towards rare diseases?
I think that's such a good question, and I think what our role as an organization can be in the future, it's something that I grapple with, and I think thinking through to our upcoming regulatory interactions that are hopefully on the horizon, thinking through how we can structure an IND to include all mutations that cause AHC. There are many mutations in ATP1A3. There are a number that account for the majority, but there are all these other rare variants as well. Thinking through how I can maybe suggest to the Broad folks, let's not just think about 5 mutations, let's think about 20. Let's expand what we're thinking. So each step that we have, I think, as early adopters, as early navigators of new, hopefully loosening regulatory restrictions and a hopefully increasingly broader acceptance of basket trials and thinking through multi-mutation strategies. Hopefully that's one immediate way that we can have some effect. But otherwise, thinking about common biomarkers that might be developed across conditions. I think that's something that's certainly of interest to us, whether they're blood-based biomarkers, whether we're doing an EEG biomarker study with a collaborator in Leiden, for instance, that's also being advanced in Dravet syndrome. So there are these, by developing tools across different conditions, I think that's one of the ways that we're also thinking through how those component pieces, putting those together for multiple related conditions might be useful.
Yeah, I would say that there are 2 aspects to which are really different. One is the regulatory itself, and which deals more about science, about clinical design and basket trials, for example. The second one, which is important because for our role is to have access to patients, is also the price, so the HTA that goes behind it. And here we advocate, especially for ultra-rare disease, for a non-for-profit model because there is no economic model, so you have to be practical. So we need to find ways to get able to bring those drugs to the patient, even though it costs money to produce, to develop. But we need to find way, and we advocate also on this matter. So the regulatory part is for part of it, scientific discussion, so in which we take part because we stress sometimes the emergency and the gravity of the disease, but on our everyday life, not just digit on the paper, because some are life-threatening, so it's kind of the ideal scenario because you can give any kind of drug, it doesn't matter because it works, it's wonderful, it doesn't work, anyway the kid would die. That's the easiest scenario. But we have many diseases which are very severe, that are fatal, but after a few years, you want to try a drug, but the regulatory authority will say, we don't want to take the risk because it's not that bad condition. And here patient organization can come, step in and say, okay, but the risk is not so high, my kid can just get a cancer and it doesn't matter, this can be cured in another way. So, well, this kind of discussion, patient organization can really make the difference as well.
Thank you very much. So I think we need to give space for the next one.
This is our last session for this afternoon, and it's a great pleasure to call back Dr. Winston Yan. He had double competence in academic, but also as an entrepreneur. Okay, it's better now? Okay, thank you. It's the last session this afternoon, and it's a great pleasure to call back Dr. Winston Yan. He had a double hat, a double competence in academic and science, but also as a bio-entrepreneur, and he will explain and talk about the Center for Therapeutic Genetics. Thank you.
Thank you so much. So I get the pleasure of presenting on behalf of David and Tim, and I'm ending a day where there have been many references made to the Center for Therapeutic Genetics, and it's also one of the last talking sessions where we get to synthesize the things we've heard into a new model of how can we operationalize, sustain, and then scale this. So this is actually a new talk that I put together just for this. I think for this particular audience with the Imagine Institute and Necker and Genathon, it's really remarkable, and there's so much we can learn from you in scaling it up. So I would love for you to shift scientific hats and into thinking more about systems and how we can build things together. And please come up afterwards and give feedback and how we can either work together or make the ideas that we have here even better. Okay, so here's some disclosures. As I mentioned, I worked in the CRISPR genome editing lab. I worked actually then 10 years afterwards in a CRISPR genome editing company, taking it from the initial discovery to the actual clinical trial. And I think it really, for me, it's just the fact that we have this amazing modular tool as genome editing, but it can reach so few patients, it did not feel right. And so pretty much 5 years into that entrepreneurship experience, I reached out to Tim and said, why can't we do what you did with ASOs, with CRISPRs? And he just said, Winston, ASOs are it's the regulatory foot in the door. It will help unlock the thinking about this whole field of interventional genetics, everything that you heard Tim present. And that's where we started working together on the N of 1 Collaborative. But finally, for all of the reasons that you heard today and with David and Tim and Alex and Nina, the pieces are coming together to make possible what I think is a new model for developing therapeutics in a procedural way. So I get the chance to reuse some graphics and go a little bit deeper. If we look at this plot, this was a plot that really indicated that the dinosaur that Tim was describing, that long tail is not addressable. And mapping onto it, a couple of patients that David mentioned. So sickle cell is it's commercially viable. It has enough patients to justify the commercial investment, and many companies are crowded in there. Chronic granulomatous disease, barely, I would say, commercializable. And then you have the true N of ones, like baby KJ. I pose to you that there will be way more N of ones like baby KJ that will come out in the coming years and decades, and ones that already exist. And we need to find a way of treating that long tail in a repeatable method. So industry actually is structurally constrained in solving this problem. If you think about how industry moves, that left to right diagram that Tim talked about, the reasons why that are the case are you have to start with the largest populations and the financial opportunities. There are returns to scale, right? You can optimize the unit economics and push down the costs of manufacture, push down the trial fees, but ultimately it's a slow progression from that left to right. And it's really not going to get us there in time. So the question I pose to you is, what if rare disease treatment could actually be delivered as a procedure instead of as a product? And the idea here was shared by Tim earlier of, take two children born at the same time, one with an anatomical cardiac defect, one with a genetic defect. One gets a choreographed team of experts who descend upon it. They produce a treatment that is tailored to the individual, but based on standardized practice. And there is then follow-up by the care team in an academic medical center. And then right now on the bottom, we have to wait 5 years and probably 10 years, $100 million, $500 million for a drug to be developed. Could there be a world where we actually have interventional genetics or genetic surgery do the same thing? I think it's not so, one question is why the name therapeutic genetics, right? And this is a thing we've thought a lot about of why is it differentiated from gene therapy in the traditional sense? Well, it turns out medical specialties have had a way of moving from observation and diagnosis to actual intervention. I joke, interventional anatomy is also known as surgery. First is just looking at structures in the body, and then you can do something about it. Then you have fields like interventional radiology, interventional cardiology, where it was first a diagnosis of looking at a film, looking at an image, and then being able to intervene with it with stents or minimally invasive surgeries. And now we go from the era of diagnosis with first pedigrees into exomes and whole genomes to using the technologies that were described earlier with both genome editing and base and prime flavors as well as ASOs. So I think it's more than just a cute name. There's actually important ways in which the connotations inspire new financial models where we can sustain rare disease treatments. So the key thing also to understand is that this is an analogy. It's not literal surgery, right? And the desired connotations we have are surgeons are incredibly skilled at what they do. It's a precise manipulation of a pathogenic thing that then gets corrected and helps the patient. It's a trusted discipline. There's a benefit-risk conversation that's very tailored. It also is discussed in close consultation with the patient. And ultimately, this is a care model that exists in hospitals everywhere. Procedures make hospitals money. What we don't want to say is that this is somehow oversimplifying this entire endeavor, that all of the work that you just presented is just wrapped up and it's like, oh, we're just going to do it like surgery. Not at all. And we also do not want to overpromise. So those are important caveats that we pose to you as a sophisticated audience to know that these nuances are there. So we have the tools for genetic surgery. We described with Tim and David in both their presentations the vast number of clinical data that have emerged to show that these modular programmable medicines can actually be tailored to patients' genetic mutations and, importantly, in time to treat the patient who is presenting. So this now gets back to the plot. Why do we think we can catalyze these treatments for the long tail? Well, if we go with therapeutic genetics, if we start at that right end of the plot, let's consider that patient who presents in need. A surgeon, they treat the patient who's presenting to them, right? That speed and the regulatory accelerations are driven by benefit risk, by the severity of disease, and by the platform efficiencies that everyone has just described. We will build templates and reference data so that we can feed off of each other and every single operation becomes faster, safer, cheaper. And that allows us to, carve that tail from the bottom up.
where as the infrastructure matures, we have these platform efficiencies that are able to treat more and more patients. Maybe I'll give you just one example I read about in the history of Necker. So 1952 was the first kidney transplant done at Necker by Jean Amberger, right? And can you guess how many kidney transplants were done last year, or in 2024, around the world? Yeah, that's pretty close. 110,000. 110,000 in the span of 7 years. And that is the type of scaling function that grows over time, but that's actually what we need to reliably, repeatedly treat lots of different angles of genetic disease. So you've seen the slide. I will just say that in addition to the institutions, it is the people that drive everything, and it is an incredible collaboration. The people from the US that you see here are just, we are in it with our hearts as much as our intellect. And there is this collaborative spirit that has, it just makes it fun to work every single day. So that's what I'll add on to the slides been shown. Here now is going to be the crux of the talk, where we have these 3 different strategic pillars that we think drive this flywheel that will help make treatments safer, cheaper, faster. So the first is actually making the treatments. We need to make genetic medicines. We need to make them rigorously while using that opportunity to build the templates, the infrastructure to go faster. So with those, now we get into this part where the template and the data that are generated actually can inform regulators to streamline practices in a data-driven way. Our goal is first to just reduce the time and cost of normal filings, but eventually get to what we call an accredited center for therapeutic genetics, an actual center that receives like a certification, just like CLIA, to be able to do it and not have to go back to the regulators every single time while still being subject to oversight. So then that streamlining needs to be paired with reimbursement. And we think that procedural reimbursement is the way to go. The sustainable revenue cannot wait. Like a hospital and departments cannot put out $5, $10, $50 million for treatment up front. They need to be reimbursed for the work that happens in the workup of a genetic disease patient, in the non-clinical development, and eventually with the treatment. But that needs to happen in lockstep versus a money up front and then marketing authorization. So that's our goal too, point of care procedural reimbursement. Then the last point at the bottom is critically important for us. Like what was done with kidney transplants going from 1 to 110,000, sharing data, sharing knowledge, the best practices, that is how we enable the entire field. We have no just thoughts that we are single-handedly going to help make a dent in the 300 million patients worldwide with rare disease. We need everyone, and that's why we've been so excited to come here and collaborate and see the great work that's happening. All right, so double-clicking into how we're actually going to develop programs. So the first is it's actually interesting seeing all of my prior speakers' slides sharing this pathway, right? We're all going from identifying patients, going through a drug development process. Mine is a bit more of a layperson abstraction. We're essentially designing it. We have to test it. We have to make the drug. We have to treat, and we have to follow up. So that's been the case for both N of 1, it's in ASOs with Milasin, as well as baby KJ's treatment. And if we take that end-to-end process, the 3 different institutes, we partnered together because we all fulfill different parts of the puzzle. The Broad, we have the incredible cutting-edge technology that we are the sort of the frontier, frontline users of it. We have the platform expertise, and we have access to the foundational intellectual property. The Jackson Laboratories, the Rare Disease Translational Center, they're industrializing the generation of animal and cell models for these rare diseases. Many patients, their first call is actually to Jackson Labs, and Cat Lutz in particular, because they've been told they need to make an animal model of their disease. And so we are so grateful to be working with them to make that process turnkey. And then Boston Children's, for all the reasons in Tim's talk, just having them as a clinical partner with the care that they provide for rare disease patients today and the thoughtfulness with how that care gets extended to tomorrow, I think is incredibly important. So then the public-private partnerships, right? I'm glad we agree that there's that academics cannot do it all. And in particular, coming from my own background in industry, I see the real value that, your industry partners have spent money on and learned. In particular, manufacturing, quality control, those are things that, at least on the US side, they don't often exist in industry, in academia, like you guys have with Genathon and Necker. So these are our partners because they've been pushing the front lines of their commercial programs. Why can't they share that expertise and help us learn? So that is actually what a few forward-looking industry partners have signed up to do, and we're very grateful for it. And then data sharing, as we said. It's not just holding on to the data ourselves, it's sharing the practices, sharing the resources, and there's all sorts of partners who this is what they do, and we partner with them in order to be able to share practices with the rest of the world efficiently. If we really think about roles and responsibilities, right, in the beginning, with the selection of the indication and patient choice, it starts off with all three groups deciding it's essentially a program team in drug development where you decide on what is the indication and the approach. Then the preclinical screening begins with the BROAD, and then there's an iterative cycle with Jackson Labs, the RDTC is the acronym. And then the BROAD helps lead the CMC program as essentially as a functional leader, just like, again, in a drug development team. The clinical teams at Boston Children's Hospital take on the clinical development as well as the design of the protocol. And then, honestly, we all are going to push on our respective insurance and Medicare, Medicaid partners, and just to make sure that a reimbursement pathway exists for the care that we hope to deliver. So this is just a schematic to say that we also hope to build definite infrastructure that can be reused across the entire pipeline. I won't go into detail here for time. And I think this is actually probably the more important slide of saying all CTG programs, our initial programs, are intentionally built on delivery and editing platforms that have validated safety and early efficacy from phase 1-2 trials. It doesn't matter if we do them or it's been done in industry. That's important so that we are not just doing first-in-man trials of new molecular platforms, but we're really taking something that's already been validated and or at least has safety and early efficacy, and we're turning that into a modular platform to treat more and more patients. So I think that's also a subtle difference from some of the pioneering work that's being done at in Imagine and Necker, where you guys are taking the bold first leap into the that new composition of matter. What programs are we going to do? Here's where we lay out the different tracks of, really they correspond to the translational readiness. Track 1 is the ones that might seem very obvious. Roman presented it very well. We have full platforms that are already validated with clinical data, and they can be reprogrammed to other genetic targets. So that's in vivo liver LNP editing, and then ex vivo HSC editing. Then track 2, it's going into places where literally the two separate components of the editing payload and the delivery vector have first-in-human demonstrated safety and effectiveness, but they have not been put together in a vectorized format. Industry tells us that it's just too risky in this funding environment, but we think that there are patients that can be helped today by packaging viral vectors with gene editing platforms. It may not be perfect, but it allows us to help patients and begin that flywheel. And track 3 is a very promising one that David described in his talk earlier of, can we use single compositions of matter and actually expand the one-to-many treatment space with a single molecular entity? It really will build on top of track 1 and track 2 clinical experience to take those same reagents, but targeting a tRNA sequence instead of a gene mutation allows you that one-to-many. So these fit together. They have different horizons of maturity. For us, I made this quick slide based on a meeting we had on Friday where we did a quick review. I wouldn't say a quick review, an initial review of patients at Boston Children's Hospital that were eligible from their genetics department, specifically the metabolism unit. And we had 12 clinically eligible patients that were essentially brought to us by a clinical collaborator. She's the head of the director of clinical activities for metabolism. So these all had confirmed diagnoses of severe metabolic disorders. They were pathogenic variants that had clearly identified. They had no treatment previously or no standard of care. The standard of care was not sufficient to them. And the liver, an interesting thing is that you can just have a proxy of could they be treated by liver transplant, like in the abstract, right? So these patients either declined liver transplant or they're waiting for liver transplant, and so they still have not had their. I mean, a liver transplant is essentially a 25,000 gene therapy, right? Like you just take it out and it's a wholesale replacement. So of those, just our first pass, we think there's 4 patients who have actionable editing just based on frontline. What we call frontline is like the most validated and canonical base editors. And so we're doing the further clinical exploration in progress, and we think that the others have paths through base editing optimizations of different PAMs, different windows, or with prime editing. So you see that in doing this process, we already are building a workflow that now this clinical collaborator knows that when she has a new presentation, she is able to come to us and say, how is this an editing amenable patient? So again, just changing the mindset of saying, is there a theoretical mutation out there that we've read in literature to here are actually the patients who are presenting to us and who are in need of treatment. That is that surgical approach that I'm trying to make clear in this presentation. So I actually modified this slide literally in this talk because I was like, oh, well, you guys have an amazing history of doing this work. And then voila, like truly all of the editing indications and everything was laid out so beautifully earlier. So it sounds like you're already on it. I urge you to think about these NFUs not as, oh, they're tiny populations, but they could be truly the templates that are developed for the precise correction. And then that allows you to just hit many more N of 1s and bundle them together versus thinking about it as, they're just too small to justify your time and effort. All right, and I think I really do want to cover this because it gets into the exciting work we're doing in the track 2 of now combining a blood-brain barrier penetrant IV-delivered capsid with a base and prime editing to be able to treat genetic diseases of the brain. So maybe I'll cover this quickly before I get into the other 2 parts of the triangle. So we are making a platform from a BBB crossing delivery of gene editors, and we're doing that as the first set of indications in DEEs. So these are essentially pediatric epilepsies, DEEs, developmental and epileptic encephalopathies. The delivery is with IV delivered. It's a transfer and binding AAV capsid developed by the Deverman Lab. It's called CapX. There will be first in human data in this, hopefully later this year or early next year. We have payload as base and prime editing, as we've discussed. The indications, that is where we carefully thought about what are indications that you can have, neurologic indications that you can have a good readout that allows you to see whether your target engagement or efficacy is occurring. And for those, with DEEs, you can measure seizures or global severity scores, and there's often a fair amount of natural history that's collected, thanks to folks like Nina, who have been so diligently collecting that background data. So the goal is that we have a first in human in 3 years, and we're already starting to get the regulatory platform pushing the FDA on it with one submission enabling 2 separate disease genes and programs. So we'll have a single CMC strategy, single GLP tox study of just the actual editor with a precision editor, but then be able to treat multiple alleles within a given gene, as well as multiple indications later. So I've talked to you a lot about how we hope to instantiate the all-important programs, but now let's think a little bit bigger. What do we need in terms of, I would say, a streamlining to our regulatory thinking? So our experience with regulators is they honestly want to help, and they really are collaborative in the discussions and thinking. And for us, it's putting forth what are the, as end users of regulatory pathways, what are the rub, if you will, the friction points that come up. So if we think about two ends of the spectrum that are being used today, on one side you have traditional product approvals. So that's marketing authorization. In the FDA, right, like your approval means you're free to market it, and then you can sell it, and that's ultimately where you get back your revenue. It's really not designed for these ultra-rare populations. We have limited evidence generation, and the patient feedback to us and to the regulators this is over-regulated. It's expensive, it's slow, it's not proportionate to the benefit risk. And at the end of the day, because you're too slow to treat the patient, it does not actually protect the patients. It's too onerous, and it doesn't treat the patient in time to help them. Then if you go back to the other end of the spectrum with compassionate use, expanded access, these programs are, in Europe, you have named patient pathways. This gives you rapid access at the discretion of an investigator. My understanding, and I was actually corrected a bit with Frederic, is that they are not economically sustainable because usually there's no reimbursement path, or there's no marketing path certainly, but there can be some cost recovery or reimbursement. I pose that this could also leave patients at risk because we're putting too much responsibility on individual investigators, and they may not have the, if we just treat it purely like surgery and it's locally regulated, there may not be oversight of it from still an emerging technology. So could the middle ground actually be accredited centers? This is like CLIA for personalized, developing platform medicine. And we think that it balances both speed to treatment with the regulatory oversight. You end up having platforms that get green lit for, by regulators saying these are valid, interventional genetic, therapeutic genetic platforms. It allows you to, once that's declared, what Tim said, you can do the do and tell of without having to check back with the regulators each time just to propose new programs and start collecting the data. Those data ultimately help us iteratively refine the regulatory approaches far more quickly than if we had to go back to the FDA every single time and iterate that just in practice. And we have already precedents in accreditation with transplant surgeries like bone marrow transplants, CAR T. So it's not inventing something new, and I think it's just slightly reframing the thinking of how do we develop precision genetic medicines. I won't go into the details here, simply to say that we have the inputs. The accredited center does a few key activities of developing the treatment, of course, maintaining the accreditation, developing the treatment, and then coordinating the payer reimbursements. And then at the bottom here, really indicating that the regulators have audit rights, right? They can come in at any time. They can, there's compliance monitoring, there's just iteration on the insights. That all could provide a system to really work hand in hand together to push these new treatments through. And this has been discussed with the FDA. There's a friend of ours who has some influence on White House. He's an investor, but he wrote an article with a catchy title. What we learned from CRISPR is catchy acronyms are just everything in CRISPR. So Standards for Advanced Genetic Editing, or SAGE, and really this is meant to be even pushing the regulatory envelope beyond the FDA platform proposals with plausible mechanism and the platform designation. So that's the proposal for streamlining. How do we make money on this? And I have to be very cautious with saying that the goal is not to make money. The goal is to make money so we can sustain ourselves and treat more patients. That's sustainability. It's not necessarily commercial success. So I'll describe to you why I think procedural reimbursement is important. If we look at the process of baby KJ happening, that still required significant upfront investment. And I would bet that also the programs that Genathon is working on require significant upfront investment. And thankfully, AFM is incredibly supportive, but that may not always be the case for countries worldwide and hospitals even within France. So with product reimbursement, it's a risky bet that you put money up front and that you have something that works with sufficient benefit risk to be approved. Procedural reimbursement is that you get paid for the activities that you do. And those activities are defined by what your peers and the process that is established. So, for example, if we evaluate a patient and perform the early gene editing screening in immortalized cell lines, that can be a cost that is reimbursable, right? Just like other procedural specialties of pre-surgical, preoperative workups. And then finally, when you do actually provide the treatment, that's when you just still have gotten continuous reimbursement along the way to cover your cost. So we think that this is going to be even more of an issue with, or I would say like as these, I call them addressable populations, become smaller and smaller, right? We've gone from gene to now individual alleles. It becomes less and less attractive to actually invest in any given therapeutic product. And I think the urgency becomes much more to invest in the process of development to keep those costs down. So for CTG, what we think about is that we need to start with short-term investment in order to prime the pump. It is still expensive to develop these treatments today. We'll start with philanthropy, donors, nonprofit grants. There are these cute things in the US called priority review vouchers that we can sell off to industry if we get approved drugs. And there are cost recovery methods under IND. Once we establish that rigorous procedural approach, and streamline the regulatory pathways and start getting the reimbursement for real in a procedural way, that allows us to get to our long-term revenue model, which is just the procedural reimbursement from payers directly. And then I think we also know that there is platform value to be created, but that's not necessarily a guarantee, especially based on tiny programs. So yeah, the last thing is just what I emphasize, that we're trying to be sustainable, it's like how a hospital department operates, right? Not trying to be lucrative, which is how a biotech, a venture-backed company might operate. Okay, so thank you all. This is, I really do think we are meeting at a pivotal time in history. We've heard it, we feel the momentum, we are doing the work. And I think it's not just the science, technology, and medicine. I think there's increasingly a lot of regulatory interest in how can we update our methods to meet the needs of patients. So I think that's very important. To me, reframing genetic medicine development as a therapeutic procedure is really just a lens for rethinking how the problem could work and how sustainability could come out of that based on existing models that are paid for and patients do get treated, right? In a hospital, you have drugs and you have procedures. Both of them help patients. We've focused a lot on making more drugs. Can we actually refactor as more procedures? And we've now described the Center for Therapeutic Genetics. For us, it really is the labor of love of how can we come together and build more treatments for patients, but start the flywheel going towards regulatory streamlining, reimbursement, and then share that as a template for others to follow if it makes sense. And finally, I'll say that we are in our early days. We really will need a collaborative global effort to capture the full benefit of these powerful platforms that we've heard about today. So thank you. I hope that we can live in a future where genetic diagnosis, every genetic diagnosis comes paired with a genetic treatment, and would love to work together to make that happen. Thanks.
Thank you very much. Thank you. Is there any question or comments?
Yeah, thank you for the talk, very inspiring. I had actually a question, like practical question on the collaboration between different accredited structure. Like if we project ourselves in like 5 years from now, I mean, we may imagine that worldwide we will have not one genetic surgery center, but maybe more. And so in the idea of streamlining and reducing the cost, the general cost for this kind of approaches, do you think that at one point we will be able to transfer knowledge between the different accredited centers? Because as you said, I mean, N of one can be N of few if you look at it globally.
Absolutely. I mean, I think that's actually a necessity before 5 years' time. It needs to happen today so that we are both looking for the patients. We're all generating data in similar ways so that they can be really analyzed in one unit. So that is, if we think about all these centers existing, that was one of the purposes of the N of 1 collaborative, is that it is the overarching, it's like the professional society, the surgical society, if you will, that can help with the knowledge sharing, the protocols, the data sharing, as Tim had described.
Following up on what was just said,
Yes.
I was wondering, because one says that, but then there are practical matters, right? Because if you want that a regulatory system in another country or another continent accepts a trial from another one, it means somehow that the people coming together work closely with the different regulatory agencies and come up at least with an agreement about, what should be done for each study in order to be easily validated. Yeah. And then my second question is, one says, if a delivery system has been validated, an editor has been validated, then we don't need to validate it again. But I don't know if the people in the regulatory system are convinced of that, and what data would need to be generated in order to convince them that this is the case, and when is it not the case? When would one need to go back and do the whole?
I would actually love to have that conversation on the next panel. I'll say two quick observations. One is that I think the method of the accredited center, maybe stepping back for a second, approval just is a very sharp black or white line in the sand, right? And so if you have an approved drug and then you do find there's some, AEs, adverse events that occur afterwards, or it's not quite what you think, you're updating the label, but it's really hard to unapprove it. Whereas I feel like with accredited centers, you can update your accreditation, your practices more live, and you can accrue those data over time to learn the things that you're describing. Two regulators thinking about how modular these data are, FDA previously had some very archaic, you have to cite, in the drug master file, they have to get access to it, regulatory access. It wasn't very easy to actually use platform data, but they just last month released, as a part of the plausible mechanism, how you can reference data. And it's actually much more streamlined. It's in the way of scientific publications, in the way of, reference access from partners. The real question will be, do other agencies believe it? And you're right, is it acceptable? That's another question. But at least on the surface, they've given interest in making it much more practical to use.
Thank you very much. We think we have to move to the next panel. Thank you very much for this.
Thank you. So we should have online Dr. De Vries from the EMA.
Yes, I'm here. Can you hear me well?
Oh yes.
I'm really
Great. Great. Can you see us?
Yes, I can see you. Good afternoon.
Okay, thank you. So it's really a pleasure and an honor for me to chair this roundtable around the regulatory aspects. Winston just gave us what is much more than an introductory perspective. It's really it's it's really a change in paradigm shift that he's proposing here, and I think it's really an interesting theme to this roundtable. So we have the pleasure and the honor of having Professor Pogam Burtz, who's the head of ANSM. Somebody, which I will not name, earlier this morning told me, "Oh, so you're having the head of the ANSM? So it's like the head of the FDA?" I said, "Yes." And so, yeah, really, thank you for taking the time. We are also having Dr. Émilie de Vries, who's at the CAT, Committee for Advanced Therapies at the EMA. The CAT is in charge of analyzing the dossier for the drugs before approval, and ATMPs, advanced therapies before approval. And we have Winston. No need to present Winston, who's also the founding president of the N of One Collaborative, which aims to be a professional society for those developing individualized medicine. So, and his experience, of course, and his overview coming from the US is really interesting, in this discussion, in this transatlantic discussion. So, maybe if we can start by you, Professor Pogam, can you, for both our US colleagues and for those in the audience who are not that much versed in the regulatory aspect, so what's the role of the ANSM in the perspective of authorizing new clinical trials?
Yes. First of all, good afternoon, everyone. So it's a great pleasure for me to be here, and I have the honor to be at the head of the ANSM, the French National Agency for Safety of Medicine and Medical Products, it means medical device and IVD, for the past 2 years. So I have to say that I am truly impressed by the audience. I am not an expert on genomic medicines, but I have heard the word regulators a few times. I think at least 20 times since I'm in the room for the beginning of the afternoon. So to be honest, at one point I thought maybe I should leave, but then if I'm here, it's because ANSM team and we do share the belief that the value of exchanges and for advanced access to treatment for the patient are really key things, and with dialogue you can do a lot of things. This is not only words, this is the way we want to work in the ANSM. Some of you can have maybe just check this for real, and so that's the reason why I accept to be there. ANSM, regarding clinical trial, is responsible for authorization on a core, let's say on the core mission of the ANSM is responsible for the authorization. As you know, most of the clinical trial related to medicines or medicines are declared in the European file, the famous CITES, and register for all medicine studies. And you can just going from phase 1 to phase 4 studies. We are working in duo with CPP, so ethics committees. This word has not been pronounced yet, but these are part of the authorization in the European landscape. And at the end, it's the approval of the ANSM, even for multinational, mononational, or multinational at the EU space. So the way we are working, if I can just try to summarize it, it's always a word that I have also already heard a lot of time. Everything is a matter of benefits and risk ratio. And this is true also when we are considering the safety, the quality. It has been said that pharmaceutical quality is an important matter, and it is an important matter. The safety also of the individuals participating in the studies, and also the compliance with good clinical practices. And our role does not stop with the approval. We are going further because we are also responsible for all the surveillance after the beginning of the enrollment. Pharmacovigilance also has been long said, but it's during the clinical trial phase, and real-time vigilance with notification of any serious adverse reaction or event that could compromise participant safety. And so we are able to amend trial protocols, and from time to time we can suspend enrollment or even order the discontinuation of the trial if risks outweigh benefits. And combined with this, we are also performing some on-site inspection. Usually it's unannounced inspections at trial site, whether healthcare facilities, authorized research centers, or sponsor premise. And just to verify the compliance with safety and right protection for participants most of the time, and also the accuracy and integrity of trial data. And if I just may jump in relation with what had been said, if we want to share or be agree about how we can just register and then rely on data register from other sites or clinical trial or coming from other clinical trial, there is maybe a word that is very important, and that's the reason why inspection is maybe important. It's reliance. So if we want to, it's one thing to give the results of other, but the matter will always be, can we trust this? And as a regulatory, this is very important. And we are currently on almost a daily basis working on reliance through inspection and, for example, but also through common guidelines. And maybe if I have a few minutes again, I can also say that we are not working as a petit village isolé, pour ceux qui connaissent, for those who know Asterix, but we are part of the EU regulatory network, and we are really deeply embedded to this regulatory network at various levels, and because we are as EU members, we are part of EMA committees such as the CAT, the CHMP, the PETCO, the Scientific Advices Working Party, the Methodologic Working Parties, and also the innovation networks. And in this network, there is a lot of ongoing initiative, dedicated to what we have been talking for this morning, and to try to enter to the specificity of rare disease topics. And I won't go into detail into that, but the last one was, and it has been a webinar, and I think it was in March, about the use of platform approaches in non-clinical and clinical domains. So Europe and France is working on this subject. And maybe just to end with the clinical trial, I am also part of the ACT EU initiative. Just a little bit, it's not specific for rare disease, but I mean, it's important for me to mention it. ACT EU means accelerating clinical trial. It's an initiative with three bodies on board: European Commission, EMA, and HMA, Heads of Medicine Agencies, also all people like me in each state member. And we are committed to work and to have the clinical research smarter, better, faster. Maybe you have heard about this. I won't go into detail, but maybe you can go to the website of the ACT-EU. But I think there is also a momentum on this side also at the European level. And the last thing, because it's also linked to what has been said, I have the chance to be the co-chair of a stakeholder platform advisory group of this ACT-EU, meaning working with industrial, academic sponsors, non-commercial sponsors, commercial sponsors, patient. And just to say that the patient, and I am co-chairing this stakeholder platform at the EU level with François Huyez, and maybe some of you know François Huyez because he's a representative for Eurodis, the rare disease patient association at the level of the EU, but he's also a French guy. So I think that as an NSM, we try to make it from a daily basis to a more general approach with the EU approach.
Thank you, Professor Pogam. So France has the highest number of clinical trials in the EU, which is a mark of attractivity in a global environment which is still difficult in between the United States and China. And we see now how China is surging in terms of number of clinical trials. So I guess there's if we keep that ranking in Europe, at least it means that there are some attractivity elements for France within Europe. Can you tell us about the type of adaptations, if any, adjustments or specific views that the agency might have on clinical trials for ultra-rare disease, gene editing or gene therapy methods?
Thank you for this question. First of all, I would say that in NSM we recognize that innovation saves lives. So it means that what we will be doing, our role is to accelerate access to breakthrough therapies, whether for rare disease, and we will go on further a little while, but also for emerging public health threat, for example. So we know that science is running faster and faster, and one of our aim or goal, I don't know, is just to evolve, and at least we are committed to evolving with the science, so and to keep the pace, which is not always easy, but we are trying. So when I come back to the clinical trial in rare disease, I would say that probably the ANSM is as an offer that would say is multifaceted offer that is so to be able to support or to assist I would say the ecosystem of rare disease. And because this is multifaceted aspects, then we have something that is attractive, I think, for those clinical trials. You know that we have this support for scientific advice, which is given through the GIO, the Guichet Innovation Orientation. I will go this maybe into detail. And then we have also like in France, yes, I don't remember who speaker said that rare disease is a history in France for a long time. And I think that rare disease is also an history for French regulatory system also. And we have dedicated for a very long time, a dedicated unit to rare disease, meaning that considering clinical trial, we have extensive experience with small patient population. And right now we are going with extremely small population with the N of 1. We have a dedicated expertise in early clinical development for first-in-human studies. We have also a strong, I think, engagement with academic teams. More recently, we have launched a fast-track design for early phase 1 or 1 and 2 for unmet needs, and showing our commitment for this to be able to streamline and to go faster when it's possible. And finally, but I already told you, I think we are in a good coordination with the EU regulatory pathway that helps also to be in a good place to support and to assist the clinical research in the field of rare disease. And this is not a new thing in the INSM. So maybe I can stop there, and then I can go further for other detail of what we are doing.
Thank you. Dr. De Vries, maybe you want to tell us about the role of the CAT and your approach to registration of products from gene medicine to rare disease and approval. So we don't hear you anymore. We used to hear you very well.
That's odd. You don't hear me?
Yeah, now we hear you.
Ah,
Thank
perfect.
you. Thank you.
You scared me for a bit. Okay. So thank you very much, first of all, for inviting me. And I wish I would have been able to join you. I can see the panel very well, but the audience, of course, I cannot really see. But thank you for having me. So I work at the Dutch Medicines Evaluation Board as a senior clinical assessor. So from that perspective, I assess the clinical parts of registration dossiers for marketing authorization. And I'm also a member of the Committee for Advanced Therapies, where we do the scientific assessment of ATMPs for the whole of Europe. So as you might know, ATMPs are per definition eligible to the centralized pathway. So they have to be assessed centrally for the whole of Europe at once. That work is being done by the national competent authority. So we kind of divide the workload. One or two countries do the in-depth assessment of the product, and then we discuss at the European level, either in the CHMP or at the CAT when it's about ATMPs. So the CAT is a multidisciplinary committee. We have quality, non-clinical and clinical experts on board from all member states. And we also have patient representatives on board and physician representatives with us. So besides the marketing authorization part, we also provide scientific advice, was also referred to previously at the central level. So I will also take this opportunity to really encourage you to come for scientific advice, either to the national agency in the country where you're based or at EMA, because I think, as was said before, regulators are also here to help you. And we really like to engage at a very early stage of development in discussion with you to discuss plans. Something that I think is often maybe misunderstood is that regulation is reactive and not proactive. So when, I mean, you are the ones that are at the forefront of innovation, right? You are the experts, you have the plans and the ideas. So I would really encourage you to come to us and share these plans and ideas, and then we can reflect on it. And I think this applies to all different areas, but especially to gene editing, because this is, of course, very innovative and we don't have a lot of experience also yet from the regulatory perspective. So we see quite some scientific advices at the moment, but of course, there are not many products approved. So yeah, and with regard to your question about gene editing, I mean, in the EU, we have the ATMP regulation, and the ATMP regulation has anchored in it the risk-based approach for ATMPs, which means that you can justify the data requirements for your product based on the indication, based on the setting, based on the individual risks of this product. And I think actually this risk-based approach provides a whole lot of flexibility. But it will need to come from you as developers to argue and to substantiate why you think you can justify things in a certain way and which data should be presented. So I think this is really something really helpful, I think, this ATMP regulation. And with respect to gene editing, I think in essence, I mean, it's not much different. By law, all the medicinal products have the same requirements on a high level. However, we have, as I said, the risk-based approach for ATMPs, but also differences, different marketing authorization types. So we've got the full marketing authorization, but also a conditional marketing authorization and a marketing authorization under exceptional circumstances. So prerequisite for all marketing authorizations in the EU is that the benefit risk should be positive. But if the data are less comprehensive than what you would usually require for a full MA, there's definitely options, right? And especially for rare diseases, we have approved products down to, I think, 11 patients in the dossier, in the clinical dossier. So we also have experience with that. And it's not necessarily a showstopper, not at all, I would say. And it all depends on the extent of the effect that you've shown. Basically, I mean, if the effect is extremely large, then only a few patients would likely suffice, and then you would have post-marketing safety characterization to help you out on, and help us out and help everybody out on that part, because of course it's different to establish a safety profile based on 10 patients. So yeah, I think I'll keep it at that, and then we'll talk later on, I think, more about the NS1. Frederic, is that okay?
That's fine, thank you. Yeah, if we come, I was going to come to the N of 1 question, because after all, many of these drugs, you can imagine that many of these drugs are not made to be approved, and the idea of transforming a product into a procedure and reimbursing the procedure rather than the product somehow means the same thing. I mean, these are not drugs that are deemed to be approved. There will never be the number of patients either, necessary to justify approval. How did you engage the FDA on such aspects, Winston?
Definitely not a me, but a we. And also I think it's, I'll give you an insight into the context of this current administration, this current FDA. There's a lot happening that is a bit crazy. There's also a lot happening that those same minds that are turning towards deregulatory, like the same folks who said we shouldn't require vaccines are saying, why do we need all of these regulations for this? And if we see that baby KJ is now growing and he is, at 99th percentile, up from 5th percentile, and tolerating a full protein diet, right? So I think that context is important to say that we actually are in a very unique time of trying different things, and they have expressed openness just for dialogue. I think that's actually one of the most refreshing things, that they've said, we think that this is one of the cornerstones of success within this administration, that we would like to encourage more of this. How can we encourage more of this? There's been a lot of behind-the-scenes work by Tim and by Julia Vitarello, who was mentioned, on plausible mechanisms to push it to where it is. We're simply saying that a product-based approval might still not reach the fullest extent of the N of that really small tail of N of 1 patients in need, and you need a well-defined separate pathway there. And they were like, okay, we understand that this is not, you're just asking for more because you want more. It's that you're trying to solve a specific problem.
Yeah, Catherine, from a regulatory agency really acts in the perspective of registration. And here we're talking about drugs that may never need registration. What do you think?
Yes, usually clinical trials, and I should say more commercial sponsors clinical trials are really designed to give the data that are needed to go to the very classical pathway to the authorization. And at this time, with N of 1, it's probably that this won't be possible. So this is the first thing. So then the second point is, what do we want to ask? Is, I'm just reflecting about your quite inspiring and convincing idea of center, and going to procedure instead of products registration. But then the matter is how it works globally at a, let's say at least at a national level, how it works for the payers and the reimbursers. Okay, this is non-profit, but they need money to work and to be able to work and to continue discovery, and then how it works. And then there is my second point to say that is that there is like say an elephant in the room, which are at least in the EU and at least in France, it's the HTA body, Health Technology Assessment Bodies. Because this is for France, when you want reimbursement, you can have your approval, and then you have the reimbursement. So, to be honest, I don't know how we can manage this. I like the idea of what you propose, but it won't. We will still with your model, at least in the French way, the healthcare is funded, which is mostly funded by all of us, and it's not private payers. We have to find the way it works, because at the end, I'm right, it should be sustainable. And to be honest, at this time, I don't know. I mean, I think we need a lot of more brains to think about how it could work in France. But I think your idea is inspiring.
But I think in terms of communication, maybe presenting clinical trials as surgical procedures rather than clinical trials would help our decision makers, our lawmakers, better understand what we're talking about, and maybe be more amenable to reimbursement at that stage than if we say, you know, we want you to reimburse what is research.
There's a lot to think about it. Yes, could be, but what we want also is, because N of 1 is very nice, but I think maybe it was one of your slides or someone else. So, but the point is, we need to get some information. We need to move on knowledge. We need to build on knowledge, and this is also important. So if we say, so we need assessment of what is done. We need follow-up. We need long-term follow-up, and we need to build on this, because it will be a continuous circle. So for France, I don't know how it could work, but I think we should maybe, there's some people in the room that I see, we can just try to figure out how it could work, the let's say global system, going to discovery, utilization, avoiding compassionate use. I love the fact that you said that compassionate use is not sustainable. I do agree. We have a recent report from CNAM showing the cost for France of compassionate. So I do agree we have to think about it, because this is going to be the future for some of the patients. So we think about it. I do not have the answer right now, as you understood.
And Dr. De Vries, at the EMA level, so what's the position on N of 1, which after all might never be, never come from for approval?
Well, I mean, to start with that, I hope it will in the end. I hope we can design a system where it will. Because I think if you talk about like a true NS1, then you have a medicine that is produced without any prior knowledge, with a unique manufacturing process for one single patient. That's like the truest NS1 that you can have. But I think we are moving more towards a situation where we are building up a lot of prior knowledge. So we are going in the direction where we might have a consistent manufacturing process, but with patient-specific targets. And if I understood correctly, for clinical trial application in the EU, you can actually, within one CTA, target, for example, multiple mutations on the same gene causing the same disease, you can put that in one trial. So if I understood correctly from a legal perspective, there's absolutely no objection on studying personalized gene editing in the EU in a clinical trial. For an INN, I think that's in contrast. I hope I say that right. In contrast to an IND in the US, you can have multiple chemical entities under a single INN. So you could actually say, okay, we have a systematic, and I think that's also one of the important words, a systematic approach. And the systematic can be either the manufacturing process or the primary endpoint, or well, whatever you can think about, and actually do that study. And I think there's probably a lot of logistical challenges when you would want to file for a marketing authorization for such a product. And I hope also with the new pharmaceutical legislation, which is underway in Europe, I think we've got an agreed text, but it will take a while to really think things through, or maybe not even agreed. But anyway, we're in the process, and it will take a while. But there are some contexts there, some concepts there. For example, the platforms that are introduced in that legislation. So I hope actually it will come to a point where we can make that actually a feasible system to give marketing authorizations for such products, because as was said also, it is very important to build the knowledge. I don't think there's a centralized European oversight of surgical centers in Europe. So, I mean, you would want to have that, right? We have to work together. We have to share data, share knowledge, build that knowledge base to a point where we can trust what we have. And at the moment now, you can use prior knowledge actually in the EU. There's absolutely no objection to using it. It has to be in the same dossier, and I understand from a logistic perspective that might be difficult, but it is possible at the moment as well. So I think there again it comes to the proactive versus reactive attitude, where I think it would be very helpful if people just came to us with plans, so that we can reflect on it together, instead of people saying, oh, it's not possible in Europe. And then we say, oh, but I don't think that's true. I think there is a lot actually possible in Europe. So that would help. And one last thing is that the CAT is now working on a Q&A document for gene editing, because we've got the investigational ATMP guideline that has been released last year. And I think in essence, that's also applicable to gene editing, but there are, of course, specific topics that are not addressed in that guideline, and we want to provide our view on it. So that's work in progress, and hopefully by the end of this year that will also be out.
Thank you. I saw you reacting, Catherine. Yeah.
No, I just wanted to reply, not to reply, but just to say about the new pharma legislation. There's topics. So this new law is supposed to enter in force at the end of the year, and there is various implementation acts to take. But there is a few things that will start immediately at the end of the year. I mean, by the law, you need to wait for 20 days, but nothing. And one is the sandboxes, regulatory sandboxes, and we could imagine that actually we don't know what is regulatory sandboxes. What we know is something that is nowhere, or that cannot fit with what is ongoing. It's not so easy because, as Emily just said, we are much more flexible than maybe people can think about it. But then a place where we can decide to advance according to a specific frame, which is not, there is no right place to put it at the moment, and to move on with this under surveillance and monitoring, and this is in accordance with our main goal, which is safety of the patients. So innovation and safety for the patients. So I think that there is also some momentum also European level to give access, to find some way to move on and to find the answer. And as you said, ASO are not in the CAT meeting because it's considered as biological or chemical drugs, and it's under the CHMP. Yes.
Okay.
But I would say that what is one of the most important thing is that, and I would say also for Guichet Innovation in France, come to us as early as possible, and just put everything on the table, what you would like, what is the idea, what could be done, and there's a lot of things that can be done. And I think that sometimes there could be some issue with the time, but nevertheless, I think that we are able to provide regulatory assessment. We can provide scientific assessment. We can so we can help and support once again this kind of clinical research.
Thank you. So. Just looking at time, yeah. So before we move to the audience, Winston, there was one point raised by Emily saying, well, we would need some. Europe has no surveillance on surgical centers, and it's true that there are surgical centers where you wouldn't want to undergo a surgery, others where you would be very happy to undergo a surgery. So how do you address that?
Yeah, so I think there's a couple of levels of this, right? There's ones that set or the precedents of like the Transplant Society. They actually have a really powerful global network of this type of expert. It's not a health authority mandating this, but actually they self-regulate and they try to preserve their field. And I think that's an example of where organizations like the N of 1 Collaborative, we're trying, let's self-organize of the practitioners and actually let's enforce ourselves so that we don't have bad outcomes jeopardize the whole field. The other thing is that I think it is valuable to provide some level of autonomy for patients to go get second opinions and maybe go to different centers and such. And it's not so much like everything has to be homogenous, right? But there could be different preferences in terms of the experience of a physician or the investigator, the experience of the site doing it, or even they might think of risk in a different way, right? In a surgeon, when you get a second opinion, sometimes a surgeon is more skilled and they might say yeah, I think this is a valuable approach. Here's the risks of doing it. And you just are truly informed in the consent, right? And so I think there's a value of engaging the patients more directly in that choice versus having a paternalistic, oh, these are good centers and bad centers and such. Yeah.
Thank you. Are there, yep, just.
I agree that in France we can be very paternalistic. You're right about this, but no. But it's we have to say I am okay. We have to. But the point is that at this point, and I do agree because I'm coming from the field of the liver transplantation, actually, Doc. So I can understand very well what is going on in this field. But the point right now, I mean, what you are talking is about, let's say, in 5 or 10 years when there will be multiple centers. At this time, then patients just want to have access to one place where they could be, they could have access to a treatment. So this is not for tomorrow, but maybe for tomorrow after tomorrow. Yes, but this could be important to consider also. And I do agree that a lot of things has to be done with the patient voice and with the information and in the rare community association. Patient association, I think we have different approaches, and it has been very well described. I think it was by you about, okay, there is some case with very severe disease with something like life expectancy, which is very short, and so, okay, benefit risk is not an issue to discuss about it, but sometimes it's a little bit more complex. And so this is what is also the most interesting thing about working with rare disease patient association and rare disease clinician, physician, and scientists and researchers.
I think we might have time for one or two questions from the audience, if there's any. Yes. Hélène et Bana.
Thank you very much for this. I was wondering for N of 1 and the approval question. I wanted to share a perspective in the sense that I believe that it is very important that the agency have a say also whether the experimental treatment might actually be safe, even if it is for one person, a neutral outside reading, so that if ever in a year or two another patient with the same mutations that might actually happen might have some precedent to say that indeed this compound, even if it was designed for a single person, has been demonstrated in the other to be at least safe and ideally also provide a little benefit, let's say. So for the question about the approval. So that was a comment. And the second point, and which is more a question directed to you, INSM, French agency, and EU also, how do you guys work together to somehow ensure a common path, at least from the non-clinical perspective, among the different countries in terms of non-clinical requirements? As far as I'm understanding, some countries might require non-human primate studies, while others might, I'm exaggerating a bit, move from cells to human. And I was wondering here if you would discuss to harmonize between at least EU.
Do you want to give a part of the answer, Emily? Do you want to start on this one?
And I could comment after.
Well, I think it's maybe more for Catherine, because I mean, from the marketing authorization perspective, we are harmonized. So that's not a question. I think it's more for the clinical trial approval.
Clinical trial approval? First in human? Yes.
And clinical trial approval.
Yes. So as it is true that is probably where there could be some variation in the way each agency in each member state could consider what they need to give an answer about if they approve or not, about do they have enough data about safety, about and to go on and to approve. So it is true. What I can say from the French perspective, at least, is that we try first of all, and it depends on what will happen with the product. But because if we know that this is a product designed to go, then next step will be marketing authorization. Then we try to rely on what will be needed for the marketing authorization. Then when we are in a case of N of 1 and we don't talk again about what are the issues about marketing authorization for this kind of drug, then usually there is a discussion about what is. I think it is the right place for having a very nice dialogue with the team and just trying to find out if we, at the end, are convinced that what we have on the dossier is enough to say, okay, to add the assessment of the benefit risk. So there is a part of less harmonized, but on the other side, this is true. And also amongst our assessors, someone says, okay, usually the head of the agency, they are very much aware, they are prone for innovation and everything. And then when we go to the one assessors, and that has to say yes, no, then it's another matter. But the fact is, for this kind of dossiers, that is what we have in the French INSM, is a unit. There is 4 people dedicated to rare disease and 4 people dedicated to early access. And they are just each week just talking about the dossier together, just to consider what they think. So I mean that also the way we are working is also a way to make a decision about what is provided, then dialogue with the applicants and to find the right way. And finally, we have also, I took, for example, the example of the webinar about non-clinical and clinical expectation that has been, I think it was an initiative for the EU Innovation Network with HMA and EMA. This kind of webinar, which is performed at the EU level, is also very inspiring for us, because there is Q&A, there is, okay, this is not white or black, for sure. But then we move forward, because also things are changing very fast.
Thank you. I think for time constraints, we will stop here. I want to thank all the participants for this very open and encouraging discussion, and thank the audience for your attention. Thank you very much. Thank you, Emily.
Thank you. Thank you for having me. Enjoy the rest of the day.
We will move to the last sessions. It's the roundtable. It's more a reflection on economic sustainability on our systems in genetic disorders. Tac, tac.
So good afternoon. Actually, we are going to continue, I would say, the same conversation, but with an economic angle. For me, it sounds a little bit strange to discuss economy, everything but an economist. Anyway, I will try to listen to these key people. So I don't think there is a need to further introduce Tim and Winston. I think you are here and you provided us already with many interesting thoughts that will be further developed. But I have to introduce Vincent. Vincent Genet is the Deputy CEO from AFM Teleton, and Bernard Cely. Thanks for being with us. He is also a Deputy Director. He's a Deputy Director of CEPS, which means in French, Comité économique des produits de santé. So this is for our US colleagues, a very important body, because they are negotiating the price of drugs with industry or anyone that is producing a new drug. So they are key people for our story. So as you know, for the four of you, we have a few questions to address. So I would like you to discuss 3 points. I will now mention. First, how to fund preclinical research and clinical trials for genomic medicine, of course, for rare and ultra-rare diseases. So what is the respective contribution of the public funds, of charities, of industry, of any possible source? You partially addressed that question already, but I think it's really good to discuss it further on the US side, but also on the French side. Second question is how economically wise, how to develop these bodies or entities for non-profit production of drugs, editors, sales, mix of them for genomic medicine of rare and ultra-rare diseases. And the third question, which is more for you, but everyone has to contribute, is on which criteria prices for such medicine, whatever they come from, industry or not, should be based. Is it value-based prices or cost-based prices, or what should we do in the future, knowing what is the present situation and the very high prices of these genomic medicines that have been developed up to now. So let's start with basically the first question. May I ask our US colleagues to start first and to give us your views on how to fund this preclinical research, as well as then the entities to produce the products we need for our patients.
This is a really important question. What we're finding is that the will to fund it from the patient community side is immense, and the will to fund it using more traditional venture capital methods to raise those funds doesn't exist because the business model doesn't exist. And of course, we would love to take advantage of any avenue that is in front of us, but funding based off of what patient communities can generate is not a sustainable model. And there are also justice and equity issues to overcommitting to that type of approach. I actually find really fascinating the idea that if we see health as a public good, if we see that enabling the profit-generating engine to create drugs using economic incentives is succeeding in many ways. There are many pharma companies that are doing quite well. This tax model that Frederick introduced at least us to today, asserting a tax on the industry to then feed back to a public good, I think that that actually sounds like maybe one of the most sane approaches that I could imagine.
Can I interrupt you and just to ask you, do you think this is feasible, politically speaking?
I would ask
in
how
the US as well as in France.
In the US. I actually do think that there are. I think that there is a window. There are, of course, requirements that we ask of companies in the United States in order to gain access to our market, and they are willing to pay the tax. They are willing to pay for the FDA. The FDA is funded through a PDUFA Act that then brings funds in to then regulate themselves. Why would they be incentivized to do that? It seems that would be the absolute opposite thing, but of course, just to gain access to the market, we have declared that this is the necessary way to do it. One more example, and then I'll pass along to see if anyone has other ideas. We have started as a public good a program in the United States to gather data from failed clinical trials. By we, I actually mean Janet Woodcock, who was a prior FDA commissioner, who created an institute in the United States called the Critical Path Institute. And she was one of the main intellectual founders of it. She doesn't operate it. She had conflicts of interest running at the FDA. But behind the scenes, this is half funded by the government and half funded by industry, who then build a database for collecting trial outcomes from their peers, and then making it available to all to help reduce the burden and reduce risks for the next investment. So I think that there is some precedent for folks who are willing to pay back to continue to participate in a system that seems to be treating many of the companies pretty well.
I'll just be very brief. And I think for me, there is a differentiation between what is purely research and what is development. And I only appreciated that having gone through what development means later. And I think even like foundational biological research on a disease, on different therapeutic windows, on if you don't know whether correcting this gene will actually have the desired effect. To me, that ends up being more. It should be state supported in terms of in US and NIH, and as well as INSERM and stuff. I think that's very important basic research. The random walk of thousands of different labs is very important. I think development costs, like once you get to the point of non-clinical development and you need to figure out the well-structured set of assays to get into a program, I think there's a cause for that to fall under more. I think Francois he said really well, like, I want to pay money, I want to support money to get program to patients, not to publish a paper, right? And then there's also the idea of, can this go under that surgical model where it is a part of the process that eventually reaches the patient? So can we start using it as like a part of the procedure, like a preclinical evaluation or preoperative evaluation type of thing? So.
Vincent.
First of all, it's a kind of a challenge to finish the day and to try to find some new ideas to inject into the debate. And my contribution as a start would certainly be to have all of you thinking about the words that have been used during the day. First of all, when I see the title of the roundtable, Toward a Sustainable Economic Model for Genetic Medicine, I think that there's one correction to be made first. There has to have an S after model, because we all agree that when it comes to genetic medicine, there is not a one-size-fits-all economic model that can be considered for all the cases, for all the therapeutic interventions, and for all the markets. That's one thing. The second thing is patients have been mentioned a lot during the day. And we talked about early access. I would like to stress the point that patients are impatient. So what the point should be for us is certainly not to have early access, but to have urgent access. And then access to innovation in a system where the words that are used are also very much indicative on the system that has been put into place, which is certainly not adapted to the rare disease and more specifically the ultra-rare disease, which is marketing authorization. And then the question is, what is the situation where there's no market? And this is really the question that was asked today. Then funding of research. I think that we are lucky. We are actually sitting in chairs in countries where I'm not sure the key issue is find some more funding for research. I think that definitely the key issue is to find a way to finance development.
Can I
I'm sure you
because then I disagree on that. That's why we don't have enough money for research altogether. Go ahead. We also need more money for development. That I agree.
That we need money for development. We need more money for research, and I was a bit provocative, but you know, this is part of the DNA of IFM as an organization to be a bit provocative as well. So financing the development, and then there has been a debate before on clinical trial. Should we talk about clinical trial? Should we talk about clinical intervention? Should we talk about clinical procedures? When it comes to N of 1, at the end of the day, the development step is the access to the treatment. So I think that on our perspective, the idea is coming back to the point that if there's no market to reach, there has to have an urgent access to secure to patients, and therefore there has to have a way to finance the development, which is access to this treatment. That's just an introduction on my side.
I have a specific question on the European side. I mean, some people are considering that perhaps what we need and that we do not have today at a European level to the development of these drugs is a kind of ARPA-H-like entity as you have in the US, which I think has been in many, of course, has been extremely successful in terms of vaccines, but not only in terms of vaccines. So perhaps, Vincent, would you react on that? Do you think that would be a good idea if, say, at the European Union level, we should have something like that may promote and ease the development of the new models of genomic medicine?
The provocative feedback I can share at that point is it's always an easy solution to say that if we are not capable of doing something at a nation level, we're going to do it at European level. I think that France has demonstrated its capacity to be a pioneer in, I would say, bringing light on the concept of rare disease. It has replicated at European and international level, and we can be proud of this. So my reaction would be, it's certainly not a bad idea, but let's start first in France and then expand to Europe.
So can I rephrase my question? Is what about dreaming of such an entity in France, developed by the public system, to have an entity that is founding, monitoring, developing, easing the trials for genomic medicine, and perhaps beyond, but at least for genomic medicine? I think this is more for you because sorry to be on you.
I'm not sure I get the question here. What is clear is that we have a lot of, I would say, tools already. Here again, I'm not sure that I fully understood to what extent this very inspiring perspective that you brought during the day regarding considering not a product but a procedure is actually offering actionable perspective in the field of non-genome editing modalities for ultra-rare disease. And I'm very much interested to listen to you on this.
So the question is, how do you think about procedures for non-gene editing? So I do have some thoughts on this, and I think the reason why I can soundly propose this idea of genetic medicine development as procedure is because of the scientific basis of these platforms. And I think the scientific basis, if you look at a CRISPR, even in the dead format, right, it's a protein in which the only change for a different target is a small piece of RNA that's actually tucked inside the functional protein. And now there's been data to show that when you go across many different targets, the safety and efficacy generally are not impacted. I think that's not the case for small molecule drugs, and it's actually a little bit borderline for ASOs. With there are sequence-specific, even biodistribution and stuff with ASOs. So I think that's where the proposal of the procedural framework is only enabled by the tools. Like if a surgeon, this tool's somehow bent a different way each time he used it, then that's not really a well-defined procedure, right? And so it's a subtlety, but I think we really have to make sure that N of 1 compassionate use as procedure is not generalizable. I at least personally, I don't think to all like repurposing experiments and other drugs. That's a separate question. Right? Did that help answer your was that the question for me?
Yes, that was the question. So, having to some extent a unique guichet, offering the possibility, to have a 360 view and process ease to secure urgent access, whatever I would say the prevalence of the diseases is certainly a dream that we can think about. So to your answer, Alain, the answer would be why not?
Yeah, I think we need the money at least to build the non-profit production of now, so of procedures of products. Obviously, today we are still on the side of products, but we need funds to do it. I mean, there could be private funds, there could be money coming from foundations, but I think we also need a little bit money from the state. You want Bernard to say something?
Just a little comment about your suggestion, Alain. I'm not sure right now in France we are really keen on creating new entities. I mean, we are more trying to simplify and to optimize the spending of the state. But that's just a small remark.
Don't kill me, please.
And maybe on the third question you were mentioning, Alain, which is should we price value or should we price cost? That's an interesting debate, especially when it comes to the fact that to demonstrate value, you have to have past cases. And you have to have a statistical demonstration that value is confirmed. So as a non-profit organization at the origin of a quite an innovative ecosystem, we are very supportive to complementary action between for-profit and non-for-profit organizations, and not to fall in the, I would say, easy and certainly narrow-minded way of looking at things, which is when it's for-profit, it's bad. When it's not for-profit, it's better. I think that, once again, every situation needs to be relying on, I would say, a very harmonious collaboration between for-profit and non-for-profit organization. What is clear, however, is that today we consider that we should exit from the one and only way of looking at price, which is based on value, for the sake of providing urgent access to treatment, and therefore certainly introduce the possibility to get reimbursed, based on cost plus perspective.
Yeah, actually, may I respond just quickly? You have a lot of thoughts on this, I'm sure. This is something I really do think the procedural model gives you an out to price it differently. And Tim, David, and I had a conversation with Medicare, Medicaid in the U.S. Right, the basically state-sponsored. The U.S. has private insurance, and they also have state insurance. And when we proposed this idea of what a procedural model is, they said, well, do you know that the CPT codes, the procedural codes that are used in surgeries and such, that's not a value-based pricing, right? It's actually a cost or cost-plus based pricing. And so if we were to adapt to that, we have to think about all of our unit effort, right? Preclinical screening, all of that. Like we have to have some unit price plus maybe a sustainability markup, right? Just because it's cost-plus pricing. I think that's actually a very interesting deviation that allows us to find what is the true cost of a genetic medicine, of going from end to end to like seeing the patient, evaluating them, building the model to at the end of the day making it and treating them. I think value-based pricing has gotten us to a point where the bar seems to keep on climbing, right? Of just like, oh, you priced it at 4 million? Well, I'll go at 4.5 million because I think I can eke out a bit more. We have better efficacy than you, and you priced it at this. And I think that the public is not really, it just makes us as drug developers feel very greedy, even though I know that there are good health economic justifications for years of life and, ICU costs not sustained and so on and so forth. The nuance, though, is that if you do genetic surgery, quote unquote, for common diseases, then you could think of it as like a backdoor to undercut the pricing of those things. And so there was a discussion that we had of very specifically outlining if that there was already a BLA, a marketing authorization for some product, you cannot apply the genetic editing tools as like a generic version or something like that. So, there's a lot of things to think about, but I'm supportive of procedural cost-based pricing to see where we can get and then optimizing that. And I think drug companies can still have their value-based pricing, just it's a separate category.
Yeah, thank you. So Bernard, I think this is really now your time to tell us how we should proceed in France.
Well, I don't know if I have a solution, but first of all, what I can share is how we do in France. So we really put a price based on the value of the medicines or the medical devices. And we have a system which is quite comfortable for us because we intervene at the very end of a process. We put the price, but before there is the HIS, the Higher Authority for Health, which gives us a report on the product or on the medicines, and which tell us if this product really brings something from a therapeutical perspective. And this is very convenient for us, and we really based our economic assessment on this purely medical assessment. The question right now in the current state, we take into consideration the cost when we are asked to increase the price. So, for instance, if a company, if a laboratory has a major increase in its costs, it can come before us and shows how much the increase is and ask for an increase in the prices based on the cost increase. That's the only situation when we can take cost into consideration. For ATMPs, for instance, taking into consideration the cost, I'm not sure it's actually an easy solution, because defining a cost, actually it's very complicated. It's probably even more complicated than tariffing on the value. Economists have been working on costs since decades in order to regulate monopolies from the 60s, and putting a proper cost on genetic medicines, it's probably not the easy way. There's another thing I would like to share is performance contracts, and we are trying to work on this in France, because obviously when you have a medicine with only one injection in order to cure a disease, you can understand that this product is going to be more expensive than other drugs. But obviously for the payers, which we are, the risk is obviously very important. So how can we minimize this risk? And that's an answer, at least, is performance contract. Here in France, considering the budgetary situation, which is obviously very complicated, we prefer the performance cost based on we see first and then we pay, over, of course, the contrary. But right now, to be completely honest, we haven't put in place any of these contract right now. We are working on it. It's going to be done. The legislative frame has been modified in order to put this type of contract in place. But right now, we have not done it. No, I just wanted to say that based on the value, we have put a price on only one genetic therapy, one generic medicine. So that's not much. There's another one. Actually, the laboratory withdrew its product from the European market, and the third one is going to come in the coming weeks or months. So, not many examples.
But expensive examples.
Expensive examples, and also the price is also going up. So if we look at CAR T cells, it's ongoing.
But regarding the cost-based price, I mean, there is one example in Europe, strangely enough, not for rare disease, but for leukemia, where this is for the CAR T cells, as everybody knows more or less. In Barcelona, they have been able to develop their own CAR T cells production, and the price at which the product is paid by the Social Security in Spain is approximately 3 times less than the price of the CAR T cells coming from the big companies. So it shows that, in some extent, it is feasible. And again, I'm surprised that it didn't come from the field of rare diseases, but the one of cancer. But still, I think it's encouraging to see a price can be determined on cost, plus a margin. I think this is exactly what you mentioned earlier today. And why not to try to do it for the rare diseases? Would you like to comment? Or?
Yes, of course. If somebody, if a competitor enters the market with lower cost, then it can reduce the price, and that's.
That comes from the public system, not from the private system.
And obviously, we could look at situation where a public structure, in order to produce these drugs, could be looked at. I don't know if there are examples in the US.
If I may react also, because it has been said public versus private. Here again, I think that the key criteria to consider is non-for-profit versus for-profit, and for sure we all know that non-for-profit is not the exclusive territory of public sector, but there are some private players, non-for-profit, and as we are. So that's one element, and then I think that we also could consider to have some eligibility criteria to some extent consider going in a kind of sandbox perspective, not with regulatory perspective, with financing perspective. And when there is a clear demonstration that there is no market, there is no commercial viability, there's a true hope enabled by science, their ability to access to patient needing this solution. We cannot wait 12 or 18 months for disease lethal in 6 months. So we have to find ways also to adapt to this perspective that needs to secure urgent access to therapeutic interventions for people living with ultra-rare diseases.
Obviously, I agree with you. I mean, my separation between public and private was a bit too quick. I fully agree. But this is not typically the industry that is going to provide cost-based prices. That's for sure. I think we have time maybe for a few questions. Please don't hesitate. Yes, there are two here.
Merci. Now, I would like to come back to Professor Tim Yu's proposal about the public good. Is it not the solution to qualify such a product as a public good? Why I am asking this question? Because I am very surprised by the fact that we could not be able to forge a cost-based price. For me, there are billion elements or product in the world today which have a cost priced, and I don't understand why it would not be possible for medicine to do that. Or I suspect that there are non-economic reasons for that, because it's very easy to allocate element of cost. I think that I was really impressed by your comment this morning about genetics, very complex for me, because I am an economist, but really, to be able to forge a price is far more simple than to go on non-code and so on, you know? So really, to fix a price is something very, very simple. You take cost allocated and you sum them up. So please, could you try to explain me why it is so difficult to find cost-based price? Excuse me.
That's for you, Bernard.
Well, thank you for this question, this easy question. Well, in fact, not that many products are cost-based, in fact, and it's not as easy as what you describe, because what do you take into consideration, for instance? When do you start your addition? And so there are a lot of questions that have to be taken into consideration when you do this type of situation. And then when the number of patients that you treat is increasing, then you decrease the price. How do you manage the price in the long run? I mean, you know, it's not that easy at the end of the day. But you may want to add something about that.
So I'll take the perspective on both sides, right? One is in a hospital setting now. How do we price a liver transplant? How do we price a bone marrow transplant? Like there's some cost associated with that, and there's some margin that the hospital takes, but there are set prices, and in some ways, because the health authorities or say this is how much we pay for things, then the end user has to optimize to how do they actually make money, or how do they make themselves financially whole doing it. So I think that is the example. I think you could really say, how do they do it today with codes and everything? It's tremendously complex, but it's doable. On the other end of things, on pharma, having, again, been in industry before, I see the insides of exactly what you're saying is when does the cost start and when does it stop, right? Do we fold in a fractional time of the CEO? What about the marketing teams, right? Because a booth at a conference is like $300,000, a really fancy booth, and all the people who are staffing it and marketing. So that in industry is a part of the cost that it all goes to the marketing authorization. But again, if there's no viable market, we should actually rethink. I mean, Jim Wilson has made the case that for a lot of these ultra rare therapies, the clinicians and the patients are the marketing arm. Like they will find the therapy versus you need someone and pay them, sales commissions and stuff that go into the cost and are hard to track. So that's just two perspectives from both sides of the world.
Thank you. You want to say?
No, exactly what you described. I mean, it's the situation, for instance, in France, in the current, in any drug or medical device, when we want to increase the price because the company had an increase in its costs, I mean, we don't take into consideration the salaries of the employees, for instance. Even if the salary is increased, we don't take into consideration. Otherwise, I mean, all laboratories would increase the salary of their employees and then ask the social security to pay. So that's just an example of how difficult it is to measure the cost of a product.
Yeah, I don't know. Yes, I had also a question. You explained that you were in a comfortable situation because you were at the end of the process. So for you, it was easy to determine the value of a product. And I was wondering, when we are in a rare disease product or N of 1 at the extreme one, when you get conditional approval based on maybe some kind of feeling that it might work, but you don't really know, and it's based on 10 patients. What's the value of this drug? Is it very high because potentially it will change the life of the patient? Or maybe it doesn't value anything because in the end, in 5 years, we'll discover that, well, it just doesn't work.
Well, that's why we have HIS, which does all the complicated analysis in order to tell us what the value, and then put a quote on the product, and based on this quote, then we will put a price. But at the CEPS level, we don't actually analyze this. We really depend on the analysis of the HIS, which is both scientific and independent. So that's very protective of the system.
May I throw something in that may be obvious? That to me, one of the problems that we're describing is a problem of scale. That there is simply insufficient scale there's insufficient data to be able to justify a price based off of whatever approach. It's difficult, maybe harder on the value side, because we know scientifically that we do not meet statistical rigor with any of our studies in any of the traditional sense. Yet one of the barriers to scaling is the current price, which is based off of the existing system, which is value-based, which greatly, actually, to my opinion, in my uneconomically trained opinion, is vastly elevated relative to the actual costs of, say, running a GLP toxicology study. To me, those seem to be running about 5 times higher than I could possibly imagine it actually costing, but that's just me complaining. I'm thinking of other ways that we can solve the scale problem that might also drive down costs in addition to the methods we're talking about. One is open sourcing some of the information that's generated around these platforms for this particular purpose. We have the ability at the NIH to infringe on patents for the good of public general research. And as a university academic, I can do the same, and as a nonprofit, many of us can too as well. But would that also be something allowable for one of the new public good entities working in this space to pull from those patent portfolios, IP portfolios generated by others in the industry, and give that back to the companies as well as to the public? That's actually the deal that we have worked out with Lilly. The information that we generate goes back to both parties. Would that also create more scale and then give you a better estimate of price, and then give us more flexibility to choose whichever value or cost-based approach you can now use with the data that's in front of you? That's a question.
Okay. I'm afraid we are not going to solve definitively the issue of prices and how to fund the development of what we urgently need, treatment for rare genetic diseases, given the fact that the science is there. But still, I think hopefully we have made one step forward. So thank you for the... you want to say something? Sorry.
Yeah, I cannot stop reacting because assurance maladie, social security has been mentioned in the discussion. And today, as an organization advocating for equitable access to care and also to cure, it's unlivable for us to consider today that because there is an economic model, some for-profit players will take the final step of development and secure access to treatments thanks to science on which funding came mostly from public incentive. And if you have a too rare disease, the science is there, the research is bringing it to a proof of concept stage, and then there's nothing to take the final step back, final step in charge, sorry, and secure equitable access. So at the end of the day, the situation today, and I think that politically in the debate, it's not put at the forefront where it should be, which is if you are suffering from a too rare disease with no commercial viability for-profit players, you do not have equitable access to solidarity through social security. And here, to us, this is obviously something that French people do have to live with. And as a patient organization, we cannot live with this.
We fully agree, but this is not only a French problem, unfortunately. It's everywhere, but I think we certainly agree with your comment. So I think we should stop here, and thank you for all four of you from this panel.
Oh, or is it a panel?
No, just give us some final about the day.
All right. I just found out that the final session, which I thought was going to be the three of us sitting in chairs, is actually us standing in front of a microphone giving what I now realize are unprepared comments, reflecting on the day. So maybe I'll just share a little bit of a stream of consciousness. First, it's very stimulating and frankly inspiring to be at this event because of the history of the contributions of French scientists, and some of whom are in this room, to the origins of this field, and also to the really impressive progress and implementation and tackling of many of the same issues that you've heard about today that the people in the United States are tackling. I think there's so much alignment on some of the key issues and a deep appreciation, which is heartening to hear as a scientist, that we're now thinking increasingly about non-science issues. That's heartening because you only think about the non-science issues when the science reaches a level of maturity that there are non-science issues to think about. Like, how are we going to get all of this medicine to as many patients as possible? How are we going to pay for it in a sustainable way? So, maybe I'll just say that it continues to feel like a sort of perfect storm moment where the maturation of science, the early but compelling clinical outcomes, the recognition from other stakeholders, the patient communities, the regulatory agencies, the groups, mostly the governments that will eventually have to pay for much of this, are coming together at a time when small differences in the course of actions that we take will end up having a very large difference, I think, in whether these treatments come, in 1 year or 2 years or 5 years or 10 years. And to patients, that is a huge difference. Obviously, can be a true life or death difference, the difference between a complete family or an incomplete family. So I think we all share this mission, and I'm very grateful to the organizers for organizing such a stimulating and in many ways uplifting to me event. So I know everybody's probably eager to not prolong the rest of our day, so I'll just hand it over to the next spontaneous remarks from Bana. Oh, sorry. Okay, Frederick. Okay, great. I thought it would be kind to just return the favor.
No, it's no. I just want to really thank the presenters and the audience. I think we've had at least I had the feeling that we had a unique day today. I mean, at a moment where we see a global geopolitical situation which is much fragmented, and where sometimes we hear that the interests here are much different. That we live in two different worlds. I mean, both in the U.S., in Europe, in China, wherever. Well, we see that people coming from coming from different horizons, but all scientists all share the same feeling and the same responsibilities, and converge on solutions that look much alike. So from that point of view, I mean, both the quality of the exchanges and I think we've had a quality of science which was really outstanding, but the quality of the exchange on the rest of the issues, and the fact, as David said, that we all feel responsible as scientists, as patient, as members of patient organization, as regulators, as member of other body stakeholders. We all feel responsible for making sure that all these drugs and this extraordinary science reaches patients. So that was really comforting, and I really want to thank. It was a pleasure working with you all to prepare this meeting on the French side with Bana, with Marina. I want to thank also Barbara. I don't know where Barbara is, but somewhere in the audience, we've had this very constructive discussions with Emily, with Romain. So I mean, it was both preparing and executing was very nice. Thank you to you all. Thank you.
So thank you very much. I am completely unprepared because I have forgotten that I have a closing remark. But so what has inspired me this meeting is starting from the first slide that I presented, that we need a bridge. Now we have a bridge between imaging and the hospital patient-driven, but today we have built up another bridge with your group, David Liu, to implement many other clinical trials for rare disease. I think that there is the wishes between the two sides to put together expertise, patient cohort, and the many clinical trials we are developing with the help of all this disruptive treatment built up by the group of Anna Rita in collaboration with us. So I think it is a great momentum. And it underlines, I think, the necessity that despite the difficulties of different government, I don't think to anyone, and the difficulty situation in financial support of research, clinical trial, the only way to be win in this situation is to bring bridge and collaboration and put enormous quantity of work together in the major benefit of patient. And I think this symposium is reflecting these wishes from both sides. Thank you very much.
I'm going to be very short. I want to thank everybody for being here, for the very productive discussions. I want to thank our American colleagues for having done this big trip and to spend time with us. But I also want to thank, all the people, Ambroise, all the people who are working with him up there, who made this day seem seamless, which is not always easy. Barbara and Corinne for organizing everything for here. And of course, my colleagues, as Frédéric said, with whom it has been a real pleasure to organize this day, be it with Emily and her group or all the French side. So thank you very much and have a nice evening.